This morning we moved to deoxygenation and co-stresses, more specifically to understanding, monitoring, and mitigating deoxygenation in the context of multiple stresses. My name is Grant Pitcher, and I'm convening this session together with Min Dai, who is sitting in China. I hope you've been able to join us. It's my pleasure to introduce our keynote speaker, who's speaking from the University of Gothenburg, Sam DuPont. Most of his research has focused on revealing the mechanisms behind species and ecosystem responses to environmental change, with the intention of developing theory for large-scale predictions. He's done most of his work on acidification and warming, but he has moved to the impacts now under multiple stresses. And here he's been specifically involved in experimental design to capture responses to change. And he has also participated in the SCORE working group on best practices of multiple drivers in marine research. And so his talk today is what we really need to address multiple ocean stresses. Thank you, Sam.
Thank you very much. And I'm really sad not being with all of you in person. So I'm going to have to do that from my office because I have a lot of teaching these days. So it was just not possible to do. But I will do my very best, and hopefully we can have a nice little discussion about these really important issues on how to address multiple stressors. And most of what I'm going to present today is summarized in this really nice document that has been led by Kirsten, who is with you in the conference and published on the IOC UNESCO webpage. So it's a really nice document that is aimed at policymakers, but can be really useful for scientists as we define there a lot of the concepts I'm going to talk about. So basically, we are all interested in the idea that the world is changing as a consequence of human activities. And I'm sure you've been discussing a lot about all these issues already, but you're focusing here on low oxygen. That's only one of the many stressors that organisms are experiencing in the natural world. And we know today that one of the main drivers is overfishing, then you have pollution, of course, habitat destruction. And on top of that, we also have all changes, and many of those are related to accumulation of carbon dioxide into the atmosphere. And we have a long list of changes from global warming, more and more catastrophic events, the ice is melting, is rising, the concentration of oxygen is decreasing, you have salinity change, and you have acidification. And all these things, so global and local stressors, are working together. As it's not complicated enough, these things are also depending on where you are on the planet. And the priority stressors that you have to have really depends on where you are. It's not only different geographically, it's also different in time. So you have, of course, fluctuation at different time scales, but you also have the fact that the list of stressors that are the main driver in location somewhere will be different in the future than today. And it's really important to understand this, to identify this combination of different stressors, because identify what are the main is the only one to develop solutions. So every location have a set of solutions that you need to implement. So at this stage, we know that we have to deal with a lot of stress, complicated, that every location has a different combination of stressors, and also that this combination will be different with time. So the first thing you really need to consider if you really want to address these issues is cataloging the stressor. And you have a monitoring component to that. And unfortunately, you have more and more interest in monitoring these different stressors at different times and space scales. The thing to really consider what is the relevant time and space scales to really be able to infer biological response. So you have all these things that you can measure, and really, if you want to identify the main issues, you need to do it at the right scale. Different talk. I'm not going to go too much in detail about this, but I really want to emphasize the importance of identified priorities. So you're in the room because you're interested in deoxygenation, low oxygen. That's a major driver for sure, but it's not necessarily the main driver everywhere. And that's really, if you work on a certain ecosystem or associated to a certain ecosystem services, that you really focus on what the main priorities are. So you might be facing a situation like this one, where stressors that are very important, it could be deoxygenation on the right, and I don't know, global warming on the left. Realize that if you face a situation like that, you need to address both stressors at the same time. If you just get rid of one, you're still dead because the other one will catch you. But in certain situations, you might face a situation like this. Threat, but one is more urgent than the other. So first, in that case, you focus on the important one locally, the adult tiger, and in the meantime, but less priority, you can work on the second one, the little cub on the left, and fortunately fix the problem before it becomes a bigger problem. But the importance here is that you really need to identify locally what main priorities, and that is, of course, monitoring temperature, salinity, oxygen, fishing, and so on and so on. But monitoring is not enough. Because if you really want to develop local solutions, you first need to understand the exposure, which is the level, for example, of temperature, of oxygen concentration, but you also need to understand how that will translate into biological effect at the species or ecosystem level. And to do that, you need to actually do your homework with the biology. So it's both a combination of monitoring and understanding the rules of biological impact, and that can be extremely challenging too. And finally, when you have identified your list of stressors, okay, in this location for that service that I'm interested to protect, here are the list of the different drivers, but then you have also to have a look at what kind of solutions are available, what kind of time scale you have to implement those. And sometimes the solutions are not there yet or not at the right time scale. So it's both, you have to think about these three aspects to really identify what are the main pieces: the level of exposure, the biological effect, and the range of solutions that you have at hand. And so you can also identify gaps in knowledge. Again, I cannot stress enough, it's different everywhere. So you really have to do it locally. For the effect size, so I said, like you monitor first, you have an idea, okay, the temperature will decrease by that level, the oxygen will decrease by that level, the pH will decrease by that level, and so then after that, you need to understand what are the potential consequences. And you know very well the Paracelsus saying that the dose make the poison, but it's not only about the dose, because for drivers, global drivers like temperature, the same dose will have different consequences in different places. And just to give you an example in the field of acidification, but you can find that in every other global driver, you can have a situation like this, where you have two populations of the same species, in that case, a copepod, and you expose them to the same level of acidification of pH or PCO2. One population is doing fine, the one on the right, and another one is doing terribly bad, so respond really negatively. In that case, there was a study on fecundity of the copepods. So that's showing that, if you have the same dose in different places, you're going to have different response, sometimes even within the same species. So same species, different responses. And the reason why you have that is because of the local aspect to that, the local adaptation. A stress should always be defined as relatively to the condition that a species or an ecosystem is experiencing today. So what is a stress for an organism will not be a stress for another one, or even, as I said, for the copepod, different population of the same species. If you expose a polar species to 15 degrees, that's going to be a major stress. If you expose a species to 15 degrees, that's fine. So stress is relative. And that's really important to understand the concept, because when you read literature, you have all these different concepts presented, stressors, drivers, stressors. But biologically speaking, that does not have the same consequences. So for example, oxygen is a driver, but stressors under certain condition, when you deviate from the condition to which the organism or the ecosystem has been adapted to. So really, I think it's sometimes understanding the definitions are key, especially when you write a paper, because it has consequences for the understanding and the conceptualization of the issue. So again, just a personal example of how a driver can become a stressor. It's an example with pH here, where we tested like a wide range of pH on sea urchin larvae in that case. And what we could see is that you were within the natural range of variability, so the gray coverage of the figure, then you see an effect. Their response, so it's a driver, so it's driving a biological response. So if you decrease the pH, they grow a little bit slower because they need more energy to basically compensate for the little bit more challenging condition, but they are fine. This is plasticity. But then you cross this physiological tipping point, and if you go lower than that, then the driver becomes a stressor, and you start to have really strong negative response in that case. In growth, but also increased mortality and abnormality. So that also explained this local adaptation, the reason why two populations of copepods are responding differently. The one on the left, they were collected in an open ocean area where in that case, the pH is very constant. So they are adapted to really narrow range of pH. So if you expose them to a lower pH, it's really negatively impacted. When the white, they were collected in the coastal environment where you have a lot of fluctuation for the parameter, and then they are locally adapted to that tested scenario. The same tested scenario doesn't have a negative effect. So in that case, it's not a stressor, it's a driver. Population on the right, driver, population on the left, stressor. So that's another way of seeing that. It's like the one on the left was collected in a fluctuating environment. So the tested scenario is within the natural range of variability. So it's not a stress. So far, we know that we have a lot of stressors, that they differ wherever you are. And if you really want to understand which of these potential stressors are actually to monitor and understand the present range of variability, and then you have to understand the biological sensitivities, and you can infer sensitivity based on local adaptation to this variability that I was talking about a little bit earlier. You also have to see what kind of options you have. So if I want to reduce carbon dioxide, what can I do? If I want to maximize ecosystems, what can I do? What is already available, and so on and so on. So at the end is the combination of all of those that will define locally what is the different combination of stressors that you should care about. But then the question is like, okay, no, you have an idea. You're in a place where you know your system, it's been monitored, you have an understanding of local adaptation, how organism and ecosystem will respond. But it's complicated to actually bridge the chemistry to the biology. So what science do we really need? And the real question, and that's an easy one, is like if you have all these different drivers and stressor A, B, C that can be temperature and so on, what's happening when you combine them? And a general rule would be that the more driver, the more stress, more negative the impact, and that makes sense. I think if you just keep increasing the pressure you're putting on your ecosystem, the more negatively it's going to be for it. There are some nice studies, for example, this one by Shin and Collins, showing that if you increase the number of environmental drivers, you have a negative effect on growth of phytoplankton. But sometimes you want to go further than that. You really want to understand what's going to happen if I decrease my pH by that, I decrease my oxygen concentration by that. And then when you read the literature about that, you have a lot of diverse terminology, like people talk about additivity, synergism, synergistic effect, antagonistic effect. And in many ways, it's confusing. And it's often presented like that. If you have a stressor A and a stressor B, you just add the effect of the driver mathematically, and then you have the effect when you combine them. Synergistic is when the effect is higher than what you expect just from additivity, and antagonistic is the other way around, is less than what you expect when you combine them. But I want to convince you that actually that doesn't help anything, because if you have this kind of thinking about additivity, you won't be able to understand really what's happening. So that's where I want to drive you through now, is showing that actually this concept of additivity, synergism, they don't work at the effect level, they work at the stressor level, making things a little bit more complicated. So just to illustrate that, let's imagine like a silly example where we use two different drivers that are the same one. So temperature and temperature. So you do an experiment that you take your favorite organism, you measure their growth, and then you increase the temperature by 2 degrees and you measure what you have, and you see that you have a 10% increase in growth. Then you increase by 4 degrees, and what you see is that you have a 15% increase in growth in that case. So the question is like, what's happening when I combine these two stressors or drivers? What's happening if I increase my temperature by 6 degrees? Can I predict that based on what I observed with my two different temperatures? Can I just assume that the effect will be 25 degrees, for example, in that case, 25%, just an increase of 25%. And you can imagine that temperature is additive to itself. So that's why I use this very nice temperature is an additive stressor to itself. So the question is like, oh, I changed the numbers there. So if you have a 10% increase at 2 degrees, if you have a 4% increase at 25, what will be the impact of an increase in 6 degrees? Would it be an additive effect, which you expect if it's an additive stressor like temperature with itself? So should it be 35, less than 35 and being an antagonistic, or more than 35 and being a synergistic effect, or it depends? So if you think about it, and you know a little bit about temperature or oxygen, you know that it depends. It depends because even if in theory temperature is additive to itself, the response is never linear. So you typically would have a curve like this. So if you increase the temperature, you first have an increase in performance, and you reach a threshold, and then you start to go into the physiological problem, metabolic depression, and you have a decrease in performance. So that explains why you can't really predict from the 2 and the 4 what's going to happen if you're exposed to 6, because it will depend on where you are on the curve. If your reference is on this curve, then you're going to have a linear effect because it's a linear relationship. So if you know A and you know B, you know A plus B, you know A plus B for the effect, and you have a pure, so it's an additive driver, you have an additive effect. But what can happen is that you're a little bit higher on the curve. And if you have the effect of A and the effect of B, it's not going to be able to predict what's the effect of A mathematically. So you need to know how to navigate on the performance curve. And you will have the impression that you have an antagonistic effect, while actually the two drivers are still additive, temperature plus temperature. This, where you actually on the other side of the curve, and then instead of having, you still have an additive driver, temperature and temperature, but have a synergistic effect in that case, having the effect size. So basically, temperature is additive to itself, but we see at the effect side depends on where you are on the performance curve. So the message here is, if you what's happening with multiple drivers, you can apply the same kind of thinking. They are additive if they work on the same physiological processes or the same ecological processes, but that doesn't mean that it's additive at the effect level. It's additive at the stressor level. And if you really want to understand what's going to happen at the effect level, you need to resolve the performance curve. So understanding yet how drivers work together, it's not as simple as just making a multi-stressor experiment and then use an additive model or try to understand what kind, if they are additive or not at the stressor level. You really need to do the extra work and understand the mechanisms. Fortunately, we know quite a lot already about the mechanisms, which is great. So that allows us to actually make some assumptions about what's happening. A lot of time in the literature of multiple stressors, what is suggested is to do these complex multi-stressor experiments. If you have temperature and oxygen, you do a cross design, you have a certain number of treatments, but that can become very, very complicated and more importantly, very difficult, if not impossible to interpret. So we'll try to show you that there's an alternative to this. What you really need to do if you really want to understand and model multiple stressors is you need to understand what's happening inside your organism or your ecosystem. They are exposed already to individual drivers, because if you understand the mechanisms, then you can infer about what's happening when you combine them, and you can understand if they are potentially additive or not. And basically, I can illustrate that with a silly example. So if you have 3 different drivers here, you can have beer, drugs or medicine, and the test. So with a little bit of experience, you understand very well what's happening if you drink a couple of beer and a glass of wine. these two different drivers are actually additive because they have the same mode of action. So what's happening is that when you drink booze, it's increasing the level of alcohol in your blood. And then if you drink 2 beers and a glass of wine, you can just quantify how much alcohol is it, and that's going to explain what impact is going to have on you physiologically. So in that case, these two drivers are additive and they don't have any interaction. Now, if you combine like alcohol, like a glass of wine and some medicines, it's a little bit more complicated because they don't have the same mode of action. And then you can start to have complex interaction. And that's the situation where it can get a little bit more complicated. So additivity doesn't mean that you have a mathematical addition at the effect level. It just means that you don't have interaction between the different drivers, physiologically or ecologically speaking. And in that case, you can use what is called a dose addition model, where you can just add the different contribution of each driver to the overall stress of the organism, and using a nonlinear model following the performance curve, you can make projections. So that's the easiest case. Then synergism and antagonistic is defined most of the time as when you have interactions between the drivers. So it's whatever is not additive. Let's go through a couple of examples of those. So let's take an example where you have a situation where you have the same mode of action and no interaction. So you have basically 4 possibilities. You can have interactions between the drivers and then can have the same or not mode of action. So one example could be you want to study the impact on the location of global warming and overfishing that you identified through monitoring and so on as the 2 main drivers. And let's imagine a situation where temperature kills 50% of the fish and fishing is killing also 50% of the fish. So how can you predict what's going to happen when you combine them? Basically, just to show you, it's a situation where you have no interaction and different mode of action. So fishing and temperature works on completely different things. So if you have a certain number of fish and the first stressor overfishing is killing half of them, then global warming is also killing half of them, but it will kill half of whatever is left. Right? So basically you have to remove the interaction between the two. So it's a very simple additive system, non-additive, where you have no interaction and different mode of action. So you're basically going to have 75% of the fish left. So that's basically the ideal simple situation. The 2 stressors that are doing completely different things. So it's not that you have it's a purely additive effect and they basically have the same mode of action. But unfortunately, it's rarely like this. So I explained to you like the case of temperature and temperature, but that could be also temperature and pH. They have basically similar mode of action, a different mode of action, and basically no interaction. So that was another case. But then you can have the situation, like this, where you have interactions and different mode of action. And a good example is if you combine pH, for example, and a toxicant. And we tested that, and just to show you that in that case, it can become really hard to predict. So we tested the impact of pH on the brittle starch growth, and what you could see is that if you decrease the pH, they grow a little bit slower, like 10% effect. We exposed them to a toxicant, triclosan, it was decreasing the growth by 30%. If you combine both, they were basically all dying within a few days. So you had a much, much stronger effect because there was an interaction at the chemical and at the physiological level. So in that case, making projections become extremely complex. They have to have a deep understanding of both drivers at the physiological or ecological level to really understand what's going to happen. So overall, let's say that even if things are additive, things are not linear, so it's complicated to do that. And you need really to have a deep mechanistic understanding of the mode of action at whatever level you work, physiology or ecology, if you really want to resolve multiple stressors. And I realized that you arrive at this stage of the talk, you thought like you can think like it's very complex. So how can I do that? And there are some resources out there that can help you to design a proper multi-stressor experiment if you really want to have the tools to model in the future. So there was this paper published by the SCORE working group a few years ago. So that's a good starting point, defining very well the concept and so on. But another thing you have to understand, you won't be able to resolve multiple stressors and model them using just multi-stressor experiment. You really need to combine different approaches, multiple stressor experiment being one of them, because a multiple stressor will be sex to interpret, and without a deep understanding of the performance curve and the mechanistic aspect of every individual drivers, you won't be able to interpret these multiple stressor experiments. So a good way of doing that, and that's my personal suggestion, is that you start by listing your monitoring work. You go there and you see what is the intensity for this. Then you spend some time working at a single stressor level. So you can check in the literature, do I understand what oxygen is doing to my organism? Can I understand what temperature is doing? If not, you just do it. You need to resolve the performance curve. So basically how the single stressor response is for your organism or your. After that, you can incorporate all that using modeling. So through my mechanistic understanding, I assume that there is no additive effect, so I can model it using a dose addition model. Then I understand the shape of my performance curve, and that's not trivial, but that's possible. And then you do your multiple stressor experiment, and you compare the projections from your model with what you observe, and you correct if necessary. That can be done in the lab. That can also be done in the field. And just to show you that it's possible, it's something that we've done recently with some colleagues. So we worked in Svalbard using protein fertilization assays. And what we did is studied the combined effect of 3 different stressors, so temperature, salinity, and pH. First, we worked at a single stressor that resolved the performance curve. Then we performed also like multi-stressor experiment using a scenario approach. And then we realized that if we use this concentration addition model in that case, we can actually predict all the different combinations. So in that case, we have a good understanding of the mechanisms. We know that these 3 stressors are additive, and we can develop the strategy to make projections for all the different possible combinations. So it works. It can work at least. But important message is that sure, it's complicated. It requires a little bit more than just doing a multiple stressor experiment. You need a mechanistic understanding. You need to resolve performance curves and so on. And it's not going to be done with one experiment or one method. You really need to develop a strategy. And you can use different resources, as I said. So you have the paper from the SCORE working group. They're going to be, I just advertise here, a training that we will organize later this year in Monaco. So that's co-funded by the IEA and the Ocean Acidification International Coordination Center and the Foundation Prince Albert II of Monaco. So that's going to be a 2-week course where we will help the participant to design their own strategy and also how to analyze and model this kind of data. So if you're interested, keep an eye on the news stream of the Ocean Acidification website. It's going to be advertised, I suspect, within a few weeks. And another good starting point, again, is to have a look at the document that was released on the UNESCO website and led by Kirsten and others on multiple ocean stressors. And I'm going to stop here. And so we have a little time if you have questions or comments. Thank you so much for your attention and giving me the opportunity to talk to you today.
Thank you, Sam, for a very good talk. Do we have questions? None? Well, Sam, thank you very much for an excellent presentation and for sticking to time. So our next speaker is Marco Fussi. And he will be talking about the global, including natural oxygen and pH fluctuations and predictability in the sensitivity of benthic marine organisms to climate change. Thank you, Marco.
Yeah, thank you. Thank you very much. Good morning to everyone. And I'm going to speak a little bit about how complex it is to predict the thermal tolerance and the response of marine ectotherms to climate change. So we know that climate change and global warming is overall increasing the increase the temperature, increase acidity and CO2, and therefore is globally also reducing the capacity of retain oxygen in the seawater. These global trends, are not always affecting at the same way the coastal ecosystem. You can see from these figures from a work from Fernando Lima that the climate change is heterogeneously impacting the coastline of our continents in a different way. Some part of coast is warming even at a higher rate. Some part of the coast is even will be subjected to cooling down temperature. Assess the response of marine ectotherms to these changes. Many indicators, many models has been proposed. One of the most successful was the Markelim proposed the Markelim project, where they studied the response of intertidal community to climate change by forming an indicator that was considering the species thermal index that was calculated, as you see here, from the distributions of the animal, and 50% of the they take the median temperature of the occurrence. So you see here two orange and the green one, and they calculated the species temperature index for both of them. So one more cold adapted, one more warm adapted. This indicator has been proved quite useful and has been adopted also for UK marine strategies, and allowed scientists to understand the different response of animals and plant communities. This indicator has been also proved useful to understand the impact of extreme event like marine heat waves on how the community will respond to this event during war and during summer. However, we saw from the presentation of Dr. Jomi yesterday, and I hope I can be able to show this video. It's not working. Okay, so this is a video that we took in the Red Sea, and you see that this is a seagrass meadows that has been colonized also from coral reef. And I want to show you is the bubbling of the seagrasses during the daylight. During this was done around 12 in the day, and the bubbling is extreme high, and the productions of oxygen is extreme, determining extreme supersaturations of this coastal environment. And the temperature of the water was higher above of the highest thermal critical temperature of most of the animals living there, like for example, swimming crab or fishes, but all the animal was there hunting, grazing, and do all the biological functions. So I want just to show this video that in coastal and highly productive ecosystem, the scenario is quite complex, because the bubbling, the production of oxygen, the photosynthesis is not just affecting oxygen, but is removing CO2, is changing pH, and create a sort of multi-environment where the animals live and the plants live. And what is that in this case, temperature and oxygen synchronous, so higher temperature increase lead to high production of oxygen, because the photosynthesis of primary producers that was coral reef and was algal community boost the photosynthesis and a huge amount of oxygen in the water. And this was not just particular features of the ecosystem we studied in the Red Sea, but you see from Venice Lagoon, from the Balearic Island, and from several ecosystems in the Red Sea, we always measure these higher fluctuations in temperature and in temperature and in oxygen release with differential predictability as shown by the wavelet analysis on the right of the graph. And what I want to show also is that each ecosystem that we study has a sort of particular fluctuation, space of fluctuation of temperature and oxygen. For example, the first one, Venice Lagoon, you see there is shape of oxygen and temperature that is similar to the one in the Red Sea, that has this similar shape. So each habitat has his own fluctuation on temperature and oxygen. So by integrating in the equation to determine the tolerance of the animal, the oxygen, we saw that oxygen supersaturation, when we included the oxygen supersaturation, the thermal tolerance of the animal changed. And some animals, here we test, all respond with a positive increase of thermal tolerance from 3 degrees from the swimming crabs, Talamita creonata, to 1 degree of the mussels or the fish. But this is true also for other drivers. For example, these studies just published by Peter and colleagues shows that the same species of mussel that live 2 different habitats, the coastal habitat that is represented by a black line and the lagoon habitat shows a different, totally different variability and predictability of the pH fluctuations. And as a response, and this is, you can see from the right side graph, lead to a different gene expressions of the animals. So fluctuation matters and the predictability of fluctuation matters on determine the gene expressions of the animals. The predictability of the thermal fluctuations is also important, has been proved very important in the traits, in determining the traits of diatoms. So the blue dot are the diatoms that have been exposed to regular and predictable fluctuations of temperature, and they have generally a higher positive effect in the traits of the species compared to the dots that instead has a complete different effect. So regular fluctuations has a different effect on the traits of the animals compared to those diatoms that has been exposed to irregular fluctuation and not predictable fluctuations. We did an experiment in Santabs where we manipulated the oxygen variability and the thermal exposures of a swimming crab and the mussels. I report here just the result from the swimming crabs that are very similar to the one we got also for the mussels. And when the species has been exposed to ecologically relevant environmental fluctuation, they have a better metabolic performance, and you see they are stress than animal that has been exposed to increased temperature but stable oxygen conditions. The right graph that the accumulation of lactate is higher in the species that has been exposed to normal fluctuation compared to the one exposed to the ecologically relevant fluctuation of oxygen. So if the single experiment on single driver design, as also Sam DuPont showed before, a response curve of a species, if we add multiple drivers, and especially if we design experiments that can depict the ecologically relevant fluctuation of those drivers in the local environment where the animal lives, we can have a shift or the shape in this performance. And this is quite meaningful because it means that we can refine the model that predict the impact of climate change on the marine species. So our project at JNCC that is just starting now is we want to, first of all, review the data that are existing on the thermal tolerance of the animal. For example, there is a database that is quite exhaustive, the GlobeTherm, that collect a huge range of species critical temperature, and I think it contains of species from terrestrial and marine environment. Do systematic review to understand the effect of the multiple driver on the performance of the animals and understand, as Sam said, the synergistic or additive effect or antagonist effect of the multiple driver. Integrate with this data with the fine scale habitat. You see before that each habitat has a particular signatures of, for example, temperature and oxygen that can be possible retrieve only if a fine scale monitoring is applied to that environment. And this is very important because habitat, for example, seagrasses, have different signatures of temperature and oxygen of a coral reef, and this can determine a different shape of the performance of the animals. And then we would like to include in the equation the variations of the drivers that we are studying, since we know now, and there is an increasing number of studies that shows how fluctuation can change the response of the animal to the ocean drivers. And also integrate data from the species sensitivity. For example, Marlin, the Marlin database has, although it's a preliminary, but it has a quite exhaustive part for several species, and it has the data for the sensitivity to different driver, from environmental driver to pollutant and human threat like pollutant and so on. So we would like to include this kind. We are working to include 5 different big data set. It will be complicated, but we're working on it to create a new indicator that can accommodate the new concept that is emerging from a more experiment, a more precise monitoring from the marine environment. It can be applied not just to the marine, the intertidal community as for the marlin, but to all the marine community, to aim for a better indicator for an accurate species sensitivity assessment to climate change. And this is my last slide, and I want to thank you for your attention.
Thank you, Marco. Do you have questions?
Thank you, Martin, for the talk. I'm surprised to see the nice connection between the temperature and the oxygen side. Was just to have your opinion about the impact of the respiration of the community. Seagrass, you also have this epiphyte, and you have the sediment, and also the regulation by the air-sea exchange, which is the reverse, because when you have increase in temperature, you will have a decrease of the dissolution of oxygen in the water. So you should have some.
Yes.
Yeah. So how does it combine? Because it's strange to see this very nice.
There is at least in the study site we measure the overall production of oxygen during the day is higher than the respiration of the entire ecosystem. Okay. In cases or some days, for example, where, for example, there's not a good irradiance, where the respiration is, then you don't have this phenomenon of supersaturation. This can be seen if I plot all the days at our level. So this is a level of variability that you can capture only if we do hourly monitoring with the new dots and everything. And it can change according with the patch of meadows that you can study. Absolutely.
And RC exchange, is it affected by?
Sorry?
RC exchange with the atmosphere.
We don't measure the exchange of the atmosphere. But
what you show, it's model results or it's observation? This
is the result of the logging that we did.
Okay.
You refer to these. Sorry, I just to these figures.
The previous one.
This
yeah,
one.
That's one. Yeah.
Yeah, but this is what we measure. We measure, for example, also on the freshwater habitats in the dikes, in agricultural ecosystem or freshwater lakes. So it is a consistent pattern. But for example, a higher latitude, as I said, if there is a cloudy day, the efficiency of the photosynthesis cannot stand the respiration, and sometimes even during the day you have, this is just 3 days, and I take as an example.
Okay, thank you.
Denise? Sorry.
That's really interesting. My question is to your results about the fluctuating oxygen environment improving thermal protection. Could you just talk about what ranges of oxygen you were seeing in that data set, and if you think that that would continue to be true if oxygen levels were also stressful under part of that oxygen variability range?
Yeah, so we need to, when we did the hyperoxia, so we showed that exposure not for a long time, for a limited time, increased the level of thermal tolerance. It is shown that long exposures, week, for example, or 5 days, of course, will start a process of oxidation. But our experiment was short in the time to reproduce as much as possible the level of hyperoxia that the animal experiences in the field. So animals usually have 10 hours, 12 hours, 8 hours super exposure to supersaturations, but then they go down to the hypoxia during the day. So we are now assessing which is the onset of exposure to hyperoxia when the oxidative stress kick in and then can be dangerous for the animals. But this is the difference. So yes, this is why it's important to measure this fluctuation at ecologically relevant site, because a long experiment on the hyperoxia would not be ecologically relevant for the species.
Short question for Denise.
Yeah, I just had a question about the what's going on in the supersaturation phase. Do you think it's something as simple as when oxygen is that high, they can essentially repay the oxygen debt and repair quicker and using less energy for ventilation and activities like that? Or do you think there's some novel physiological thing going on at supersaturation?
What we measure definitely is they are quicker in recover from hypoxic driven stress, like lactate, for example. In the experiment, last experiment that I show, for example, we measure also the hemocyanin, for example, and we saw that animals that experience fluctuation and phase of supersaturation, they have more significant amount of hemocyanin in the hemolymph compared to the being exposed only to 100% of saturations. So we are looking in this mechanism process, and I guess there is some, I would say that it is a quicker recovery of the night hypoxia-induced metabolites.
Okay, I think we need to keep it there. And our next speech talk is by Anna Marie, and it's on the global impacts of warming and deoxygenation of marine species of the 21st century. Thank you.
Hello, yes. Let's see, yeah, thank you. Good morning, everyone. Thank you for having me here. So my name is Anna Moret, and I'm working together with Bern and our colleagues at the University of British Columbia, Taylor Clark and William Cheung, on the effects of both oxygen and temperature of marine species. And I think I will be the first one at the conference looking into the future. So I will give you an idea of projections forward into this century. So let's first get a quick picture of what kind of changes we are talking about. So here from Earth system model projections, sea surface temperature are increasing. Are projected to decrease. And we've seen this figure many times already, about 2% loss of the global oxygen inventory until now, since the 60s. And also for temperature at the surface, there's already been a 0.5 degree warming in a global average. So within that framework, we asked the following questions, or these are the results I will show you today. So 3 points: how does this warming and this deoxygenation affect global habitat viability? And how, and this relates very well to Sam Dupont's talk, does species vulnerability matter in this warming and deoxygenation, so species-specific impacts. And then last, I will show you results on habitat volume for the species of interest in our study. However, how do we go from warming and deoxygenation to a quantified impact on species? And for this, we use this aerobic growth index, which has already nicely been introduced by William Cheung. And it's very simply put ratio between oxygen supply, which is environmental oxygen, and oxygen demand, which is species specific. So why this index? There are other indices around. We chose AGI because it's really widely applicable to about 10 times more species than earlier developed indices. It's in good agreement with these earlier developed indices. And it's excellent way to integrate both the effects of temperature and oxygen in one index and assess habitat viability across very many species. So a little bit more detail on the aerobic growth index. I show here in blue the environmental state, so oxygen supply and temperature. And in orange, you have species-specific components of this equation, which is relatively simple. This is weight and length related. For instance, you have preferred temperature of the species and an oxygen threshold, which we have already heard quite a lot about. So with this tool in hand, we can calculate AGI for about 1,000 species, but to really get to impact, we need some kind of threshold, right? So this threshold we define by the 10th percentile of all the AGI values in a species distribution. And this method has been applied before and seems to work really well. So at this 10th percentile and variability in space and in time, we say below this threshold, this critical threshold of AGI, we do not have AGI values which are able to sustain a viable population of that particular species. So keep this critical value in mind for the remainder of the talk as we will use it in impact. So now I've shown you how to calculate AGI and how we define our impact using AGI. We have both oxygen and temperature coming in, but we need some information on oxygen and temperature towards the future, right? So for this, we use a CMIP6 model output from 6 models such that we can also assess model uncertainty. We involve a low emission and high emission scenario in these models such that we have also uncertainty assessed, and we bias and drift correct these models for our study. So one more slide before I show you some results. All our results are presented for 3 species groups: epipelagic, mesopelagic, and demersal species. And the species within these groups, it's in total 48 species, have been chosen such that the diversity in depth, climatic zone, and body size is really represented. So at the bottom of this slide, you can see, you can get a sense of the diversity of the habitats of these species. So now we're ready for some results. So I promised to show you, first of all, changes in habitat viability. And this is really just a change in this AGI index, which I promised you combines temperature and oxygen really well to assess habitat viability. I compared it between the end and the beginning of the century. So you see that for these 3 different species groups, AGI generally decreases, and more so at the surface, so about 10% decrease. So reduced habitat viability, more so at the surface. And by plotting it this way, I do now, showing here the medians, by the way, I can split this up in drivers of oxygen and temperature. So let's start with oxygen. So here you can see oxygen is relatively uncertain here in the equatorial regions, I should note, but oxygen is less important at the surface for these total changes. So you see here only 1% median contribution to the changes in AGI out of 10% total. While at depth, it is a more important contributor to the general decrease in AGI that we observe. And for temperature, this picture is really reversed. It explains most of the changes at the surface and less so at depth. And that's really quite intuitive, as this temperature, the warming, has penetrated well into the surface layer and less so at depth. So these pictures are entirely species independent. It's general decrease in habitat viability. And in order to go to species specific impacts, we need to have an idea of the initial state of the species environment. So what kind of temperature and oxygen does it have around itself? Thresholds the species has. So I just want to show you an example of one species I picked from our 48. It's the Atlantic tuna. And this Atlantic tuna, it lives in this region. I'm afraid that the actual habitat was colored here, but it has dropped out of the PDF. I'm sorry for that. But its habitat is about here. And this species, if I plot in a probability density function here, all the AGI values in that species distribution, I get this PDF. And you can already see that this PDF is really quite skewed towards lower AGI values. And I can tell you that's true for almost all species. So it gives you a sense of species vulnerability, right? Because this represents a relatively large volume in that species habitat with AGIs close to this critical threshold. But it's still okay. And by definition here, you have this critical threshold at the 10th percentile, like I explained to you. So if we then look at what this PDF looks like at 1.5 degrees of warming, you see it already shifts slightly to the left. And if I go forward in time in general, of course, depending on what kind of climate we will end up having in the future, here at 2 degrees, 3 degrees, 4 degrees of warming, you see this PDF slowly walking towards opening up a large volume of this species habitat to subcritical values, which would imply mobility in the case of this species, and it might become invasive in new areas. So I can apply this exact same process to all these 48 species to give you a picture of what kind of habitat volume losses. So here are all 48 species relative to the original habitat volume, what kind of percentage losses they are projected to experience when the models reach 1.5 degrees of warming. So for each of the species, you have a little whisker here, and you see that they're relative to the original habitat, they lose less than 1% at 1.5 degrees of warming. Although individual species really show a different pattern. And this species here is the Atlantic tuna, which I just highlighted. I want to also tell you that within these whiskers falls also the threshold uncertainty, the scenario uncertainty, and the model uncertainty. So we've really tried to combine very many sources of uncertainty in these whiskers. So if you show the same plot for 3 degrees, I'll just go back and forth one more time. You see this entire plot moving downward, right? So at higher levels of warming and deoxygenation, you would have higher relative losses of volume, which hopefully is a motivation to meet the 1.5 degree Paris goal agreements. So this picture, it makes it really fair to compare across species because it's expressed as a percentage. But if you're maybe interested in volumetric losses, as maybe it's more interesting for fisheries, this picture looks quite different. So I show you once more for what the cubic kilometers, millions of cubic kilometers of loss of habitat would look like for these different species. And if you put them all along the same axis, you really see that the epipelagic species have the largest volumetric losses. So to appreciate this picture a bit more, I just want to highlight 3 keystone species. So these are generally considered in the literature to be central to their ecosystems. They're generally top predators, fall away in a specific location in a cubic kilometer. They will have drastic effects on that specific ecosystem there locally. Then still, I will be honest with you, millions of cubic kilometers is a bit hard to grasp. So I here express it in volumes of, still difficult to grasp, but maybe it hangs around a bit more. So these epipelagic species generally lose 15 volumes of Baltic seas in the median, the mesopelagic 5, and the demersal species about 1% of a Baltic sea habitat volume loss, according to these predictions at 3 degrees of warming. So with that, I will wrap up and show you some take-home messages. I introduced you to the aerobic growth index, which really nicely combines oxygen and temperature in one index and is really widely applicable. I've shown you that AGI generally reduces, so habitat viability reduces, and this is more driven by temperature at the surface and more by oxygen and depth. But that the impact of this reduction in habitat depends on species, on its thresholds, but also really on its initial state, as shown by these PDFs. But that for all species, contemporary habitats are compressed independent of warming, and that this warming level really matters for the amount of. Thank you for your attention.
Joanna, questions, Veronique?
Thank you, very nice talk. I was wondering how sensitive are your results to your choice of AGI critics?
Yes, so we've definitely assessed that. And if I remake this 1.5 degree plot with this whisker plot for all 48 species, it does shift a little bit, but it falls still within this whisker uncertainty. So if you choose a different, we choose really different critical values of AGI, so you can involve different amounts of time variability. So now we did monthly World Ocean Atlas time variability, but you can also do it at higher variability. Like we've seen, you can even get to supersaturation. If you involve all that, you get a wider PDF you take your 10th percentile from. But the values and the conclusions are really still holding. Yeah, thank you for that question.
Other questions?
Yes, hello, thank you. A very nice talk. I have a question. So in the keynote this morning, we learned that the same species with the same stressor reacts differently in different locations. And now you have the AGI and you don't do that anymore. So can you comment that maybe?
Yes, I had the same thought, of course. So this is just stressors from temperature and oxygen combined, and you zoom out at the global scale and you project into the future. So you work with immense uncertainties. That's why also there's quite a large whisker on each of these. And I would, my guess would be that one part of the community might fall in the top part of that whisker and the other one in a lower part of that. Not the whole community is on the median, but it's within that spread, their response. That will be my guess.
Thanks very much. I'm surprised that the deep ocean appears to be so stable from these model results. First, I think the models may underestimate the oxygenation there quite a bit, but also you showed oxygen is more important in the deep ocean, but I guess for each species, range of oxygen concentrations or partial, which may, I'm not sure how robust this finds. So each single species cannot move across an entire ocean basin, so they may be affected by changes that your AGI PDF might suggest. So do you have any estimate or any way to not to say the deep ocean is safe? That some people may get from your talk. I'm a little worried.
Yes, thank you. I think demersal species, especially on the last plot I showed, if you put it on the same axis as epipelagic species, they look like nothing is happening. But if you give them their own cubic kilometers of volumetric loss, you really see large losses. They generally have smaller habitat volumes, so that's why it's more fair to look at these pictures percentile. They lose quite significant percentages of their own habitats. So relative to their relatively small initial, it is significant. So then I would really refer to this picture. Yeah.
Okay, I think we should call it there. Thank you very much.
So
wonderful
much.
talk. Our next speaker Zirai Santana Falcon, and his title is Irreversible Loss in Marine Ecosystem Habitability After a Temperature Overshoot Informed by the Ecophysiological Demand.
Hello, everybody. Thanks for the opportunity of presenting this work in this conference and in this incredible place. I would like to show you an overview of the work that I'm developing with a number of good colleagues from other institutions. I'm from the Met de France in Toulouse. It's irreversible loss in marine ecosystem habitability after a temperature overshoot informed by the ecophysiological oxygen demand. This is a work that is much in line with the work that presents Anne-Marie. Well, the ocean has been undergoing a number of unprecedented changes, mainly due to global warming-induced alteration of temperature, oxygen, and pH, the so-called deadly trio. Because of these changes, the ocean has been warming, acidifying, and losing oxygen during recent decades, and will continue to do so during this century, according to greenhouse gases emission projection scenarios, as indicated in this already seen figure from the IPCC. The limiting marine life decline is caused by ocean warming, major changes in biodiversity, productivity, and global biochemical cycles. Therefore, it is key to track the persistence of these changes affecting marine ecosystems and to know whether they can be stopped and, if possible, reverted with mitigation plans and how the ocean will respond. With this, sorry, in the IPCC report, several studies have indicated that several phenomena linked with ocean warming and deoxygenation are mostly reversible at the surface, while it will take centuries to millennia to recover at depth. However, the reversibility of these stressors, temperature and oxygen, has been studied in isolation so far, and the combined effect to is poorly known. That's why we want to estimate the component effect that changes on temperature and oxygen will have to the ecosystem habitability after a temperature overshoot. For linking temperature and oxygen, we will use the metabolic index that gives insights of the habitability of a marine habitat. For a marine habitat to be habitable, the supply of metabolic demand. Following this, we can this metabolic index, like the one from Amaret, the AGI, by the ratio between the oxygen supply and the oxygen demand. Along with temperature and oxygen, metabolic index or Phi also depends on a specific species parameters, mainly the temperature dependence and the vulnerability of a species to hypoxia. And we have tried to compute the Phi for 72 different species compiled in this study of Deutsch et al. And this is the distribution of these two different parameters. Some key values of Phi that we have to have in mind is that in theory, the minimum requirements for survival of an organism, oxygen supply and demand, will be equal. That is when the metabolic index is 1. It's what we call the resting rate. However, the oxygen supply should be higher in order to allow organisms to carry out several key activities such as feeding, growth, and reproduction. In viable marine ecosystem, it has been shown or had been observed minimum values of Phi that range between 2 and 5. This is what we call the critical rate, the resting rate plus additional energy to accomplish these activities. And for temperature and oxygen, what we use instead of scenarios into the future is this highly idealized experiment developed within the CEDAR-MIP, which is an intercomparison project that aims to know how the Earth system, or to study how the Earth system will respond to different levels of overshoot, of a peak of CO2 and then a decline. In particular, the experiment that we use start at pre-industrial values of CO2, increases at 1% per year until the concentration reach 4 times initial conditions, and then decrease at the same pace until reaching back the initial condition, at which point it held constant for additional 60 years. So at the end, we have 340 years of monthly data for the first 1,000 meter depth from a suite of 7 Earth systems in this project and for 72 different species. And with all of these together, this is how the experiment looked like. I plot here in red the CO2 to have a point of what the experiment looked. And then for the surface, epipelagic and mesopelagic water, we have the multimodal global average, ocean temperature, oxygen. As we see in the surface, temperature and oxygen respond almost linearly to the increase of CO2. However, there is some lag between the peak of CO2 and the peak of oxygen and temperature that occurs several years later. As we get deeper into the epi and mesopelagic waters, this lag between the oxygen temperature and the CO2 increases the multimodal standard deviation, the measure that we use for uncertainty. Phi, which is in green, is highly correlated with changes in temperature and oxygen at the surface and also on the epipelagic waters, but detaches, or the behavior of Phi detaches from the behavior of temperature and oxygen at the mesopelagic waters. In this layer, Phi reached the peak 8 years before the oxygen, after the oxygen, sorry, and 40 years about before the temperature. So the behavior of that detaches completely from its drivers. We can also see here some hysteresis, meaning that the end values of the experiment has not correspond to the initial values. And we indeed can see this hysteresis in this regional picture. We compute the difference between the last 30 years of the experiment, the after peak, and 30 years from the PI control that we call before peak. What this map shows is that the global ocean or the after peak global ocean will differ completely from the ocean at the beginning of the experiment, even though we have the same levels of CO2. The after peak global ocean will generally be warmer, especially at depth. We will see complex variability on the hysteresis of oxygen, including some increases of oxygen at some outwelling regions and depleting over vast areas of the Atlantic and especially the Southern Ocean. In the Phi, we will see a general loss of habitability by lowering the values of Phi, except at the deep waters of the mesopelagic waters of the North Pacific. We wanted to see what drives this hysteresis of Phi. So since oxygen in its solubility, that depends on the water masses properties, temperature and salinity, and of the biological activity and ocean circulation that can be approximated by the AOU, we just separated Phi, into these, the components of the solubility and the components of the AOU. We see that the solubility components drive negative hysteresis, decreasing the values of Phi across all levels and across mostly all regions. And we see that AO components induce more variability into the hysteresis of Phi. In the third panel, we have computed relative dominance between the solubility versus AO. And we see that in general, solubility components drive the majority of the changes, except at the mesopelagic waters of the deep of the North Pacific, in which the ocean circulation accumulates the oldest waters. Now that we have seen how the Phi is related with temperature and oxygen, and how or what the drivers are of his hysteresis, we can look at what this matter for the ocean's ecosystem. This is the main result of the work, is the global ocean volume, or the evolution of the global ocean volume at which Phi is below 1, so unfitted for life. Phi is between the 1 and the critical rate, which only some species could live, and the ocean volume at which Phi is above that critical rate in which life thrives. I computed here the relative or the fractional change with respect to the initial value for the full distribution, the 72 species and the 7 Earth system models. What we see, well, before that, I show you that these big numbers represent the percentage of each kind of water with respect to the total. And these smaller numbers, represent the hysteresis at the end of the experiment. What we see is that the deadly zone, that zone unfitted for life, will present positive hysteresis in the epipelagic, meaning that a bigger volume of water will be unfitted for life after the peak. This is also true for this critical zone. And here we can consider this as a transitional waters. Here only those species that can develop some strategies to cope with these low levels of oxygen may thrive in these waters. By contrast, the happy zone or the comfort zone, the area in which Phi is bigger than the critical Phi, will decrease after the experiment. This is mostly consistent across models. This indicates the peak of each of the models that we use, and indicates a 5% reduction of the habitable waters in the epipelagic, in the first 200 meters depth, where most of the biota live. Finally, we want to make an approximation of the time needed for the global temperature to recover from hysteresis. For that, it's just an approximation. We have taken the last 60 years of the experiment and projected forward in time. What we see is that the Phi will need, in the first 200 meters depth, especially, we may need more time to recover than temperature or oxygen alone. And the time will be in centuries. So getting back into the intro, we can now add a new line, this one, to the table extracted from the IPCC. We have seen that the combined effect of warming and deoxygenation will induce hysteresis that may take longer than the oxygen and warming to recover alone. Persistently affecting the habitability of marine ecosystems by expanding the volume of water that are unfitted for life at the expense of those water that can support it. Just the take-home messages, our data reveals a 3 to 5% reduction of the habitable volume after the strong overshoot. The solubility component of feed mainly drive is hysteresis, and this hysteresis will take longer or may take longer than the time needed for oxygen or temperature to recover. And just some things that we want to do, obviously after publishing the manuscript, compare with other physiological indexes, especially the AGI presented by Am. Testing, we have high frequency data to track compounds, and using this framework into a predictability framework to see if elements can be detected in advance. And many thanks for your attention.
Thank you, Jero. Question at the back. We'll get the microphone.
Thank you for a very nice talk.
Yes.
Is the slide before last or so? You have the table. Yeah, this one with medium confidence. So my question is, what as a framework to really go from medium to high confidence in order to predict with certainty the loss of habitat volume, aerobic habitat volume of species of the world ocean?
Yeah, in our case, I just put low for confidence because at the end we are only based on modeling. So I think that for increase the confidence, we will have to find different ways of measuring both the temperature and the oxygen in this case, not only based on modeling, but also based on observations and well, and another all the ways that we can measure.
Last question.
Yeah, thank you for a really interesting talk, and it's kind of the point of the hysteresis and kind of looking at recovery time of both of these stressors together is, I think, a really important perspective. Really, I kind of need to make a little bit of a comment, though, a word of caution on the metabolic index. I've looked at it for species in the Southern California Bight, and we know that the metabolic index are quite sensitive to the initial kind of laboratory conditions, and we know that very rarely do laboratory conditions the ecological variability we see in those systems. And I have a lot of fish that live under a metabolic index of 1. So I think we should be very cautious about how we interpret these results.
Yeah, for sure. Actually, we have tried to measure the uncertainty and also the uncertainty given from the species. And actually, it's interesting. I have it here. And what we see is that especially for the comfort zone, we see bigger uncertainty from the species, not from the model. So it means that we have to try to constrain the uncertainty of the species to get real good results in these evaluations.
Okay, thanks, Jero. Our next speaker is Karin Limburg, and she's going to be speaking on fish as mobile monitors of hypoxia exposure and incipient acidification in the Baltic Sea. Thank you, Karin.
Thank you. Can you hear me okay? Yep, good. Okay. Are we ready? Can you put it in? I like to see the whole screen and not the. It doesn't matter. It's okay. It's okay. I'll deal. Okay, thank you, folks. So this is going to be kind of different. This is looking at fish themselves, and something I've been doing. So my window on fish and fisheries has largely been through the lens that we're going to look at today. So I have a project right now that we have nicknamed Project Breathless, and I talked about it in a webinar about a few weeks ago for GONE. And we are looking at, you know, sort of in many ways, we're looking at the impact of low oxygen zones around in some very large ecosystems of the Baltic Sea and Lake Erie and the Gulf of Mexico. And so I won't talk about really that, but really I'll be building on some work that we did in a previous study and expanding now. So the focus, or the lens, if you will, that I look at is myself is looking at what is recorded, so to speak, in fish earstones. And these are, here's a, this is the, I think, but this is, you know, looking at inside of a salmon head. And you can see if you squint a couple of little white things in there. Those are the otoliths, or earstones of fish, and they're part of the hearing and balance in fishes. And what is interesting about them is that they time because they accumulate, they accrete a little bit every day. Particularly in young fish, you can see that growth. But indeed, I really have started to think of them as kind of the ultimate logbook for every single fish. Every fish tells its own story. It's carried along this way. So this is a cod otolith, the star of this particular presentation. They're pretty small. They're not the smallest otoliths, but you can take a cross section through an otolith and look at the whole exposure like this. And this is a picture all the way from birth to death, because, you know, they form their core in the beginning of life and they grow and accrete material throughout life until they're caught. So, and sort of superimposed on that is the chemistry that we can pull out of that too. So it's sort of like unlocking the flight recorder box and seeing what's in it. So one of the things that we explored in our project Hypoxilus was developing and validating proxies of hypoxia using manganese, primarily for all the biogeochemical reasons that you know and heard yesterday, but also in fish otoliths, very often there's physiological influence. And so to correct to some degree for physiology, we also look at, we developed a proxy that sort of works, helps a little bit, manganese by magnesium, because even though magnesium relates in seawater with salinity, it does not do that so in fish otoliths, so it's different. But so we can see that, so the low zones in blue, this is a synchrotron map of this otolith. We periods of elevated and low hypoxia is low, and these are episodes of seasonal hypoxia, summer hypoxia. And so the Baltic Sea is where this story is playing out. It's a place, I was interested that you gave your volumes in Baltic Sea volumes. It's a place that's really a poster child of hypoxia, of course. When we did our initial work looking at hypoxia exposure, the blue are the indexes of hypoxic volume in the Baltic Sea. And a completely different index, if you will, is the exposure, year by year exposure of fish, of these cod to hypoxia. So that's the duration during the year. It's one of our better indices. And I was shocked when I saw how well this. With Baltic Sea cod, there's this very strong contrast between what one might call the good old days. There was a period of extremely good conditions in the Baltic proper for cod. It's no longer there. Today they look like this. You saw some of these images before perhaps. And my colleague Yvette has calculated in her dissertation that between 1990 and 2019, a 7-year-old fish has lost 40 centimeters of growth of size. And for those of you who think in English, me, that's 1 1/3 feet. That's a lot of growth. And we can see in this graph where I've got parsed out the hypoxia exposure groups by quartile. So these are the least exposed, these are the most exposed. And what you see is by age 3, a 39% loss in length translates to 64% loss in weight. That's a biomass, that's your fish, that's your fisheries, right? Okay, so let's dial up to my new project. And now I just have to tell you that we have, we use, for a lot of our work, we use laser ablation mass spectrometry. It's 2 instruments that you hook together. The old one finally died. So by a miracle, we got a new one, and it's much more powerful. We can do more with it. So because of that, I started to monitor the element boron because Lisa Levin thought it was an interesting one to look. And it's much more sensitive. So we can see some interesting patterns in boron. And I analyzed some cod from 2000 for Yvette to help her out, enhance her data set. And so I found some interesting patterns. But then for this new project, I looked at fish that were collected in 2019 and 2020 and found a really marked decline in boron ratios. And I won't go into all of the details about this, but they've both been shown, both the boron calcium ratio and the boron isotopic ratios have been shown to correlate positively with pH and are used to reconstruct past pH environments in paleontology. So where we're going to focus, the data are going to be mostly from here, but also from these other sites. But the first thing I'm going to show you is from the southern Baltic area right there. So this is from 2000, and this is a trace where I take run a transect with the laser from the ventral edge through the core and out the dorsal side. And that's what we have here. And that's how the data are going to look. And you can see that there's some real variation in here, even though the numbers are really low. Contrast that to when I started analyzing fish from 2019, I thought something was wrong with the instrument, so I reran everything from 2000 and from 2019, and everything checked out. And so we sort of have these pancake-like traces if you put them on the same scale. So what's going on is the question, right? So I started reading up on boron, right? And I found that there are 2 forms that are pH dependent, but the one that is called borate is positively correlated. Oopsie, sorry about that. Is positively correlated over here. And borate is found in marine carbonates. And so because basically the bottom line here is that carbonate boron to calcium ratio should be positively correlated with seawater pH. So this pancake appearance suggests possibly looking at you know, a decline in pH. So I started looking for literature in the Baltic Sea, right? So it turns out there wasn't a whole lot of literature. In fact, I just found 2 reports last summer. One of them had come out just a week or so before I started looking for literature, and they don't really say a lot about it. What they say is a few take-home messages, but what they say is, for example, that the ecosystem effects of acidification are currently small. I contacted my own biogeochemical contacts. They were kind of surprised that I was getting concerned about this. They say, nevertheless, that we should be starting to monitor for it, right? And they also told me that the Baltic Sea was buffered by an increase in alkalinity coming out of the watersheds of the Baltic because of increases in precipitation. But then they also kind of said that this was probably going away. And in fact, if you look at the data in deeper parts of the Baltic, the acidity has increased rapidly, and acidification's impact is low, but it's projected to start having an effect. So what's that look like for our other cod otoliths? So I went back into my archives and I started pulling out otoliths and reran them with boron and to reconstruct a time series. And you'll also notice here that these are actually smaller than these. They're getting smaller and denser, which is in agreement actually with some experimental work in Norway. So this is what my time series looked like finally. And we see it sort of looks like a roller coaster. It sort of bumps up to this peak and then kind of comes down like this. Really, I don't understand it completely. But I knew that from other work that boron's positively correlated with salinity. And so maybe I was looking at a change with drops in salinity, right? So what I did was I studied it, I studied these patterns in relation to salinity. And in fact, salinity is increasing, if nothing else. It's at least as high as it ever was. And it's plenty high. But the boron to calcium ratios seem to be at an all-time low, at least in the data that I've got so far. And if you plot that against pH, we also see, with coloring the pH, we also see a decline in the pH. So this is just looking then at the period from 2001 onward, and we can see there's just been kind of a general decline. Now, you can also look inside the otoliths within these transects, and this is one from 2014 and 2019, and you can see patterns. So the brown is our hypoxia proxy, and the purple is our putative pH proxy, right? And we can see that in this one, there's kind of an inverse relationship. In this one, there's sort of more of a congruent relationship. So there's a mix, but what I did was I took just the dorsal side and I calculated regression, just computed regressions of these two variables on each other. So for this particular otolith, we see this is just all the data points on that transect for boron and for manganese and magnesium. And we can see this negative slope, which you'd expect if the organic matter decomp produces both acidification and hypoxia. And so calculating those slopes and parsing it out year by year, which is what you can do with these chronometric structures, then what we see like an ontogenetic effect, where you have a positive slope at younger ages, perhaps these, and we know these fish are being more coastal and they're in coastal nurseries. So maybe the alkalinity is buffering them there. And then as they get older and move into deeper waters, they're seeing more of that acidity, I think. And so what I think is happening is that you have these massive blooms of algae in the Baltic Sea nowadays, and that is exported as organic matter to deeper water. The microbes get to work on it. And then if you look in places like the Gotland Deep, you can see over time changes in both dissolved oxygen and pH. And the effect is also there in Bornholm Basin in the southern Baltic, but not as strong. But if you look at the entire, if you look at oxygen versus pH, you've got this almost classic relationship, and color it by temperature. It's cooler up here and more oxygen and higher pH, and it zips down. This is really a time series that we're looking at here as well. So just to summarize then, boron has gotten very low. It seems to be at an all-time low, despite the fact that the salinity is higher now. There's mostly an inverse relationship between boron and manganese, and that seems to increase with age. And the hypothesis is that, hypoxia is being produced by blooms. There's weak acids being generated too, and this is being picked up. That's what we think. Very much a work in progress. Stay tuned, unpublished data. Thank you very much.
Thank you, Karen. Questions?
Yes, Andreas.
Thanks very much, Karen. Very, very exciting. Do you see difference between the northern and the see this calcium changing as well? Could that be part of your signal?
So regulated. The calcium is very regulated in otoliths. So, but what I do see is that even though the Southern Baltic cod have pancaked as far as barring the calcium goes, the pancake is even thinner in the northern part. So what I can see so far. But there's another secret, which is a whole nother talk, so I can't talk about that.
Okay, I think it's sake of time. Thank you, Karen. Our final speaker is Michael Roman. And he'll be speaking on the interactive effects of increasing temperature and decreasing oxygen on coastal zooplankton. Thank you, Mike.
Okay. Everybody hear me? Yeah, good morning. I'm not going to show the Brightberg slide, but I am going to thank the Ocean Carbon and Biogeochemistry Office in funding the travel to a number of us. And I'm also going to thank Mary Lohr, who's done 99% of the work that made this a great conference so far. So, you don't hear copepods mentioned a lot in metabolic index, right? And also when I look at the audience and gone to the poster sessions, I didn't see any copepod posters. So I threw this in, this little diagram, just so that I could give you a little 101 before we start. So, you know, copepods have a life cycle where they lay eggs. It goes through 6 naupliar stages. They look like little bugs, or if you live in the United States, a tick. And then they go through 6 of these copepodite stages before they molt into an adult. They're pretty good at grazing large phytoplankton, but if the phytoplankton are small, they eat the microzooplankton that eat the small phytoplankton. They're important for biogenic flux because they produce fecal pellets that sink hundreds of meters a day. And the fisheries people here, they're the important food or forage fish, and also the juvenile stages of important commercial fish. And these fish, you know, for the size of the copepod and the nutritional content of the copepod is very important. So if the copepod food web is changed because of temperature and oxygen, it's going to have ramifications on both biogeochemical flux and the food web. The oceans are getting warmer. We know this, right? But shallow bodies like the Baltic, Chesapeake Bay, where I live, Long Island Sound, are even getting warmer. Sometimes 2, 3, 4 degrees temperature change. So here's an example from Long Island Sound, where since the 40s, the temperature has increased 3, 4 degrees. And to the right is my favorite copepod, Acarcia, that dominates coast areas around the world. And you can see that early on in this time series, the copepods are larger. And in the latter part of the time series, they're smaller. And there's also seasonal variation. They're larger in November and they're a little smaller in the summer. So if any of you study genetics, you will go, hey, Roman, you know, maybe it's not the same copepod. They look the same, but it's a genetic adaptation. So certainly that's possible. I hope not, but it is. And so these are copepods that were cultured. This is work that was done in the Baltic. And their favorite temperature is right in here. And here are the stages of the copepods, copepodite 1 to adult. And you can see that they grew them at 9 degrees, 13.5, which is about the ambient, and 17.5. And as the temperature increases, all these stages were smaller. So the temperature affects the size of the copepods. And this has been done a number of times. Why is this so? So copepods have these life stages, and it's like the Tin Man and the Wizard of Oz, right? You know, they're in this carapace, and then temperature and ecdysone says, okay, it's time to molt. They strip off their cape, and then they're soft, and then they grow. So that molting cycle is influenced by temperature. as is the growth rate, how they put on body mass. So as you see, as temperature increase, molting at a higher temperature, they're molting faster than they could catch up to grow. So that results in smaller copepods. It's the flip side in colder water where molting rate slows down more than growth rate. So they could really bulk up between the molts. And that's one of the reasons why you have larger copepods at colder temperatures. So I went on a cruise. I don't know where you get it, in Iceland or Greenland, and go to Norway. He's going to see copepods like this, big, right? 1-2 centimeters in length. In the Panama, over the Caribbean, anywhere in the tropics, you see small copepods. And this has been true for a long time. We've known this in ecologists, all ectotherms, where you get small copepod or amphipods, and then larger ones in the poles. And this is a study done using different IPC scenario. And this is a century prediction on where copepods will go in the North Atlantic. And if you focus on the top one, these are diapod copepods that go to sleep. They have a lot of lipid, and they're really important for cod. And you could see that that population over the next 100 years is going to move from here, where they go down, and move up north. Why? Because the water is changing in temperature. The same thing for this last one. These are big copepods, 2.5 centimeters in length, and they're going to move up. So as the water warms, they're going to move towards the poles. And this has been shown for a lot of things. So does temperature alone explain these global patterns in the size of copepods? What's the name of this conference? Oxygen. Good guess, right? So it's not only temperature, it's also oxygen. So copepod respiration is function is exponential, right? The warmer the temperature, the more they respire. And body size and temperature transcend species differences. So you could model this change in respiration of copepods. And these guys recently did a global analysis, and copepods in both oceans and lakes increase their respiration about 7% per degree, right? So in the Long Island example, it went up about 3 degrees. So that means over that time, the respiration increased about 21%. Does respiration always increase with temperature? These were copepods from the Baltic. They like these cold waters. Increased temperature and looked at the rest and found that at the higher temperatures, 10 degrees, the respiration fell off, so that it didn't keep increasing. Well, why is that the case? Oxygen solubility decreases, increasing temperature. And see, these lines are closer together at low temperature. So if you raise temperature up in the Arctic, the solubility is going to decrease more than that it's going to have tropics. So these animals might be more sensitive. So if you look at respiration versus dissolved oxygen, here's a couple of copepods. You can see that it stays linear and then it falls off. So at some low oxygen, the respiration is going to be limited by the amount of substrate. You've seen this explanation over a couple times in this P-crit, and this is my version of it. I'm kind of bifocals. I'm trying to see how the screen is here, but I'll look over here. All right, so this could be a worm, a copepod, a fish. If you look at the respiration rate versus the partial pressure of oxygen, there's some critical concentration, which we call the target. And above that, their activities are not limited by the amount of oxygen, right? So it's independent. As you decrease the partial pressure, there's a level of oxygen where things start to be limited, right? And we call that P-crit. Below that, respiration or growth or feeding or reproduction is going to be limited by oxygen, and it's a linear function into a concentration called P-lith. So that's the limiting factor. but we didn't measure respiration. I've never measured respiration. I don't know how to do it. What we did is we used a bioenergetic model and we converted egg production, we converted somatic growth, and we converted ingestion to respiration units, right? So you can see that these different things up here to the right were not limited by oxygen. And then we looked at the curve and we decided that at this level, which turned out to be 8.1 kilopascals, these vital functions were limited, and then they were linear related to the oxygen concentration until we hit this 3.1 kilopascals when the copepods died, right? So this isn't, you saw sharks in a box yesterday, you saw corals in a box. These are free-range copepods, right? They're swimming around in a beaker. So this is active metabolism, not resting. I don't know how you do resting metabolism with a copepod. All right, so remember these values. So it's 8.1 kilopascals, and the critical thing is 18 degrees. So how do you scale up with different temperatures? Well, we applied a Q10 of 2.3, and then you could see as temperature increases, peak crit goes up. That's because respiration is an exponential function of the oxygen. And also the lethal temperature goes up. And a lot of local management agencies use 2 milligrams per liter. So this is this 2 milligram per liter line. So you could see, above about 15 degrees, 2 milligrams per liter is not really predicting the limitation of the copepod. All this area here is when they're limited by oxygen, but it's greater than 2 milligrams. So here's playing a little game. This copepod is pretty amazing. It lives in water 1 degree all the way to 32 degrees, right? So if you apply this Q10 value, that means that at 2.5 kilopascals is the peak crit at 1 degree, and at 32, it's a whopping 20.8, right? So that's a limitation range of 1.5 to 6.2. So at 32 degrees, cars would experience limited respiration slightly below atmospheric concentration. So that just shows the way oxygen can limit things. So I was doing the synthesis of hypoxia, and I was comparing the Baltic to the Gulf of Mexico and Chesapeake Bay and the Arabian Sea. And they say, oh, it's hypoxic because it's less than 2 milligrams. It could be 25 degrees or 5 degrees. So really, the concentration is meaningless for predicting the effect of the organism. You really need to know the partial pressure. Oh, well, this is interesting, and this was the only experiment that I've seen. We kind of played around with data from Richmond, and this is a really laborious study, but she grew egg to adult copepods at 3 different oxygen concentrations and 2 different temperatures, 25 and 15. So here I estimated what the peak crit would be. So you could see at this higher temperature, if they're raised in a sequence of lower oxygen concentrations, they actually are smaller by the time they get to adult. So the oxygen is limiting their growth. So that's another reason why you could get smaller copepods. And in 15 degrees, there wasn't that much difference because here's the peak crit, 6.21, and so that decrease didn't make that much difference because it's colder and the respiration is less. So you really need to know the temperature as well as the oxygen to predict the effect on the copepods. So this was some analysis of that data and some others that we had COVID, right, play with data. And we looked at growth rate as a function of oxygen. And the warmer things are, the faster they grow. So here are the ones at 20 degrees, 15, 20, I'm sorry, 25, 20, and 15. And then I showed you where the peak crit values would be. So you see at the higher temperatures, when there's plenty of oxygen, they grow faster in warmer water. But with decreasing oxygen, these higher temperatures, higher metabolic demand. So there wasn't enough left over really for growth. So these points emerge. And then we combine this data in a multiple regression model. And you could see that the sweet spot for this copepod growth rate is at the higher temperatures, but the higher oxygen. So all of these things kind of swerve up to that direction. So we could use this equation, P crit, to predict what the temperature and oxygen effects are for this copepod. Here's some two meta-analysis. It's kind of like the molting rate and growth rate. So this is the relative change in temperature. See, at high temperature, respirations using up most of that oxygen compared to growth rate, right? Which is why they're smaller. Just like molting in temperature, when you get down to lower temperatures, the respiration decreases faster than growth rate. That's why you get these big bulky copepods up in the poles. Okay, this is one copepod, right? Acacia. So great, only have 250 more to go, right? So we're not going to do this. So we have to think of some other ways to use allometric scaling so that smart people like some of the ones in the room could model this effect on ocean ecosystems. The good thing about copepods is they don't have gills, right? So it's not like fish, big gills and low oxygen, little gills and high oxygen. They don't do ventilation. You know, they're pretty simple. They get their oxygen through little pits in their skin. And it's just based on the surface area, integument thickness, and diffusion. So these are the shape of copepods. They kind of look like American footballs, right? They're all spherical. And using a geometric model, we could estimate their surface area and volume. So you see these big copepods, Calanus, like are up in Norway on North Sea, you know, their surface to volume ratio is about 5.6. These small copepod tropics are an order of magnitude higher, right? So there's a rich body of literature that small phytoplankton are favored in oligotrophic conditions because it's better transport to the surface, to the interior. And we're hypothesizing that the same is true in copepods. So it might look like something like this. As you increase the surface to volume ratio, P crit goes down, right? So that's why a smaller copepod might be able to do better at higher temperatures and lower oxygen than a large copepod. So it would look something like this, where, you know, the P crit for the large copepod would drop down here, and the P crit for the smaller copepod would be there. And then you could predict which one is going to be favored in this different temperature and oxygen environment. And again, we have acoustics that has size spectrum of zooplankton. If we could figure out how to scale this, it would be fairly easy to model up to potentially show the effect on fisheries. So you've seen this metabolic index. This is what we're working on. I think Jody mentioned this early on. You know, an animal living in an aquatic environment really is up against it for oxygen. There's 34 times more oxygen in a liter of air, maybe not in this room, but, you know, in a liter of air than there is in water. And more importantly, the diffusion rate is 300,000 times, right? So it's really hard. You might ask, in terrestrial, about a month ago, I was in Colorado doing mountain climbing. You know, where I live is at sea level, and puffing and puffing, right, at the lower oxygen in the atmosphere. You can see that there are terrestrial insects that are also smaller as you go up mountains. Well, here's scary to leave you with. In the Paleozoic, oxygen levels are about 40% of the atmosphere, not 20%. And these dragonflies were fossils that they found in France. They were 30 centimeters long, and their wingspan was 70 centimeters. Live in the Paleozoic, she's shown there as examples. So this is an example of how oxygen, even in the terrestrial environment, could influence the size of something.
Thank you, Mike.
Great talk. I really enjoyed that. And I kind of feel like we should all have a favorite copepod now. A little bit more appreciation for copepods. Just out of curiosity, did you play around with some different use for the model?
We did not, but we did do a meta-analysis of all Q10s, right? And you know, they're down as like 1.8 to 2.6, and whatever was 2.3 was right in the center. So it isn't a huge amount. Thing is, some of these copepods, you know, say in the blacks in the Baltic, they're at 9 degrees, and then you put them in this different water. What varied is the acclimation time of some of the investigators, and that could affect the Q10, but that's a good point to bring up.
Thank you.
Okay, I think we'll leave it there. Thank you very much, Mike. And I'd like to thank all the speakers for a great set of presentations.
So it's our pleasure now to welcome Dacia Atamanchuk. She's a chemical oceanographer. She's a researcher from the Ocean Frontier Institute in University of Dalhousie in Halifax, Canada. She's also a member of our steering committee of IOCCP, the International Ocean Carbon Coordination Project. And today she will tell us about the ocean losing its breath and some insight from the Susten observation in the deep water formation region. So the floor is yours, Dacia.
Thanks so much. I'd like to thank organizers and sponsors of this event, but also Marie-Laure and Veronique to kindly invite me to give keynote presentation today. And I hope you'll enjoy it. I'll leave some time at the end for the questions, if there is any. So today I'd like to talk about the role of deep water formation region in the problem of ocean deoxygenation. And I would like to start with a bit of first time the scientists were able to observe the deep water formation and its relation to the increased inventory of oxygen was actually in the Labrador Sea in between 2003 to 2004, and it's coincided with the onset of development of oxygen opto technology. So the scientists were able to see the progression of the deep layer here shown in blue, and the increase in the inventory of oxygen here in red. And then 15 years later, 2017, 2018, we accumulated enough knowledge based on observations, not only those spanning 15 years, but also the years preceding observation with the optodes. We accumulated enough knowledge to tell us that ocean is actually loses its breath now. And observational studies show that this decline is as much as 2% last 5 decades. And then it touches all the parts of the global ocean, from coast to the open ocean, from the surface ocean to the deep. Two nice things about, this ocean loses its breath and ocean takes a deep breath analogy is that, it helps us relate to the problem of ocean deoxygenation to this personal level, because we liken it to the human breathing. And then if we look at the global ocean and the exchange that happening between the surface ocean and the atmosphere, we can see that this exchange is not uniform, and that there are certain regions that act as a sink of gases. Some of them act as a source of gases. And we can say that ocean breathes through its skin, let's call it that way. So the only way the oxygen can get into the ocean is through its surface. However, there are a couple of regions of the global ocean. One of them I marked here with the red circles, where there is an intense uptake of gases, both oxygen and carbon. And in these regions, the uptake goes from the surface and reaches the very deep depth. We call these regions the region, and there is just a couple of them in the global ocean, some of them in the North Atlantic and subpolar North Atlantic, and there is a couple of regions in the Southern Ocean. All these regions are characterized by the heat release to the atmosphere that in gases, so the gases ocean, but then it's through the process that is called deep water convection. These gases eventually reach the deep ocean, and then by the means of the conveyor belt, this oxygen-saturated water finally being distributed through the Atlantic, Indian, and all the way to the Pacific Ocean. So what the analogy with ocean breathing here is that we can, if we think about the global ocean and that breathes through its skin, then the lungs of the ocean are actually these deep water formation regions. That's where the ocean takes a deep breath and then keeps it for about 1,000 years. This is how long it takes for the water to travel from the deep water formation region in the north all the way through the Pacific and back. So what will happen with ocean breathing in the future and how this ocean breathing can be changed. And the model estimates tell us, all of them tell us that the northwest oxygen will be reduced, and that's mainly due to the fact that the water becomes warmer, but also deep water formation of the ice in Greenland. So the melting of ice make water lighter, and that doesn't allow thermodynamically for the efficient mixing to happen. So the questions we have as a community is how good are these predictions and what can we do to make them better, but also how do we make them better? And that's, I guess, the question that us observational oceanographers are keeping in mind. How can we improve the model estimates through observations? And projections are not very good, I should disappoint you. I took this figure from a presentation of Andreas Oschlies in, I think, 2018, 2019, where he shows that the decrease in oxygen according to the models, climate models, show about 0.6% decline over the last 50 years, while the observations show only 2% decline. I think most scary part here is that, to me at least, that if the underestimated models give us only 4% decrease by the end of the century, what the real decrease might be. So that's a number kind of quite scary. And here we put deoxygenation in different terms. We think about the surface ocean and we can think about the deep ocean. The surface ocean quite well characterized, and models are quite, actually doing well. They predict decline in the surface ocean as a result of warming and decrease in oxygen concentration. However, whatever is happening in the deep is really hard to predict, or that's where the biggest disagreement between observations and models are. And I'd like to take this approach of sinks and sources of oxygen to the deep ocean in order to frame my today's talk. So the reason why the deep ocean is losing its breath is because there is either a decreased supply of oxygen or there is an increased demand in the deep ocean. Supply of oxygen, of course, has happened through this deep water convection and deep ocean breathing events. But then the sinks of oxygen are numerous. These are related to the circulation uncertainties of our understanding how the internal climate oscillation are performing. These include the Pacific Decadal Oscillation, North Atlantic Oscillation, and also how the circulation may affect oxygen concentration. So the slower water moving, the more chances it has to accumulate the respiration signal, so the lower concentrations would be. And finally, the biotic control on ocean deoxygenation. is through understanding of the mechanism and the amplitude of the export production. So the primary production that happens at the surface, where there is light and nutrients, eventually that organic matter falls down through the water column and reaches deep ocean, and that what causes decreased levels in the deep ocean too. How do we deal with this problem? And I think there is a consensus that we need more observations, but what we also need is a targeted studies to find out what are the mechanistics of these processes that can influence the oxygen. We need to keep an eye on what's happening, but also we have to understand the processes behind what's happening. And in today's talk, I'll be using some of the examples from the Northwest Atlantic, and I will be using the underwater winch profiler. I'll come back to that later, but also the data from the 53 North Mooring Array and the Argo and BiArgo data in the region. So we'll start with the ARC exchange. And what do we know about our ARC exchange in the Labrador Sea? We did this simple exercise. We measured surface concentration of oxygen in the Labrador Sea, and then we applied the known parameterizations of ARC exchange and wind forcing to calculate the oxygen fluxes. And this is the top panel here. In this panel, you can see that the classical diffusive models on the left, and then the models that include bubble inclusive parameterizations on the right. So bubbles are being injected during winter storms, and then that increases concentration of oxygen in the water column. And would be really hard to decide which one works well, because there is a really, really high disagreement between them. And especially if we look at the bubble diffuser model, disagreement between those two. Fortunately, we had Argo data, Argo flow data in the region that were there throughout the year, and they told us that there was an increase in the inventory of oxygen of about, and that served us as a reference point. So then, and with a wind speed product, we were able to say that inclusive parameterization of Stanley seems to work fine. However, it's just a matter of fitting data to the number that we obtain. So it's not really a constructive way of doing it. So we need to really know how much gas is being pumped in through the convection event. The other interesting fact is that if we take different wind speed products, so in the Labrador Sea, we used North Atlantic reanalysis product because it was verified against actual observations from, there are available a number of wind speed products. And if you, the spread of the values they see, it's actually quite fascinating. Some of them, difference in oxygen flux is about twofold. So here we already have two problems. One of them is that the parameterization that we have is not sufficient to resolve these bubble fluxes, but also the wind products that we have, they disagree. And the biggest disagreement they have is at high wind speeds. Why high wind speeds are important is because it's particularly these high wind speed events, about 2/3 uptake of oxygen is happening. So there's the higher wind speed is, the high uptake is. So if the model doesn't represent this high wind events well, it means that we'll not be getting the right number for the oxygen flux. The question why the parameterization do not work well, I think lies in the fact that the region of the Labrador Sea is different from the region for which the parameterizations were developed. So most of the parameterizations are based on the empirical data, and they developed for the regions where the water column, the the air-sea exchange lasts just for a couple of days, months. Here in the laboratory shows that there is an oxygen still stayed at the end of the convection, and it doesn't depend. It depends on how long the convection lasts here on the x-axis, but also the depth of the convection here on the y-axis. So still with the shallowest mixed layer, with the shortest there's still under saturation of oxygen at the end of the convection season. And that points to the fact that there is a take rate, which means that the air-sea exchange will continue as long as the surface stress, external forcing will keep the gas flux into the ocean. So the representation of winds can have a fundamental impact on our understanding of circulation as well. And here it's important for us to link the circulation on the global scale to the circulation region. And it's normally that circulation, or so-called overturning circulation, the change in the density relate with the changes in oxygen. However, this fact was never verified based on real observation. That's one of the work that's been going on. A group that has been equipping transport mooring North Atlantic. Here is showing one of them. That's the Orion Array that stretches from the Canadian coast all the way to UK through Greenland here. And by equipping this transport mooring array that measures a transport of water with oxygen, able to relate transport of mass to the transport of oxygen. And why this? So I would say creative in a way is that we already have years of data recorded from this transport mooring arrays. So if we manage to find a relation between the transport and provide us as invalid transport of oxygen from the region down to North Atlantic. And here is shown the way we're doing it. So we simply clamp additional sensor next to the instrument that measures CTD. So it's quite easy to do. Doesn't require establishing a completely new program and infrastructure. And at 26 North, there is a rapid mooring array that also has mooring at this deep western boundary current that will also measure oxygen in the future, which means that will be oxygen from its formation in the Labrador Sea through its and direct export pathways all the way to the North Atlantic. I should mention that this oxygenated water feeds a lot of life along the eastern coast. We managed to tackle just the first part of it, or try to tackle the first part of it. So we're looking at transport of oxygen that is following the formation of deep water in the Labrador Sea. So here in the green lines on the left-hand side, direct export path of that, the dashed lines show the path. So we are able to relate the increase in concentration in the deep water formation region and then its passage through this. And what we managed to discover here is that there is a signature of this newly formed oxygenated water that passes 53 North Array roughly a couple months after its formation in January, February in the central Labrador Sea, and it's about half. So meaning that about 50% of the air that's of oxygen that's been taken up during the convection months makes it this direct southward export pass into the North Atlantic. The rest gets recirculated in the region and eventually gets mixed into the deep western current and exported too. However, the most direct route responsible for about 50% of oxygen increase. And to understand the importance of it, we also calculated the oxygen demand in this player in the North Atlantic, and it's about, and that equals 71% of oxygen demand in the region. So the importance of deep water convection in the region and sensitivity to the changes in the supply of oxygen to the deep sea and the change. Water is being exported out of the Labrador, the North Atlantic, and later other oceans. The main process that will be consuming oxygen is biological activities. Here, we need a knowledge about the biological carbon pump in order to understand really the extent of oxygen decline in the deep ocean as a result of biotic activities. There are a couple of pathways of biological carbon pump, export production so-called. Here I list only 3, but I'm listing the main one, the gravitational carbon pump, that is dominant in the most regions of the global ocean. And the export path can be described by the Martin curve. So production at the surface is followed by remineralization in the subsurface. And as the water, as the organic matter sinks, it gets respired more and more. It means that only a fraction of organic matter that's being formed on the surface eventually makes it down to the deep ocean. Should also mention that because of the process of this organic matter is not really, a top-notch product anymore, and the energy benefit from it is really low. However, in the regions like the deepwater formation region of the Labrador Sea, other pathways take place. One of them that is observed in early spring before we have the establishment of this shallow mixed layer. And what happens is on nice sunny days, there is production happening at the surface. However, the water column is not stable yet. So the organic matter that's being produced at the surface get physically mixed into the deep layers and it stays there. So instead of going through this pathway, a slow pathway of respiration, fresh, very desirable, very high energy organic matter gets straight to the deep ocean and stays there. Another pathway is called eddy-driven carbon pump, and it's observed in the regions of high eddy activity, and Labrador Sea is, of course, one of them. And this pathway is characterized by the subduction of organic matter, particularly organic carbon, along the edges of the eddies to the depths. You can see the blip here. That was actually observation from the glider that were able to detect the aftermath of this eddy-driven pump. And similar to the mixed layer pump, it delivers small non-sinking particle to the deeper layers, which otherwise wouldn't get there if we're thinking only about the gravitational carbon pump. And to emphasize the importance of these alternative pathways, I'd like to show you a modeling study and highlight the regions in the North Atlantic, which shows that about 30 or 50% of total export of organic matter happens through the eddy-driven pump. We would also been able to confirm the existence of these pathways in the Labrador Sea through the direct observations. So the export basically is a balance between production and respiration. So by constructing a mass balance, we were able to see that efficiency of export is about 36% during the conventional spring bloom and summer period, which is more or less consistent with the gravitational carbon pump pathway. However, if we take into account the period before, the pre-bloom period, where the water column is still unstable and it's been constantly mixed, we get about 99%. What it means that organic matter that's being produced at the surface almost entirely makes its way to the deep. And that has an implication towards the preformed, so-called preformed oxygen levels in this region. So by supplying organic to the deep ocean, in the future lowering the oxygen concentration there, because this organic matter will require analyzed. To summarize, what we know, what we don't know, and how can we decrease the errors in our estimates. I would say that we need a targeted studies about the mechanistic understanding of the air-sea exchange and the mechanism and the amplitude of export production. And its relation to the oxygen transport can, I would say, easily be addressed, not easily, but can be addressed by adding sensors to the existing transport arrays that already measuring transport of water. But on the global scale, that the study, especially the study of mechanisms and the amplitude of export production, should be studied better. And that has to do with the fact that we may have a change in the productivity of the ocean, but we may also have a shift in the mechanisms that transport organic carbon to the deep ocean. And these have to be carefully examined one by one. And of course, how we do it by means of and thinking about the observing network that can address these issues, in addition to already existing Argo program, platforms like moorings gliders and wave gliders. A good addition would be to have a set of these profiling reference ocean sites, just like the Seacycler or any other profiler, basically, that can accommodate a suite of sensors that usually demand more power, and then they cannot be simply put on the mobile platform due to their size and other limitations. And these, and of course, specialized moorings that will help us to really study the processes in hotspots such as Labrador Sea, Southern Ocean, Pacific Ocean, in order to better understand the processes behind oxygen, deoxygenation. And I would like to thank, of course, all of my collaborators for providing their expertise and time in collecting, analyzing, and making sense of the data. It's an enormous amount of work, trust me. Thank you.
Thank you, Dacia, for this very stimulating talk where the ocean is freezing. So we have time for a couple of questions.
Yeah. Thank you, very interesting talk. Indeed. I have a question. When you speak about the concentration of oxygen in the surface water, do you mean upper layer directly involved in the exchange with or without bubbles, or is it averaged for upper mixed layer, the layer where you can measure with sensors, for example, in Argo floats?
I'm talking about the layer, the Labrador Sea layer that is formed as a result of the deep convection. So it all has uniform concentration of oxygen and the same density, basically. So it reaches down to 2,000 meters, depending on the year.
Andreas?
Great. Question about abiotic transient traces. Since I think most of the mismatch or gap in our understanding is probably due to physical transport processes. And have you measured in this taking a breath events, the CFC or SF6 or other, well, like Argon 39 or some traces help to better constrain circulation also in models in the end?
and not in relation to the oxygen uptake, in relation to the anthropogenic carbon, yes, but we're not being, it's not a part of the oxygen work, but we have the data, yeah.
Oh, great, okay, yeah, we should lobby for that, thanks.
There is a question right in the back with Mike, and then we'll proceed.
Oh, great talk, thanks. Just out of curiosity, how do you kind of correct for or take into account, water from the overflows and their oxygen concentrations and things like that?
So when we construct the mass balance, we take into account lateral fluxes, including the water that comes with the eddies from the western boundary current. So by constructing this budget, we for entrainment lateral fluxes and also the overflows that comes with the boundary current, but not the overflows at the deepest part itself. That makes sense. So the GoSNAP program is designed as that. So we're going to add the flow of oxygen around Greenland, and then the mixing in the overflow waters into the deep boundary current there.
Last question from Mike, short one.
It better be good, right? Thank you for a wonderful talk. A number of people are working the problem of potential decreased global primary production because of restratification. So I was wondering if there's any data or what are your thoughts on reduced primary production in these areas of deep ventilation that would reduce the organic material coming down for that reduction in oxygen?
Yeah, I don't have enough observation to say whether there is a decrease or increase. We are mostly targeted like the process understanding rather than trying to measure the amplitude of it. So I don't have an answer, sorry.
Well, thank you, Dacia. I think we need to move on. And so now we have, I think Daniele is online. And it will talk about characteristic, drivers, and consequences of from climatic trends to local scale variability. So I guess the floor is yours.
Thank you. And thank you to the organizers for organizing this wonderful meeting. I couldn't be there in person. I'm actually in South Korea, so it's 7 p.m. here. Thanks for moving my talk to an earlier time. But I remember being there 8 years ago and it was really wonderful. So I hope to be there in the future. So what I want to talk about is work that we are doing at UCLA. So it's part of a big collaboration. I want to acknowledge collaborators at UCLA, Pierre McWilliams and Anh Pham, collaborators at the Southern California Coastal Water Research Program. So if I let me know, and collaborators Curtis Deutz and Evan Howard. And this is going to be a talk about hypoxia along the US West Coast shelf based on model simulation that we developed at UCLA. All right. So this is a sort of the take-home messages and the outline of the talk. The first one is that hotspots of hypoxia emerge along the US West Coast shelf, and they are driven by the seasonal upwelling and really by sub-mesoscale variability. And you need a high resolution model to capture variability on the continental shelf. The second message is that local pollution can actually worsen hypoxia even in a region like the California current that is very, very dynamical and exposed to the open ocean. The third message is that climate change drives a inevitable of hypoxia along the US West Coast, and we see that with model simulations. And finally, there are consequences to biology that include species migration and even local extirpations. So I'm going to focus first on the first point. So the talk is going to be mostly about the first point. Then I'm going to give you a few vignettes, short slides about the last 3 points. And this is all work done by collaborators that I introduced earlier. All right, so the tool for our work is the UCLA Regional Ocean Modeling System, ROMS, coupled to a ocean biogeochemical model, the biogeochemical elemental cycling model. And we develop a set of nested configurations, so coast-wide, and then we start zooming in. So we have 2 smaller nests at 1 kilometer in the northern and southern coast that we together and analyze in a seamless way. It's important to go to this high resolution, especially if you care about processes that happen on the shelf where their ecosystem and resources are found. And the width of the shelf a lot going from the north to the south, but it's generally a few kilometers to a few tens of kilometers. And so you really need models that capture these scales if you want to say something about the shelf itself. We run the model for about 20 years with realistic forcing, and we save daily output so we can do statistics on that. We also have 2 fine scale nests at 300 meters, and I'm going to talk maybe today about one that we used in the Southern California Bight to study the effect of anthropogenic nutrient inputs and pollution. All right, so this is a model. This is what the model reproduces on average, oxygen at 50-meter depth. And of course, we want to compare that to observations. And we have been engaged in an extensive validation of the model that is really enabled by the richness of observations in the California current. And actually, I want to take a second to acknowledge and all the people that are involved in collecting, analyzing, and sharing observations. There will be no modeling if there were not this type of rich observations. So the model does reproduce the patterns in oxygen, the low coast, and the transition to higher concentrations offshore. It also reproduces seasonal cycles. So I'm showing here for the Newport region where there is rich observations at the surface in blue and below the surface in red. This is a climatological seasonal cycle, but you can see observations as dots, obviously for an entire, for a period of several decades. The same thing can be done in different regions where we have observations. And in general, our model is quite good at reproducing seasonal cycles. I should say it also reproduces some fraction of the sub-seasonal variability. But I should also say that there is more variability in the observations than in the model. And that might be due, well, the scale of the model is not obviously only 1 kilometer. There is also, we save daily output, so there is sub-daily variability. And there was a great talk yesterday showing a huge variation over the course of a day. And then there might be other unresolved scales. So this is something to keep in mind, that we're still underestimating variability with this model. We focus on shelf processes. So I'm going to focus on shelf processes for the rest of the talk. And we actually take a long isobath coordinate system that really allows us to highlight what happens on the shelf itself. So I'm going to illustrate what I mean with a plot of primary production. This is units of carbon per cubic meter per unit time on a log scale. So it's really strong on the shelf. Less strong offshore. And notice that I'm showing the shelf on the right panel. Maybe you can see my my mouse, but on the right panel, basically, we average all the observation using isobath as a coordinate system, and then doing a meridional along isobath average. So at every point, we use this coordinate system to really see what's going on the shelf. We go from 0 to 200 meter. We take 200 meter depths as the boundary of the shelf. And then on the left panel, we go offshore. And so we revert to a depth coordinate system, still going parallel to the coast, but showing distance versus depth. So anytime you see this type of plot on the right is a along isobath coordinate system that really highlight what's happening on the shelf. So there is a high production on the shelf. It's really enhanced compared to the offshore region by several times. It drives strong organic matter export and remineralization at depth. So the remineralization is shown, oxygen consumption is shown below. In the blue values show you consumption. And the peak of consumption is shallow, tends to be near the shelf, and is really due also to the fact that sediments accumulate organic matter and suck in oxygen as well. This upwelling is seasonal, and so there is an interplay between seasonality that moves water to the shelf and drives the productivity. And by doing so, it also moves low oxygen waters along the shelf, which you can see here. But there is also a role for this local respiration that further draws down the oxygen on the shelf. There is also a lot of variability. And so with the model, we can start estimating the variability. In this case, I'm showing you the root mean square error of oxygen. So something like the standard deviation. It's also in a nonlinear scale, but I really want to highlight the strong variability on the shelf center at a depth of about 50 meters. And this is all for the central coast, but of course we can characterize that all along the coast. So we do that here. We show the root mean square oxygen at a depth of 50 meters or shallower using this isobath coordinate system. So we go north to south, and we're really expanding the shelf region. This is the total variability in oxygen, so it's the root mean square. And the model allows us to decompose that into a seasonal component that corresponds to the upwelling time scale, and a sub-seasonal component that reflects wind events, eddies, and sub-mesoscale fluctuation. They are different. The seasonal is stronger and explain most of the total variability, but the sub-seasonal, the eddies, and the sub-mesoscale events are quite important, especially at isobaths or depths shallower than 50 meters or so. And they tend to be important throughout from the north to the south of the domain, while seasonal variability is particularly strong in the northern part. The high frequency output of the model allows us to do statistics on hypoxia, and so start characterizing a hotspot of hypoxic conditions. By hypoxia here, I just use a threshold of 60 millimole per cubic meter, just a representative threshold. And so we can start looking in our isobath coordinate system at the frequency of events that are below that threshold. So on average, you are not below 60 millimole per cubic meter, but there might be short events that go below this threshold. And they tend to be, at least in the north, quite shallow along the shelf, and they are driven by this combination of upwelling, but also local respiration. In the north is more seasonal upwelling than dominates. If you start going towards the central and southern part, the picture changes. Is similar in the central, quite different in the south, but really we see the emergence of these hypoxic events on the shelf. We can look at how they vary in a little more detail using again our isobath versus latitude coordinate system. And there are several where hypoxia is more frequent. So these are really what we call hotspot of hypoxic condition. They have different drivers. In the south is more, or in the center south is more remineralization. In the north is more upwelling. And the model allows us to do all sorts of statistics that we started doing, including things like duration of hypoxic events. So we see how in some hot spots, hypoxia can last up to several days. The most extreme events are up to a month or so, but in some other regions, it's very ephemeral and it lasts for only a few days. We can also look at other properties, intensity, recurrence, co-occurrence with other stressors, and we started thinking about potential biological impacts on the shelf. So this is what we are doing with the high resolution modeling, but I want to switch gear a little bit, and if I have a few more minutes, I want to talk about local hypoxia, climate change, and biological impact. Local pollution. So we started investigating local pollution starting from the southern part of the domain using a very high resolution set of simulation that are around 300-meter horizontal resolutions. They really represent the sub-mesoscale and the shelf very well. And we have two sets of simulation. One that represents anthropogenic nutrient inputs that we spend several years actually putting together from all sorts of observation. They include urban wastewater, agricultural runoff, and we compare this simulation with anthropogenic inputs to simulation that have no anthropogenic inputs, so they represent something similar to the natural state. And this has been published last year in a paper by Kesuri et al. So what are the impacts of anthropogenic inputs? There is local eutrophication, shown here as a change in primary production and phytoplankton biomass that is really intensified all along the coast. The arrows here represent point sources of nutrient inputs from large underwater outfalls. And this increase in primary production obviously drives increase in organic matter formation and export, and in turn, oxygen loss. So here is increase in respiration and carbon export going up to 2 times all along the coast in certain hotspots, and a decline in oxygen shown by the blue line down to concentration of a reduction of several millimole per cubic meter and down to 20 millimole per cubic meter or so. Another way of looking at these anthropogenic effects is taking a section from the coast to offshore that crosses one of these local hotspot of eutrophication. And again, we see this intensification of oxygen loss right on the shelf at 50 meters or so, and a decline of 15 to 20 millimole per cubic meters during certain times of the year. So we are currently investigating the patterns of this anthropogenically driven oxygen loss in the California current and the biological consequences, and of course, transmitting the results to stakeholders. Another quick vignette is climate change in the California current. So we were engaged in a downscaling of global Earth system models from the IPCC, so the fifth assessment report. Basically, here is a summary result from a paper published last year by Auer et al. Basically, what I'm showing here is oxygen change below the surface all along the California coast from our ROM simulations, showing the oxygen lost at the end of the century relative to the present day under a high emission scenario. And this is driven by climate change. And oxygen can go down by several tens of millimole per cubic meter and up to 30 or 40 millimole per cubic meter. So coast-wide oxygen loss is inescapable. This is actually a multi-model ensemble mean. And so no matter what model you use, you get well, you get similar responses, but this is the mean. Other people have done similar calculations. So I want to highlight the group at University of California, Santa Cruz, and the paper by Pozobuil et al. That also shows in the California current at a similar resolution, a coast-wide decline in oxygen. And finally, one point that I want to make is that these are coarse impacts on the shelf. You really need to higher resolution. And apologies if I'm a little bit breaking down, but there was a nice paper last year that showed that you need high resolution to capture climate change on the shelf at scale similar to what I shown earlier. So this is my last point. We are engaged in doing biological impact assessments based on these model simulations. And so what we do is applying, using model output with the metabolic index framework developed by Curtis Deutsch et al. That was actually introduced on Monday by William Zumstock to do basically calculation of aerobic habitat loss for different species. We apply this framework for anchovy species in the California current. And so we basically show, or Howard et al. Show that the metabolic index and the aerobic habitat correlate very well with abundance of anchovies along the California current and their range. And you can actually use the metabolic index to calculate changes in aerobic habitat over time and project them into the future. So this, I promise, is my last slide. Let's see if it comes up for me. And it's basically showing by the end of the century, based on our simulation and this aerobic habitat calculation, what's the loss of habitats for anchovies in the California current. So it's a quite dramatic loss of habitat in the southern part. Is driven both by temperature and oxygen loss. And by the end of the century, a lot of the southern part of the domain will not be viable for anchovies. The same calculation can be done for other species for which you have metabolic index information. And so we are engaged in that effort right now. So I'm going to stop here. Hopefully my summary should come up, and I'm going to take questions if there is time for it.
Thank you. Very nice talk. So, Lisa.
Thank you for that really nice overview. So I have a question about the ability of these models to capture short-term events. I know you only went through 2017, but in 2020, Southern California experienced a massive dinoflagellate bloom that led to coastal hypoxia and I believe anoxia in the open coastline, something I had never seen in 30 or 40 years of working there, and it lasted a couple of weeks. Can the models, and it coincided with unusually warm weather, unusually high precipitation, and probably some nutrient inputs resulting from that precipitation. But can you capture that sort of event?
This is an excellent question, and we have not looked at, well, we have not run the simulation for this period. There are two elements to your question. One is the ability of the model to capture the very coastal scale where you might observe this sort of coastal stratification, eutrophication, and the hypoxia. And I would say maybe going to the 300-meter resolution, you start having the right scale for these really nearshore events, but there is a lot of physical processes that we're still building up in the model. There is another element. You mentioned dinoflagellate blooms, and so this goes into the biogeochemical formulation. Our model is relatively simple. It's obviously very expensive to run. We use a model that has been validated. It's actually quite good at getting the very high chlorophyll during bloom. So we get concentration of chlorophyll up to tens or even 20 milligram per cubic meter, but we do not resolve diversity of phytoplankton, for example, dinoflagellates versus other types of phytoplankton. So I really don't know if we can capture the details of these events, but we do see all sorts of events that we are starting to study right now.
Let me just say this event extended 100 kilometers, so it wasn't a small event, and there were massive fish kills on the beaches.
In that case, there is a better chance of capturing if it's a widespread event.
Okay, thank you, Daniele. I think we need to move on. Remember, you can ask questions on the Padlet, especially for those who are online. So we'll go to the next talk, which will be given by Zouheir Lashkar, who must be also remote. He will talk on the Arabian Sea oxygen minimum zone, variability, recent trends, and vulnerability to future climate change. Hello, Zouheir.
Hello. Well, thank you very much. So hi, everyone. I'd like first to thank the organizers for organizing this great meeting and for giving me the opportunity to present this work. So I'm going to present some of our recent research on the Arabian Sea oxygen minimum.
Sorry,
in terms of
excuse me, we're not seeing your slides, so
okay, so
it's getting there. Yeah, sorry. You can start again.
Okay, thank you very much. So I'm going to present some of our recent research on the Arabian Sea oxygen minimum zone in terms of its recent variability and future vulnerability to climate change. But let me first start with providing some context. Okay. So several features make the Arabian Sea quite unique. First, it is subject to seasonally reversing monsoon winds and currents. It hosts the only major western boundary upwelling system. And as a result, its ecosystem is highly productive with 2 bloom seasons. It is also relatively poorly ventilated due to its landlocked geography from the north, and its hydrography is strongly influenced by marginal seas with extreme conditions like the Arabian Persian Gulf. And most importantly, for the purpose of the presentation, it hosts the world's thickest and one of the most intense oxygen minimum zones, making it a global hotspot of denitrification. But the Arabian Sea is also a hotspot of global change. First, it underwent a very fast warming over the last few decades, as can be seen in the left panel, showing trends in the sea surface temperature in the region between 1982 and 2010. In particular, the Arabian Gulf and the Northern Arabian Sea have warmed at a much faster rate than the global average over the period. Trends in satellite chlorophyll observations shown in the top right panel also suggest a recent decrease in productivity in the region. And finally, a decline in wintertime nitrate to phosphate ratios in the subsurface of the Arabian Sea has been observed over the last few decades, as shown in the lower panel. This change has been proposed as a potential factor contributing to an observed recent shift in the wintertime phytoplankton dominant species in the Northern Arabian Sea from diatoms to Noctiluca sentiens, a dinoflagellate species. So, however, this decline in the nitrate to phosphate ratio does also suggest a potential increase in denitrification in the region. And recent observations in the region indeed suggest an intensification of the oxygen minimum zone in the northern part of the Arabian Sea. So here I show examples from 2 studies that illustrate this change. So on the left panel, you can see historical oxygen profiles in the Gulf of Oman region. Recent observations collected after 1990, shown in indigo, generally display a stronger oxygen depletion in the subsurface than other observations shown in yellow. On the right panel is shown the average oxygen profile for each decade from the 1960s to the years 2000 in the region around Oman. And again, you can see that oxygen depletion and hypoxia are sharper and occur at shallower depths in the recent decades relative to the previous decades. So this leads me to formulate the main questions I would like to address in this talk. First, how much and where did oxygen decline in the Arabian Sea? And the second question is, to what extent did recent warming cause or contribute to these trends? And finally, will the Arabian Sea oxygen minimum zone expand or shrink in the future? To answer these questions, I will be showing results based on model outputs from both regional and global ocean simulations. So here I present results of an ED resolving hindcast simulation of the Indian Ocean and show trends in oxygen inventories in the layer between 100 and 1000 meter on the left panel. And on the right panel, I show oxygen trends along a north-south section crossing the Arabian Sea at 60 degrees 5 east. So the trends are shown in percent of the average inventory per decade and reveal clear deoxygenation in the Northern Arabian Sea that is particularly strong between 100 and 300 meters, as can be seen on the right panel. So as a result, the volume of the oxygen minimum zone represented here by the volume of hypoxic water, and I use the same threshold of 60 millimole as a definition of hypoxia, 60 millimole per cubic meter. So you can see the volume of hypoxic water in green, and it increases at relatively weak yet statistically significant rates of around 0.6% per decade, while the suboxic core of the OMZ, defined here by oxygen below 4 millimole per cubic meter, which is shown in purple, as well as denitrification in blue, increase at a much faster rate of about 14 to 15% per decade. Now, the question is, what drives the oxygen decline, this oxygen decline? So to try to answer this, I show in this figure the accumulated sources and sinks of oxygen anomalies over the study period. In blue are shown the oxygen anomalies associated with ventilation. The part of ventilation driven by vertical mixing is shown in light gray, and the advective fluxes are shown in dark gray. And finally, oxygen saturation levels in purple. So as you can see, most of the oxygen decline is driven by ventilation changes, while neither biology nor solubility does show any negative trend over the study period. The second observation is that most of the oxygen decline associated with ventilation is driven by decline in vertical mixing, which explains almost 80% of the ventilation negative trends. Now the question is, what ultimately causes the reduction in the ventilation of the thermocline in the Northern Arabian Sea? And in order to answer this question, I show here the results from 2 sensitivity experiments. So to test for the role of warming, we have run a simulation that is similar to the control, but where we used climatological heat fluxes. So we repeated the same, what we call normal year forcing for the heat fluxes. And so in this case, the ocean experiences no warming. And to test for the role of warming over the Gulf region only, we have run a simulation where we use the climatological heat fluxes over the Gulf region only. The reason we performed this test is that it has been recently shown that the Gulf is a major source of ventilation for the upper OMZ in the Arabian Sea, and that warming of its waters can reduce their subduction in the Northern Arabian Sea. So the 3 panels here show the trends in oxygen inventories in the same 100 to 1000 meter layer in the control simulation, as well as and these 2 forcing scenarios. So as you can see, in the absence of upper ocean warming in simulation S1, the deoxygenation trends in the Northern Arabian Sea are actually replaced with oxygenation trends, with an increase in oxygen. And even when the absence of warming is restricted to the Gulf only, the deoxygenation trends are significantly weaker, which suggests that the oxygen decline in the Northern Arabian Sea was mostly driven by upper ocean warming, including in the Gulf region. So now the question is, what will happen to the oxygen minimum zone in the future? Will it expand or shrink? So here I show a figure from a recent paper by Matthew Long and colleagues that presents the CMIP5 models projected oxygen changes under the business as usual emission scenario as a function of depth and latitude on the left, and the contributions of changes in solubility, the so-called thermal effect in the middle, and that of the so-called apparent oxygen utilization, or the non-thermal effect, which is basically driven by changes in biology and circulation on the right. And the point I would like to make here is that although globally the deoxygenation signal dominates in the tropical thermocline where the OMZs are located, models project only either a weak deoxygenation or even a slight increase in oxygen. And this is because while in the mid and high latitudes, the effects of oxygen, or the effect of oxygen solubility and the biological utilization add up, in the low latitudes, these two tend to oppose each other, resulting in a relatively weak and uncertain future trends in the tropics in general and in the Indian Ocean in particular. So for the Arabian Sea, most CMIP models show patterns that are consistent with the changes simulated here by the CSM large ensemble model displayed in the upper left panel. That is the oxygenation in the upper layers of the northern Arabian Sea and oxygen increase in the southern part of the Arabian Sea. So in this particular study, as well as in other studies, the changes in oxygen appear to be mostly driven by changes in the apparent oxygen utilization, and more specifically, changes in the ventilation of the thermocline, where the Northern Arabian Sea, with the Northern Arabian Sea getting less well ventilated, whereas the Southern Arabian Sea gets more ventilated, as can be seen in the simulated changes in water age shown in the upper right panel. There is also the reduction in biological production and export fluxes shown in the lower right panel that may also further contribute to increasing oxygen in the southern part of the Arabian Sea in the future. However, these projections come with very large uncertainties related to the fact that oxygen changes translate a change in a subtle balance between supply terms and consumption terms that are generally poorly understood and misrepresented in the current climate models. In particular, processes such as the future changes in upwelling, dust deposition, denitrification, as well as the biogeochemical feedbacks and mixing are known to be poorly represented in the current generation of global models, and hence may affect the reliability of oxygen projections for the Arabian Sea, as these processes have been shown to control the dynamics of its OMZ. So to wrap up, I would say that using a set of hindcast simulations of the Arabian Sea, we find a strong deoxygenation signal in the upper thermocline of the Northern Arabian Sea, in agreement with recent observations. And this results in the intensification of the OMZ there. Our analysis also reveals that this oxygen decline was primarily driven by reduced ventilation caused by surface warming, including in the Gulf. And finally, minimum zone in the Northern Arabian Sea in the future, probably because of reduced wintertime convection and also reduced subduction of the Gulf water, as well as a weakening and potential shrinking of the OMZ in the southern and central part of the Arabian Sea because of the enhanced ventilation of the thermocline there. However, important uncertainties characterize these projections because of the poor understanding and representation of key processes that have been shown to control the dynamics of the Arabian Sea OMZ in the global climate models. And I just would like to add that the topic of the future of the Arabian Sea oxygen minimum zone will be further developed and discussed on the basis of regional downscale simulations by my colleague Parvati Valivattilam on the Friday morning. And with that, I would like to thank you all for your attention, and I'll be happy to take questions if there is. Thank you.
Thank you, Zohair, for this great talk. So we have one question there.
Thank you, Zohair, for the nice talk. I was wondering, you were showing the time series of the oxygen, ventilation, and oxygen, and there was, and you say there is a strong trend, negative trend, but I also noticed at the end that it was like a kind of increase. When I'm just curious because I don't know the Arabic seas, but when I see something like that in the North Atlantic, I would think to compare that for the North Atlantic Oscillation to see if there is some natural variability. Is there something like that also in the Arabic seas or not?
Yes, thank you very much for your question. Indeed, there might be a stronger component of the trends that I presented may be driven by natural variability. And we know that there are oscillations that affect the circulation Indian Ocean, the IOD dipole, as well as ENSO. So it's very possible that, and we see it very clearly in the time series, that it is very strong interannual and potentially also strong decadal variability in this time series. And as you notice, the study period is quite short. So, I'm not claiming this is really driven by at least entirely by the climate change perturbation. So I think that possibly there is at least partially there is a signal from the natural variability there, but we haven't looked at that.
Thank you, Zohair. Is there another quick question? Quick, Lisa.
Yeah, okay. I know the Northern Arabian Sea upper boundary of the oxygen minimum zone is highly seasonal, driven by the monsoon, and it's oxygenated for some of the time and not for other parts of the year, with a big effect on fisheries and so on. Do you think that seasonality will change, or can you resolve that, because it's so important for the survival of the shelf animals?
Thank you for your question. So in the model we have, of course, we resolved the season cycle. We haven't looked at, especially for the hindcast simulations, whether there was a change in the seasonality, but I think it's an important question. Thank you for asking this. So this is something that we probably have to look at.
Okay, thank you, Zohair. We'll go on for the last talk of the morning. It's Salman Tarek, and he's here, and he will talk about long-term trends in chlorophyll A and sea surface salinity in Arabian Sea. Yeah. Okay.
Paper. I will talk about long-term trends in chlorophyll A and sea surface salinity in Arabian Sea and their inter-association with aerosol nutrients. Climate change and ocean warming has a great impact on physical ecosystem in which the phytoplankton grow. Modifications in the vertical stratification and advection of ocean currents also impact by the phytoplankton and the sun rays coming and the solar rays. By using in situ measurements and long-term satellite data, the response of marine ecosystems in the global ocean have been extensively studied over the past 2 decades. Vital nutrients have been supplied by the phytoplankton through atmospheric deposition that serve as a base of nutrient food web. Around the globe, nutrients have been carried in plume either through the emissions from the desert dust, biogenic debris, volcanoes, wildfires, and vehicular and industrial pollution. In this study, we aim to investigate the long-term trends and spatiotemporal variability in chlorophyll A, sea surface salinity, and their association with aerosol nutrients in the Arabian Sea. Also, the correlation of chlorophyll A and the sea surface salinity and aerosol optical depth, angstrom exponent, and dust angstrom AE will be determined. The data, we use data of chlorophyll A, AOD, and AE from the MODIS, and the data of sea surface salinity have been acquired from Aquarius. Dust aerosol angstrom exponent has been obtained from MERA2 model. And the spatial resolution of chlorophyll A is 4 kilometers, while the resolution of dust A is 0.5 degree into 0.625. The spatial resolution of MODIS derived AOD, sea surface salinity, and AE is 1 degree. Ocean chlorophyll A is a key property to assess photosynthetic burden. Spatial distribution of chlorophyll A has been shown in this figure. It can be seen that we obtained high values of chlorophyll A near the coast of Pakistan, Oman, and part of India. Salinity profiles are the key factor in controlling ocean currents and density. Spatial distribution of sea surface salinity over Arabian Sea has been shown. A latitudinal shift has been observed that mean as we move upward from lower latitude to higher latitude, the value also increases. Evaporation of ocean water and formation of sea ice, both increases the salinity of ocean. However, the salinity raising factors are counterbalanced by the inflow of fresh water from the rivers, rain, and melting of the ice. Aerosol optical depth is a key parameter to assess total aerosol burden. Aerosol optical depth has been shown that is derived from the MODIS. It can be seen that there are 2 major hotspots near the Arabian Sea. One is close to the coast of Pakistan, near the city of Karachi, and the other one is near the Oman. And a little higher, comparatively higher values are also obtained near the coast of India. And it can be seen as the as one moves towards a lower latitude and open ocean, AOD decreases. This is dust angstrom exponent. Low dust AE and high AOD indicates dominance of dust aerosol along the coast. It can be seen that the blue color represents that the dominance of coarse mode particles, means large size particles are present on the coast of Pakistan, Iran, and in the Gulf countries. While one moves towards lower latitude, the aerosols become smaller in size. This is basic statistics of the parameters used in this study. It can be seen that we obtained a negative percentage change for chlorophyll A, sea surface salinity, while positive trend is observed in AOD and AE. We have also correlated the parameters. The first map is showing that the AOD, that the hotspots of AOD are maximum and showing minus 0.98. The value of dust AE is minimum minus 0.98. That shows that the dominance of dust aerosols, while rather than the hotspots, there exists a major chunk of Arabian Sea where the independence of their variables are also observed. The mean AOD and AE of 0.390 and 0.7. Overall percentage change of 11.02% and 0.60% are observed during the study period over the Arabian Sea. This image shows a correlation between chlorophyll A and sea surface salinity. We found a good correlation near the deep sea between the sea surface salinity and chlorophyll A. Although sea surface salinity is negatively associated with chlorophyll A, but I think there would be a kind of plant that are more tolerant towards sea surface salinity, or there would be more effect of another parameter that is increasing chlorophyll A. Trends in the variables has been shown. The time series of chlorophyll A, angstrom exponent, AOD, and dust angstrom exponent. A generic trend is observed that during the peak values of AOD, low values of chlorophyll A is observed in the deep oceans. With the passage of time, a decreasing trend has been observed for chlorophyll A and sea surface salinity. AOD and A is showing an increasing trend over the time, while no change is observed during for dust AE. The highest peak of chlorophyll A is in the year of 2004, while the lowest in 2007. In this study, long-term trend in chlorophyll, sea surface salinity, and aerosol nutrients have been observed by spatial number correlation analysis. A high positive correlation is observed between AOD and sea surface salinity along the coastline of Arabian Sea, and the positive correlation of AOD and dust AE with chlorophyll A is observed along the southwestern and northwestern coast of Arabian Sea. Low dust AE and high AOD indicates dominance of dust aerosol loading along the coast. The mean AOD and AE of 0.3 and 0.7 with the overall percentage change of 11.02%, 0.60% are observed during the study period. An overall increase trend of AOD and AE has been observed. The mean chlorophyll A and sea surface salinity of 0.65 and 0.20 and 35 point, if I can read it, 6.52%. Chlorophyll A and sea surface salinity of 0.656 and 35.85 is observed during the study period, with overall percentage change of, as I told earlier, that we found a negative percentage change for chlorophyll A and sea surface salinity. The percentage change shows an overall decrease in surface salinity and chlorophyll A content. For more in-depth analysis of chlorophyll A variability, longer time period data is required with other variables, like more variables from climatological data and meteorological data. Thank you.
Thank you very much for this nice talk. We have time for one quick question. Yes, Erica?
Yeah, sure.
Hi, thank you. I have a curiosity question about how we can connect these results to like macrofauna. So for example, are you familiar with in these areas where you see high nutrient input from, say, dust, do you see changes in fisheries productivity?
There's a good point that we should have discussed, but we have used just remote sensing data to explore the relationship between aerosol nutrients and chlorophyll A, but it can be the topic of further research.
Thank you very much for all the speakers from this session.
So, like I said, I will be presenting my very preliminary results of my work here. In the morning session, Dr. Sohail Akhar before me already explained the Arabian Sea OMSEs very well, and I'm thanking him for that. Okay. So Arabian Sea oxyminimum zone is a very interesting region to study about it, because it have a very large vertical extent of hypoxic water among all other world ocean OMSEs. And we expected to expand this Arabian Sea OMSEs in the future due to the climate change effects and global warming. Like Sohail sir said in the morning, The Arabian Sea is located in a landlocked position, so the ventilation will be low. So my work is based on a model. I have used a coupled physical biogeochemical model, which is known as hybrid coordinate model, and ECOSMO model, which is a coupled model, HICOM ECOSMO. And I have simulated the deoxygenation from 2000 to 2020 in Indian Ocean. The HICOM model is a physical model which is using hybrid coordinate system, and the ECOSMO is an ecosystem model. In the flowchart, you can see the major processes involved in the ECOSMO model. And this is the study, this is my study area I have modeled. The first one depicting the topography, that is the red highlighted region is my focus, that is Arabian Sea. And the next picture is showing the model resolution. In Arabian Sea, we are getting 10 to 20 kilometer resolution from this model. So I have forced this model with some atmospheric forcing data that is derived from ECMWF ERFI reanalysis data, that is a 6-hour resolution, temporal resolution. And the variables I have used to force are the 10-meter wind components, then air temperature at 2-meter height, then dew point temperature, cloud coverage, and precipitation data. Additionally, we have used river runoff data from ERFI also. So in the coming slides, I'll be explaining the validation of our model that we have done with the World Ocean Atlas 18, and we have showing our validations in cross-section plots. In the first figure, you can see the sections we have taken. And secondly, we have looked into the variability of OMS at core boundaries, and that is wherever the dissolved oxygen is below 20 micromole per kg. That is what we consider as OMS at core. Then we have computed the volume of OMS at core from the area and thickness of water. Then for the analysis, we have divided the Arabian Sea into 4 regions, that is Northeast, Northwest, and Southeast and Southwest. That is, you can see in the plot below. Then we have used some seasons. We have taken seasons as December, January, February as Northeast monsoon, then pre-monsoon season in March, April, May, then Southwest monsoon as June, July, August, and post-monsoon is September, October, November. That's a common season we have taken. Then we have used correlation analysis to find the relationship between the variables. So in this slide, you can see the validations of oxygen, dissolved oxygen, and nitrate. The first layer of plots depicting the validations of oxygen, and you can see there is no much difference between our model and World Ocean Atlas as per the climatology. And this white contour lines depicting the OMS at core boundaries. So our model is well reproduced the profile of oxygen as well as nitrate you can see in the below layer plots. So in this slide, I will showing the validation of temperature. In the temperature plot, we are also getting similar results with the World Ocean Atlas. Then moving to our study on variability of core OMS boundaries. So we have noticed that the upper boundary of OMS is varied between 90 to 200 meter depth and in the Arabian Sea as per the climatology analysis. Then we have noticed a deepening trend. As you can see here, there is a deepening trend of OMS core boundaries from south to north. So the thickness of OMS is showing a meridional variability than a zonal variability.
So there is a thicker, we can see a thicker part in north while a thinner part in south. So we have computed the climatological values, and we have plotted them to show the seasonal variabilities. So here you can see the dissolved oxygen as well as nitrogen is very much similar to each other, and they're showing us clear seasonal variabilities because during the monsoon season, we have a lot of nutrient inputs, and in winter season, due to the deepening of MLD, we are getting this dissolved oxygen values in depths also. So moving to the volume of OMSZ core, we are getting this volume in between a 2.1 million meter cube. But as per the World Ocean Atlas, it is 1 million meter cube. But it is comparable with less bias. So the internal variability of dissolved oxygen. So in the Southern Ocean, we are getting the boundary of OMSZ in 400 to 800 meter depth, while in the northern parts, we are getting 900 to 1,500 meter. So the deepest lower boundary was observed in the northern Arabian Sea, while the shallowest observed in Southern Ocean, Southern Arabian Sea, sorry. This is because in the Southern Arabian Sea is highly ventilated due to intense underwater masses and Persian Gulf water masses. Here you can see some statistical analysis. There is correlation we have done with nutrients versus primary productivity versus oxygen. The surface nitrates are highly positively correlated with the primary productivity, while the surface nitrate is also positively correlated with surface oxygen. Because whenever the nitrate is higher in the surface, it will enhance the primary production, and surface oxygen will be more. And you can see the surface nitrate eventually leads to the decrease of oxygen in the OMSZ depths. So we get a negative correlation there. So in this slide, you can see a clear warming trend effect on this model variables. And all the regions that in the Arabian Sea, the SST is increasing, while the surface productivity is also seems to be increasing, because the increasing surface sea surface temperature in Arabian Sea draws so much of southwest monsoon season, and it enhances the primary production in the Arabian Sea. So this enhancing primary production eventually leads to the decrease in surface oxygen in the Arabian Sea. So some more evidence of warming is the looking to stratification. We have computed the stratification using Brand-Weierstrahl frequency, and you can see in the Arabian Sea the stratification is increasing over the time, and the correlation between the surface oxygen between the SST is highly negatively correlated. So what are the climatic events influence on oxygen, dissolved oxygen? So we just compared with so we have listed out the main events years, that is El Nino, La Nina, and positive IOD and negative IODs. So we compared with our dissolved oxygen data with what happens with these years. So we found out that whenever the El Nino is El Nino and positive IOD is happening in the Arabian Sea. The surface SST, that is sea surface temperature, will increase, and the dissolved oxygen is very less in our water. So we have to explore more about this climatic events effects on Arabian Sea. So coming to the conclusion, the model outputs were validated with the World Ocean Atlas, and this model is never been used before in our Indian waters. This is the first time we have been using, and we have shown that our model is capable of producing most of the trend of biophysical and as well as biogeochemical variables, and the model lacks some capability of explaining DO trend in Southern Arabian Sea. So we are suspecting because of the lack of bacterial processes in the model. So deepening of lower boundary of OMS was found from south to north latitudes. So the deepest lower boundary was observed in the northern Arabian Sea, while shallowest was observed in the southern Arabian Sea. So we have noticed some warming influence on the dissolved oxygen as well as, and we have looked into some El Nino and positive IOD effects on dissolved oxygen. So for now, we have observed the El Nino and positive IOD caused a decreased surface dissolved oxygen in our Arabian Sea. So thank you all.
Thank you, Akash. Any question? There is time for 2 or 3 questions.
Hello. I was wondering, you said that the model was lacking capability to reproduce the DO trend in Southern Arabian Sea. Yes. Do you have any clue or thing? Why is that?
Yes, because we are suspecting that we are lacking some bacterial influence in our ecosystem model. So we have to add some more bacterial activities.
Question from the audience or at distance? Yeah, Andreas? Okay.
Yeah, thanks a lot. I was impressed by the pretty good agreement with observations. I wanted to ask first, how long did you spin up the model? And what do you do with the marginal seas and the slope currents affecting the Arabian Sea central waters?
Yeah, the spin-up time was for 8 years for this model. So right now we are not looking into the currents effects because we are lacking some time. And this is our only preliminary results of our work now. So we will be looking more into the future. Yeah.
Okay. I have just a quick question. So I have seen from one poster that the evolution of the OMZ depends on the threshold concentration that you choose. You choose 20.
Yes.
Did you try to choose also threshold concentration? So for instance, lower than 20 to see if the trend is the same.
Right now we have only looked into the OMS core, that is 20.
Yeah. Okay. Yeah. Thank you. Yeah. Thank you so much. So now we move to the next speaker. So Alexandra, she's at distance. Okay. Hello, Alexandra.
Hello.
So, hello. Nice to see you. So we see you very well. So maybe we will see you in big format. Yeah. Okay. So hello. So we speak about oxygen deficiency index for forecasting oxygen deficiency in coastal areas.
So exactly. Thank you very much for inviting me. And I'm very pleased to see a lot of old faces there and to see a lot of people I haven't seen for a long time, even if it's just from the distance. So what I'm talking is about the improved oxygen efficiency index, shortly ODI, for forecasting oxygen deficiency zones in seasonally stratified coastal analyses as one part of the InfoWAS project. The InfoWAS project is a joint project of the Alfred Wegener Institute and the Federal Maritime Hydrographic Agency, where I belong to. And I'm very happy and appreciate it highly that we have Fabian Grosse here with his expertise on the ODI, and he is from the Federal Institute of Hydrology. Welcome on board. Okay, so I will give you shortly an overview of the InfoWAS project, which aims to develop an information system for forecasting potential harmful algal blooms and oxygen deficiency zones in the near future. It further should inform environmental agencies and the public about potentially upcoming harmful events, which then could act in advance in order to diminish environmental and economic consequences. So for that, we use remote sensing data, we use in situ data and forcing data for the model. Remote sensing data, satellite data, and in situ data are integrated in a parallel data assimilation framework, which is part of the AVI, and the ecosystem model is the operational ecosystem model at the BSH. It then should create daily outputs, for example, oxygen, temperature, and salinity, which jump into a toolbox with functions who calculate, for example, the oxygen efficiency index, which then the outputs are fed into an information system on various platforms. In order to do so, it was the wrong one, sorry, wrong direction. I apologize, I really have a very long delay with my internet. Okay, so just to shortly introduce you, we use the operational model at the BSH. It's the Huron-Bose model, which is coupled to the biological ergo model, and it has a coarse and high resolution. And the parallel data assimilation frameworks at the AVI is then coupled together with the operational model in order to give a higher accuracy of the model outputs. What we define here is the oxygen efficiency index, so forecasting potentially harmful oxygen efficiency zones. And the ODI ranges between 0, which should then represent the low risk, and a high risk of development of oxygen efficiency zones. This ODI is modified after the original developed oxygen efficiency index by Grosse et al. In 2016, which was applied for North Sea and used the EcoHam model. And the main differences to the original OD we do have in calculating a mixed layer depth, which we use now the formulation after Miero and Huyang in 2009, which is based on seawater density, so thus a function of temperature and salt, in order to better constrain and consider both regimes, the North and the Baltic Seas. Without any mathematics, because they're quite heavy, the ODI is a product of 3 different indexes. And I will show you results now, our first results, which are taking into account a reference period of 30 days, so hindcasting 30 days back for a daily output of an ODI calculation. And all these results I'm showing you are without the data assimilation. So the indexes consist of a stratification index, which is basically given by the longest continuous stratification period. The depth index, which is representing the sub-water volume below the mixed layer depth, with an assumption that with high primary production and sluggish ventilation, with the lower water volumes or smaller water volume you have there, oxygen can be consumed quicker and is more prone to develop an oxygen deficiency zone. And then we do have this net primary production index, which can be used as a proxy for particulate matter and sinking out into the depth, which then can be used up by biogeochemical processes. It's depth integrated, And we do have a mean of a reference period, and we are testing now minimum and maximum net primary production thresholds. Shortly to show you a graphic, what we are doing, we are checking a reference period here now for 30 days, especially we are checking the last 10 days prior to day 0, which is the daily output, in order to check if we did have a stratification in the last 10 days or not. Then we're calculating the depth index with the subwater volume of the MLD, and we're combining it with the net primary production index, so we get results of the ODI. And how that looks in detail, I show you the graphics here. At the very top left, we do have the stratification index, and as I said before, it's without data assimilation, and I just show you 1 day. So red means it's very likely that an oxygen deficiency zone can develop, because we do have stratification here. The same is with the depth. The reddish it is, the shallower are the sublayer depths of the MLD sublayer. And the same on the bottom left with the net primary production index. The higher the net primary production is, the more reddish it is, and the more likely we do develop an oxygen deficiency index. All these 3 indexes together give one output on the bottom right, which is the oxygen deficiency index, ranging from 0. It's very unlikely or low risk to develop an oxygen deficiency zone. And whilst it goes over yellowish to greenish to yellowish, orange, and red, the risk is higher to develop an oxygen deficiency zone. So what does that mean? Or do we have anything to compare with? Yes, we do. Basically, on the left-hand side, this is our output. It's just daily output, because that's what we get, and we will feed into this information system. We compare it to the historical output of Grossi et al. In 2016. He uses the index in 2002, which is picture number A, and compare it to 2010, picture number B. Although you can see differences in the intensity of the index, the shapes are really similar in 2002 and 2010, and I think they also agree very well with our newly defined oxygen deficiency index. But how does this really relate to oxygen? And it's always the question, do we use saturation versus bottom oxygen concentration? So at the very top image, this is our output showing the oxygen efficiency index and the likelihood if it will develop or not. And at the bottom, we show bottom oxygen saturation. And on the bottom right, I show bottom oxygen concentration, and I was really surprised how good it works with the bottom oxygen saturation, and how good we can see the dynamics of it. It also works quite well with the bottom oxygen concentration, but here I think since we are using the MLD and we're defining it on temperature and salinity, and as well as bottom oxygen saturation is a function of temperature and salinity, I think it makes sense that they better can be compared, and I probably will use that also in the future for comparing. However, there is still a lot to do. So as the results are really fresh, but we are thinking to get into more detail and better constrain the outputs. We will think to constrain better the net primary production thresholds. Maybe we should separate for the North and Baltic Seas. And also the first output shows that the oxygen efficiency calculation could be rethought in order that we are not using multiplicative approach, or we compare it to the additive approach and see which one is better. Moreover, we might think about to take into account longer reference periods. However, these are just cosmetical operations, because what I found out in the last 3 years working in database management and geodata information system administration, I found out that it's really, really important that you do have a system where you can immediately manipulate things, where you can immediately impose changes and then basically upload the data somewhere without shutting all servers and just creating really a lot of inconvenience to the entities. So what we did, I call it now the ODI toolbox for this information system, InfoVAS system. It is programmed in Python and uses basic packages. Moreover, the code runs on all common Python operation system. It doesn't matter if you use Windows, Linux, Unix, or whatever. We use output files of models, and here we use NetCDF files. However, they can be of any kind, the output files, to feed into the toolbox, into the function, as long as the file format is Python editable. And moreover, we also create output files. Now we create NetCDF files, for example, but as long as the file format is Python editable, we can basically create everything and we can very quickly modify the code. Which means also this toolbox is independent of model versions and model types. And the output products we get, NetCDF files or GeoTIFFs, which are georeferenced pictures, raster images, they are very easily to process with open source GIS applications, which are widely used in public entities. And moreover, they're really graphical. And I think that's very important, because the aim of the InfoVAS project is to develop an information system for water quality, which is easily accessible, applicable, and understandable to different entities, stakeholders, and public, and then it comes into citizen science. So what we are doing now in the last phase of our project, except then calibrating a little more and getting some cosmetical corrections on how to make it quicker, faster, and easier, we have to check where our output should go. And one aim of the project was to upload our outputs on the Copernicus Data and Exploitation Platform, shortly DE. Or we also would like to put it on the Geo Information System at the GeoC portal at the Federal Agency, Maritime and Hydrographic Agency, at the BSH. So in order to do so, I will rely heavily on a lot of experts, and I would like to thank all the guys from the IT, my colleagues, and everybody else, even though some of my project colleagues I have never seen in person due to home office restrictions. And I would like to thank the audience. And well, do you have any questions?
Thank you, Alexandra. Any question, Mitya? There is one there.
Hi, thank you so much for such a great presentation. I like to ask on the overview of the InfoVAS that you presented at the beginning. I would like to ask, what are the barriers or the challenges that you think that is still very challenging to face in order for us to move forward from the forecasting your potential to all the way to the public, to inform the public, all the way to the act in advance as you first slide. Thank you.
Thank you very much for the question. And yes, as you're right, it's very challenging because we have to consider a lot of things. For example, integrity. We have to consider open data. We have to consider open source licenses. And of course, once you're dealing with data which come off federal agencies, and they will be fed into the public, you have also to take care what is about the critical infrastructure and critical data. So this has to be defined. Grateful that I have a great team of experts behind me and I don't have to go this way alone. And still, I'm very positive because over the last 2 years, I see a high trend that things are getting easier and that we really made a lot progress and a very large jump into digitalization. I'm very grateful to that and thanks to all the IT
Thanks,
experts.
Alexandra. I guess that we have an online question. So we will take it. Just a few seconds.
I see it. I think it's the ODI toolbox available on GitHub.
Can you ask the question online? You can ask, it's in the
It's in the chat, I think it's from Kate.
Yeah.
Is the ODI toolbox available on GitHub? Well, in the moment, not, because we first have still to make it a little bit more user-friendly. And then, of course, also we have to see how the open source licenses and open data policies will then kick in, and we have to define this. But there's still some process to make. But I hope in the future it will be.
Okay, thank you, Alexandra. Thank you again.
So I finished. Thank you very much.
Okay, thanks. So we move now to the next talk, which is also online. It's Liam Fernand. Yeah, hello.
Hello, good
afternoon. And yeah, we can see you and we can hear you very well. Okay, and then we will speak about observation, optimization of observation network to improve ecosystem forecast.
Okay, thank you. Yeah, and thank you very much for the opportunity to present to you. Yeah, I work at CFAS, which is the UK Marine Government Advisors. And the reason for mentioning that is that as such, we fit on the scientific evidence and policy interface. So what I'm talking about today is perhaps less technically detailed than the presentations you've seen before, and more how to implement it and how to make things happen. I'm hoping that will. Oh, hang on, I thought I'd clicked. Right.
You should be able to change yourself now.
Yes. Oh, there we go. Fine. Right. Yes. Yeah. I'm grateful to Alexandria for her moving away from the Arabian Sea and into the Northwest European Shelf. As previous presenters have mentioned, then oxygen isn't a problem in its own, and certainly in seasonally stratified seas, then eutrophication and links are an issue. So one of the mechanisms for addressing these issues is, and it's a grouping of about 15 countries that have grouped together and form assessments for various substances, including hazardous substances and eutrophication. So they're considering dissolved organic nitrogen, dissolved organic phosphate, and as level 1 indicators, and chlorophyll and oxygen as secondary indicators, and they're the ones which drive assessments. So the results on the screen are just very preliminary, but they are combining assessments, but I want to focus on oxygen. So the oxygen status, and this is taking all the oxygen measurements that are in the ICES DOME database and looking at the bottom 5th quartile of near bed measurements. And when you categorize them into bad, poor, moderate, good, and high status, where the boundary between good and moderate is 6 milligrams per liter, you get this map. And as you can see, I'm not sure if I can, if you can see my cursor. Okay, yeah. So the area to the east of the Dogger Bank is categorized as being moderate. Areas in the Kattegat are moderate, and areas off the Loire and Gironde Plume are moderate. Okay, that's fine. And therefore, areas in the whole of the Celtic Seas don't appear to have a problem. That map, and many of you seen maps from Diaz and Rosenberg, or more recent ones showing areas around the whole of the UK as being hypoxic, is quite a contrast to that. What's the data that it's built upon? On the right-hand side is the data that map was built upon. When you grid grid them down to about, I think they're 12 kilometer squares, 20 kilometer squares. And you can see that actually there are very large areas where we actually have no data at all in the 2015 to 2020 assessment for near bed oxygen measurements. The left-hand map is effectively the same plot within the 200 meter contour for the whole of the data sets in ICES or the BODC between 1920 and 2017. So there are very good areas of high coverage in the German Bight and southern North Sea, and becomes much more sparse when you come out into the Celtic Seas and even down to the French coast. This lack of data is relatively obvious and is recognised at OSPAR. So there are developed tools to assess not just the status, but also the confidence in that assessment. And there are a number of different ways of generating confidence and then combining them. So as you can see, that the overall status is that there's high confidence associated with the assessments being undertaken in the North Sea, but very low confidence with those associated in the Celtic Seas. Okay, so how do we get around this? And can models help? Well, the obvious answer is yes. So this is results from a paper by Stefano Civiata, one of my co-authors. And this isn't just one model run. This is an ensemble run of 100 models. Well, one model run 100 times with different starting conditions, slightly different starting conditions. It's the Ursem Polcoms model at about 12 kilometers resolution. So that is quite coarse, but it does mean it runs quite fast, hence the 100 model ensemble. But as you can see here, so what we're seeing on the left here is from the 1998 to 2009 average period. And you can see there are quite significant areas, but certainly beneath 6, and possibly even get down to say 4 milligrams per litre. And the plot on the right-hand side is the certainty in red of the minimum values and how often they were recorded in that 100 model ensemble. So if it was red, then it meant that there was a value beneath 6 in every single one of those 100 ensemble runs. So that plot has certain very similar in certain parts of the German Bight to the observations, but that lack of observations is the real issue in the Celtic Seas. Do we believe the model? So the plot on the left-hand side is the small plot here is from Stefano's paper. There's a reasonable line, and most of the observations are lying. The model comparison to observations lies along that line until you get to higher values. So some of the observations give much higher values than the model. The model appears to have a top upper limit. There is still quite a reasonable spread down here. This isn't a very tight line. The right-hand plot here is from a benthic benthic lander at this site. There's a little dot down here. The model are the blue solid lines for the bed and dotted line for the surface, with observations at the surface and at the bed. So the ones in red are at the bed. And you can see that the model is tending to sort of overestimate the amount of oxygen, but it's not doing too badly. But it's not perfect. And there's some other features which are so can we believe believe the models? Well, in many ways, we don't have any choice because we don't have observations everywhere. And it's about how we wish to quantify that difference and accept that uncertainty. What about better observations? Well, gliders should be able to help with this. Gliders are great in that they give you vertical coverage, they give you spatial coverage, and really quite good temporal coverage. So these are a couple of observations from the central North Sea, right here, and also from a more recent AutoEco project, where gliders were maintained out in the central to northern North Sea for a whole year. On the plot on the right here, but these are expensive, and typically these programs, which are research programs, tend to cost quite a lot of money. In the UK, we only really have one operational, as you might call it, glider program, which is continuous, which is the Samdron Ellet line. And that's costing about 120k a year. And that's only for a glider operating for about 140 days a year. So they are quite expensive. We also have a bit of an issue with effectively what we have at the moment, which is a gap between technologies. So we've heard a lot about the issues with simple samples from bottle samples, and the fact that they don't get the right resolution, they miss the very high the effect of the high frequency events and so on. But equally, cruises are being reduced in the UK, and we also have issues with the amount of time it takes to get data from sensors from gliders and moorings validated, calibrated, and into data centers. So the data centers can take them. There's a lot of data that we collect, not just from oxygen, but from things like barometers, which doesn't actually get into the data centers at all. So we've got to sort those problems out. Right. So what do we do about this? And one of the solutions would be to get, in terms of the OSPAR assessments, is to get models accepted as evidence. And there's been quite a lot of work on and progress on this. Herman Lennart at Hamburg has led the ICG EMO group, which is bringing together a group of ecological modelers and using an ensemble and bringing those together in quite clever ways, observations. And they've been used to set the thresholds for nutrients and for chlorophyll for the next assessment. But the next step on from there is actually to get them used and accepted as evidence themselves, in which case the oxygen gap could be filled, the lack of oxygen measurement could be filled. And that would help with some of the issue, well, a lot of the issues, but we do need to make sure there's confidence in those models. Right. The other part two of this is a proposal for a better network. Again, oxygen is measured in isolation. We need to think about measuring it associated with nutrients and chlorophyll. So part of the elements in the UK is having to justify why. What is it that giving a better network would do? Scientifically, that's obvious to people, certainly this audience, but beyond this audience, it's having to justify what are the benefits. And if you do have a better network, how do you get greater confidence? So what we've been doing is we can simulate better data into systems, and then when you propose a new network, new moorings, new glider lines, what that gives and how that increases the confidence you have in any statement you make. And therefore, you can then come up with a system which is effectively cost effective in relation to the confidence associated with your assessments compared to how much it costs. Yeah, thank you. That's all I wanted to say. Only other thing would be, I would be delighted to hear from people that have managed to develop networks and combine them with observation, combine them with models, and solve this problem of convincing funders to pay for them. Thank you.
Thank you. Time for one question. Done. No, done. Please.
Thanks. Yes, I have maybe a comment question. I, for one, am a little skeptical about accepting models as evidence. I think one has to be really careful about that. And in my opinion, it's kind of a slippery slope. You know, the adage, all models are wrong, some models are useful. And I think that one really has to be careful before accepting models as any kind of real evidence.
Yeah, if I may just briefly come back. I agree with you. George Box's quote is one that I try to live by. It's making them useful. So, I mean, the issue with oxygen is that unlike other parameters, you can't measure it from satellite. So you're always going to have this spatial problem. So in areas where we have no or very limited data, what are we going to accept? So I would prefer to be saying we need to quantify the errors in the model and say that our error is this much, or whatever that much is, and have that acceptance, rather than otherwise you're right. You can drive down a route where you don't need observations, which is completely not true. You absolutely need the observations, but we're not going to get observations everywhere.
Okay, thank you. So the best is to combine both observation and models. Yeah, thank you so much. Yeah. So now we move to the next speaker, Bachar Guo. Yeah, sorry for the French pronunciation. Okay. I hope I didn't not too bad. No, it's okay. Good. So oxygen utilization rate differs from true respiration rate in the mesopelagic zone. And you are from DOMAR.
Yeah, exactly.
And I have heard that it is your first year of PhD, so congratulations.
Thanks. I hope you can hear me clearly. Yeah, good. Check. Good afternoon, everyone. I'm Hachou, and I'm from GIUMA. As the first year PhD student, I'm very honored that I can have a chance to give a talk here in this very nice conference. The topic of this talk is oxygen utilization rate differs from true respiration rate in the mesopelagic zone. We know that from observations, global ocean oxygen inventory decreased by about 2% in the past 5 decade years. But models only show just half of observed oxygen loss. And that means perhaps we still lack the quantitative understanding of deoxygenation mechanisms. And in this figure, I show some mechanisms which will lead to the oxygen loss in the ocean. And you can see that respiration processes are involved in most of these processes. Therefore, respiration is regarded as engine of the oxygen deoxygenation processes. And unfortunately, the oceanic respiration observations are very sparse, perhaps because it's very hard to measure, and it really limits our understanding on deoxygenation processes. And to improve the model performance, to give better projection, but what will happen in oxygen, we really need more oceanic respiration observations. And there are several ways to measure the respirations. Here I introduce a method called oxygen utilization rate. We call it OUR. And to calculate OUR, we first calculate apparent oxygen utilization and which is just a difference between saturated oxygen concentration and the measured oxygen concentration. And then the OUR is defined as changes of AOU divided by changes of age. And then the age of water mass is just defined as time since the water last contact with the atmosphere, which means since the water have some air sea gas exchanges. And in the real ocean, the age of water usually derived from CFCs and some radioactive tracers. And in the models, we usually use IDH. But every method have uncertainties, and so as OUR as well. But it's very hard to evaluate the OUR the accuracy of OUR method, since in the real ocean, we have very sparse observations, but that is not the case in models, since we have exactly the true respiration rate, and also we can calculate OUR in models as well. And here I use a model called FOSSIMOPS. It is an Earth system model. It contains atmosphere, ocean circulation, land, CS, and biochemical processes. And the resolution of the model is high as well. We have half-degree resolution. There are 46 layers. Here's just how one chose research sections. And here's the distribution of IDH, AOU, out curves, distribution on different potential density surfaces. And out curve is just a region where potential density layer is shallower than the winter mixing depths. So the air sea gas exchange can happen. And I chose a region where the isolines of H and AU are parallel, and I select a section which are perpendicular to these isolines to avoid any, as much as possible, the lateral mixing. And also I chose the section with different biogeochemical features and dynamics. You can see, sorry. Yeah, I chose one in North Atlantic section, which we don't have impacts from upwelling system, but we also chose one in the tropic Atlantic, which you can see these tone-like features, which means we have impact from upwelling system. And just for convenience, and say this section is North Atlantic, and this section is tropic Atlantic. And here's a comparison for OUR and true respiration rate. And you can see that OUR perform very different in these two sections. In the North Atlantic section, OUR always smaller than true respiration rate, but the difference between OUR and true respiration rate decreased following depths. And the ratios give somehow fluctuations, but overall trend is that OUR and true respiration rate ratio approach 1 following depths. And the tropic Atlantic, in the relative upper part, the upper 300 meters, the OUR is also smaller than the true respiration rate, but below it, OUR is becoming larger than true respiration rate. And the same with North Atlantic part, the difference becoming smaller following depths, and the ratio also approaching 1 as well. So what is the reasons for the description of true respiration rate and OUR method? Here I show the distributions of respiration and out curves again. And you can find that true respiration rate is always higher near out curves. And also at the distance between out curves and research sections becomes longer following depths. Why that lead to the difference between true respiration rate and OUR? Here I give two figures. First, let's discuss the figure on the left. The red curve is calculated OUR, and the black line is imposed respiration. And here's a pattern which I try to explain what will happen if we don't have impacts from upwelling. And you can see that if we have higher respiration near outcrops, we have higher, the AOU will increase dramatically, and then we have higher gradient for AOU near outcrops. And since we have diffusion, since we have diffusion as well, this AOU will lost to the outcrops. So we have less preserved AOU on density layers. Therefore, we have the ratio of OUR to respiration rate is smaller than 1. But if we have upwelling impacts, let's see the figure on the right, then the upwelling will bring lots of age relative AOU, because in the deep ocean we have very low respiration rate, so the age is relatively larger than the AOU. And then we have higher gradient of age here and less preserved age, and therefore OUR and true respiration rate ratio is larger than 1. But we still can see that if we become far from outcrops, the ratio of OUR and true respiration rate also become 1 as well, which means OUR in these regions perform much better. And here summary, OUR and estimate true respiration rate in the upper mesopelagic zone by up to 6 folders, which is pretty large. OUR perform differently in deep mesopelagic zone. Without upwelling impacts, OUR underestimate true respiration rate by factor of about 1.2 to 2.6. But with upwelling impacts, OUR overestimate true respiration rate by factor of 1.6 to 3.4. And the ratio of OUR and true respiration rate approaches 1 for in-depth in both sections, which means perhaps we can use OUR method in some deeper part and use Martin curve to rebuild the respiration distribution in the ocean. And here's the outlook. We try to use OUR. We try to research that whether OUR can reproduce the changes of respiration in the past and in the future. Here, I used RCP 8.5 scenario. And the result is not very well. I mean, because OUR and true respiration rate ratio always changes because it changes of difference distance between outcrops and research sections, and also distribution of true respiration rate and upwelling strands, and also the changes for advection and diffusion, a lot of things. So still far to explain details why we have these differences. And yeah. And we, next step, we will also use RCP 2.6 scenario, and hopefully the OUR can perform better since all these changes will be smaller. Yeah, that's all, and thanks.
Thank you very much. Question? No?
Thank you. I was wondering, maybe you said and I missed it. When you say the true respiration, do you integrate over the Lagrangian trajectory of the water mass in the model, or is it just you layer in the respiration rate at that location in the model?
Just calculate the average respiration rate, simulated respiration rate.
Yeah, but one parcel that is along your section, it has a respiration rate where it's here, but it also the history of that water has seen respiration along the pathway of So do you integrate over the pathway?
No. I just calculate the local respiration rate. So I'm not calculate the
Thanks. There is an online question by Hernan Garcia.
Sure. Thank you so much. Great presentation. I get lost with what you mean by true respiration. What does that mean?
Yeah, so true respiration rate is just a simulated respirations. So we have motors and we have output, which name respiration, and that is name it true respiration. So yeah, sorry for confusion, but yeah.
So the idea was to compare what the model can compute for the respiration with what can be listened for observation or for
I think it's not observations, but only model output.
For the model, but yeah, but the people yeah. Any other question? Yes.
Thanks so much for the talk. And I might have missed it, but can you talk a little bit about the AOUR on this slide? It seems like it's doing a much better job.
Yeah, sorry. In this conference, I'm mainly focused on OUR, but we also do some research about AOUR. But
could you give the definition or? Like,
yeah, it's just the ratio of AOU and the H. It's not gradient, but just the ratio of AOU, and it really represents average respiration from outcrops to your sample location. So yeah.
Thank you. Thank you very much. Very nice talk. So we move to the next speaker, and Daniel. So it's observation in the coastal ocean. So assessment of coastal water quality by monitoring using low-cost instrumentation.
So, Ceci? Okay. So good morning, good afternoon. Everyone. So my name is Anne Daniel. I work for Ifremer in France. I will present you today the works that we have conducted as part of the REC project to develop a low-cost instrumentation to measure oxygen bottom concentration in coastal water. This instrumentation was developed in order to be able to assess water quality in a reliable way. So the context, as mentioned in previous talk, only few bottom oxygen data are available today in coastal waters as in the open ocean. We have also some disadvantage of the traditional on-site discrete sampling, which is too limited to record episodic events or those that are changing rapidly. It requires also a large effort in human resource. And now the oxygen parameters is used as an indicator to assess water quality or ecological status, but many regulation as the European Water Frame Directive, the Marine Strategy Water Framework Directive, Spark Convention. What we need now is it's a high frequency monitoring to assess the extent in time and space of this oxygen concentration. So only high frequency monitoring with low sampling interval, about a minute or hour scale, can capture the episodic oxygen depletion. But the classical mooring system are not so much used because of its costs and its maintenance over time. So for environmental and economic reasons, a new kind of is required to meet the following criteria. So the first one, it should be the cost should be lowest as possible. This instrumentation should be light to be implemented only by a small vessel with 2 operators, and not need oceanography ship. It must be simple to be implemented without scuba divers, and it must be robust to be reusable. And we don't want to have a service buoy to avoid vandalism and to limit the impact of storm. And for the last one, it should be adaptable to allow the reuse of most of the optop sensors commercially available. So we develop a mooring system called a Mastodon Oxygen. This Mastodon oxygen will be soon marketed by the French private company at a price about €2,000. It consists of an electronic compartment placed in a float bore. And here you can see the protective cover in stainless steel to avoid damage and the accumulation of materials on the sensor, for example, microalgae or seagrass. When the protective cover is removed, you can see the frame that works as a ballast. Here, the oxygen sensor, which is placed very close to the bottom, about 10 or 20 centimeters. Here we use the mini-dot sensor, which is distributed by PME, which is an expensive sensor with good analytical performance, but other sensors can be used as well. Here you can see the hop stool that allows the float ball to be attached to the bottom frame. And we decide not to use the acoustic release system because of its cost. So we developed a low-cost release system called Burnwire, which consists of an electronic erosion of a copper wire loop under low voltage. It is a time release system. The recovery date has to be scheduled before the deployment. So here is a short animation, if it works. No. So the pilot of the boat arrived on the GPS point of the deployment station. The Mastodon is launched by simply resetting it, and the oxygen sensor requires data during the entire deployment. So on the recovery day, programmed before the deployment, the electronic device switch on the current for the electronic declaration of the copper wire. It is completed in about 1 hour, releasing the float and the electronic compartment. And the LED placed in the electronic compartment blinks to facilitate its location for recovery. So the buoy is grabbed and brought back on board, and then the frame is brought to the surface by pulling up on the rope. So the Mastodon were deployed in the Bay of Vilaine, which is located in the west part of France in Brittany. So it was in 2020 and 2021. The Bay of Vilaine is a mesotidal open bay under the influence of the Vilaine and the Loire River discharge. This shallow bay has a maximum depth of 30 meters. It is partially protected from the swell of Atlantic Ocean by an arc of peninsula and Iceland as well. So this partial hydrodynamical confinement makes this water particularly sensitive to the contribution of nutrients from the Loire and the Vilaine rivers. So since 1982, hypoxic conditions are observed almost every year in the Bay of Vilaine. Sometimes the threshold of anoxia. This is well known as one of the most sensitive coastal water to eutrophication in France. It has undergone several eutrophication events over the recent decades, mainly due to the high nutrient inputs from the Vilaine and the Loire rivers. And the most visible consequences are so the brown black waters here in 55 in 15. There is also some red waters due to Mesodium rebrum bloom, and other red waters last year due to Lingodium polyhedra bloom. And we have also have some green waters, which were followed by a deposition of sludge, and which induce fish mortality by anoxia. So despite this obvious eutrophication problem, the water body of the Bay of Vilaine is in good status with the WFD assessment. So so we deploy the Mastodon on 6 sites located on the Vilaine Plumes, some of them next to the shellfish farms. It's important to notice that the sediment of the northeastern of the bay here are dominated by silty fine sand, while the northwestern part is rather silty. There is also in the center of the bay a moor buoy that measures biological and physical chemical parameters at subsurface and at the bottom. So here's the first results. So first of all, it works. So you can see here the change, the first deployment of one Mastodon, which has been replaced by another one, and we don't see any cut of the oxygen allure. So as expected, the variation of the oxygen concentration can be very brief on some stations and reach anoxia, for example, here in Kervoyal or here in Lema. And this episodic events can be something like 1 week long. The potential influence of tide have been studied, and we can see that much less very low oxygen concentration are observed during spring, during neap tide, and not during springtime. The low oxygen concentration observed during or after the fresh water in the bay, when the low oxygen concentration observed in July 21 are associated with these freshwater inputs in the bay. And here you can see the salinity observation done by the MOLIT system. Interannual difference are observed in the dynamic of oxygen concentration measured at the station, and this can be related to the amount of phytoplankton burn areas which is produced in the bay. You can see on this satellite chlorophyll observation that there is a spatial heterogeneity on the bay, from the west to the east, and as well as a temporal heterogeneity from 1 year to another one. If we apply the metric usually validated by the regulation, which is 10 percentile of all the measure value, we observe that all the stations except one, here Basse Normande, are classified in good status, whether we use a significant number of data, the high frequency monitoring, or not with the low frequency monitoring. So we can say that this metric is thus not adequate to highlight the importance of this episodic hypoxic event. A metric that could be interesting to apprehend the distribution of this high frequency measurement could be the number of low value measured every year during the period of risk of hypoxia. This metric allows to separate the station that are subject to episodic events. So here in Saint-Jacques, Ménarway, and the station which no episodic events are measured. Another interesting metric is based on the duration of these hypoxic events. So here you can see that in 3 stations, the status will be moderate, and in 2 stations, it is a good status. The metrics that we need to work on will be a combination of this 3 metric, so the 10 percentiles, the frequency of hypoxic events, and the duration of this hypoxic event. So the importance of any of these metrics should be considered in the light of high frequency measurement in other areas, subject also to episodic hypoxic events, just to fix the threshold value. So in conclusion, so it is now possible to increase the acquisition of spatial temporal oxygen data in coastal area with a limited budget. The high frequency observation provide a greater understanding of the dynamic of the bottom oxygen concentration, are essential to supply models to understand the complex interplay of the physical and biogeochemical factors, which control the process of the epoxy development. Threshold value applied by many regulations to assess water quality statute need now to be reconsidered to take into account the fact that oxygen depletion event may be very brief. And future work, because it's just the beginning, the Mastodon system will be commercially available soon. And we want to extend the Mastodon monitoring along the French coast, even in areas that are not yet suffering from IOSA, but maybe in the near future. We can also imagine a citizen participation with the sailing school, the yacht club, but this is a lot of coordination. And we also want to improve the calibration of the sensor, allow oxygen concentration, because the Winker calibration is not possible due to chemical interaction. And we want to send all this data to database to improve the results of the models. So thank you. Thank you very much.
Thank you, Anne. Time for question? Yes? And
thanks for the talk. Really, really great. I have actually 3 questions, all related to the same thing. I think first, I think I may missed for how long you keep your sensors in the water, and what's the limiting factor there? Is it fouling? Is it battery life? Is it the need to download the data?
Yeah, so last year we leave the system during 2 months long. And we didn't have really fouling problem because there is a copper plate on the sensor. And it works quite well. The main problem was the silt, because sometimes the silt arrive and your sensor can be under the sediment. So it is because we are working already very near the bottom. It's only 10 or 20 centimeters, so it is a risk.
Mm-hmm. Great. And then what will be the price of this Mastodon without the sensor?
2,000 euro, about 2,000 euro.
Okay, yeah, perfect. Great. Thank you so much.
Thanks. One question there, and then we move to the next speaker. You want to ask a question? Yeah, yeah.
Really abrupt recovery.
Mm-hmm.
What? Yeah. Yeah. Some of your time series had a really abrupt decline and then a really abrupt recovery.
Yeah.
Do you have a clear idea of how that worked?
We think it's due to the phytoplankton bloom, which can be very intense some days. And when they fall on the sediment, due to the organic matter decomposition, it can be very brief sometimes. So I don't know if you see the picture here. This bloom, you can see that they are very limited in space. So sometimes it's a great amount of organic matter which fall on the sediment, and this can be the explanation.
Okay, thank you. So we move to the next speaker now. Koen, yeah. And so we speak about habitat compression by extreme event in the Eastern Pacific.
All right.
Okay, hello everyone. Thank you for giving me the opportunity to talk here. This is a great pleasure to talk to you about oxygen extreme events in the Eastern Pacific. That's what I'm working for my PhD and the group of Nicholas Gruber. So throughout the conference, this has already been outlined nicely by a few talks, also by Marisa Vidor, that the presence of subsurface hypoxic volumes or the oxygen minimum zones really constrains the available habitat for heterotrophs in the upper ocean. And this is also shown by the study, for example, of Strum et al. In 2012, where they looked at the vertical diving depth of blue marlin in the Eastern tropical North Atlantic. Observations also suggest that the oxygen minimum zones are expanding with ocean deoxygenation, not only horizontally, but also vertically. And that means that the upper boundary of these oxygen minimum zones gradually comes closer to the surface. And bringing this together, then that means that the oxygenated habitat for these species would be more and more compressed. But then superimposed on this long-term trend, there's obviously also internal variability. And this internal variability can then affect the upper boundary of the oxygen minimum zone. And this is what I'm going to be focusing on this talk. And I'm going to be talking a lot about THREES. And that's my acronym for Transient Habitat Reduction Extreme Events. And I care about THREES because obviously there's a strong habitat compression during those moments that might affect ecosystems, but they might also provide us with a window into the future, maybe more deoxygenated state, and they might be a good study ground to see how future ecosystems respond to this more expanded oxygen minimum zones. But to kick off this work, first of all, we need to figure out when and where these THREES occur. And this is what I'm going to be talking about mostly and what their drivers are. To do this, I turn to a model study. I use, also introduced by Daniele Bianchi already, the RUMS-BEC model. I use it in a Pacific Basin setup outlined here on the slide. In the plot, I run a model hindcast from 1979 to 2016. And here you see basically the model grid outlined by the red lines with the pole located over South America. And then the model grid branches out and over the entire Pacific Basin. So we get our highest resolution in the Eastern Pacific. We force the model with error interim forcing, and we save daily output from the model. And the map shows also the mean oxygen concentration at 200 meters depth. And if you compare this to observations now, this is the orange lines from World Ocean Atlas. Basically, the take-home message from this is that the model does a pretty good job in reproducing the oxygen minimum zones in the Eastern Pacific. So THREES, how do we define them and how do we detect them in the model? First of all, we need to define the upper boundary of our oxygen minimum zones. I take it to be the 60 millimoles per cubic meter oxygen isopleth. And at each location, I look at the time series of this depth, and then I establish a threshold depth, which I take to be the 99th percentile of this hypoxic interface depth time series. And then obviously, whenever the hypoxic interface moves shallower than this threshold depth, then we have a 3. But I also require that the minimum duration of these events is 5 days. So everything shorter than that, I discard. And once we have defined our 3, we can then go on and define some characteristics and calculate them, such as the 3 onset timing, the duration of the event, the intensity, and the severity. What does it look like in the model? Here is just an example snapshot on the left from October and December in 2010. The orange, this is in the Eastern tropical North Pacific. You might you might see the coastline of Mexico and Central America there. The orange colors show you the habitat reduction in those snapshots. The orangey colors show you that there's a habitat reduction of 50–60% going on. And if you look at the locations of the blue axis, in the maps, then on the right-hand side, this is the corresponding time series for these moments, and shown are the oxygen concentrations and depth versus time. And basically the thick orange line shows you the hypoxic interface, and you see that during this A snapshot, that's the moment when it really shoals up closely to the surface and thus compresses the oxygenated habitat. We can do this then at every grid point of the model output. And now these are the composite of these characteristics of the events. Here on the left-hand side is just the number of events that we detect in the model run. So the yellowish colors show you that we have high numbers of events ranging up to 13, 15 events over the entire hindcast from 79 to 2016, mostly so in the Humboldt Current System and in the transition of the Eastern Tropical North Pacific to the subtropical North Pacific gyre. The mean duration is somewhat of the mirror image of the number of threes. So wherever you have elevated numbers of threes going on, there the mean duration of these events is somewhat shorter, just in the 5 to 15 or 25 days range. The most important or most interesting map probably is the one on the very right, showing you the composite mean habitat reduction of all these events going on at each grid point. And there you see that during those events, the habitat can be compressed usually around 50%, especially in the tropical Eastern Pacific, and in some locations it can go up to 70 or 80%. So it's pretty strong habitat compression going on during those extreme events. Right, then if we basically look at the entire Eastern Pacific here now outlined by this orange line in the map, we can basically calculate how much of the Eastern Pacific is affected by threes over time. And so what I plot on the right is a time series of these of the area fraction of the Eastern Pacific covered or affected by threes. And we see quite some interannual variability over the entire time series superimposed on, just a lot of variability going on. The mean value is about 1%, which makes a lot of sense because these threes are rare by definition. If you have interannual variability in the Eastern Pacific, first thing to think of is ENSO. And so I plot now here the Oceanic Nino Index. And there's a strong anti-correlation of the upper time series with the Oceanic Nino Index. And basically it tells you that we get the Eastern Pacific is preconditioned for the appearance of threes during La Nina periods. On shorter time scale, we can also look at the seasonal time scale, and what I plot now here on the left-hand side is a composite mean of the average onset of these events. And wherever there's a color, that means that these events, they prefer to start at a particular time of the year, where it's light gray, sort of masked out. That means the events, they sort of start during random times of the year. And basically throughout the subtropical and subpolar regions, it's rather random. But towards the tropical Pacific, we get quite strong seasonality. And if you look closely, even maybe between 10 south to 10 north, you even see somewhat of a westward phase propagation or westward phase shift, indicative of maybe a signal of the Rossby waves propagating westwards and lifting the hypoxic interface up. So now we've looked a little bit at the timing of these events, but we haven't really dove into the processes driving the events. So I turn to more of a dynamical understanding of these events, and I classify all my events based on the relative vorticity and the Ukubo-Weiss parameter of the horizontal flow field. And I basically plot here on the right-hand side the number of days that are extreme in the respective 4 classes that I identify. And these are cyclonic eddy threes, anticyclonic eddy threes, strain threes, and sluggish circulation threes. And basically what you can take home from these plots on the right is that towards the tropical Pacific, Eastern tropical Pacific, it's the sluggish circulation threes that dominate, whereas in the subpolar and subtropical regions, it's more the eddy-related threes that play a big role. Interestingly, if we look closely at the anticyclonic eddy threes, we looked at also the vertical density anomaly profiles associated with these events. And we get a signal in the density profiles that is really reminiscent of the isopycnal stretching that is really a typical signature of anticyclonic mode water eddies. And we pick out this anticyclonic eddy signal really at the edges of the core oxygen minimum zones. And so this really seems to show again that these anticyclonic mode water eddies play a big role in transporting the low oxygen water out of these oxygen minimum zone cores. So the question is, what are these sluggish circulation threes? I looked also at the density anomaly at my hypoxic interface, and I wanted to find out more about this. And I plot basically, again, the composite mean of the density anomaly at the hypoxic interface during threes. And I see that in the Eastern Tropical North Pacific, basically here, we don't see any density anomaly during the events compared to the normal state. And this is what I tried to sketch here in the sketch on the low right panel. This is basically where the hypoxic interface comes up close to the surface, but this is associated with a corresponding lift or uplift of the isopycnal. So this really shows you that the isopycnals move up and bring with it the low oxygen water. And the last thing, also addressing in the context of the multiple stressors, I also wanted to figure out whether other stresses or variables are extreme at the same time. And so for this, I turned towards the threshold depth, and I looked at other variables such as temperature and pH, and I averaged those values over the duration of the threes, and then I compared them to the general distribution of these events at that location. And what I find basically, again, here on the right-hand side, this is composite mean maps, but now I plot the mean percentile of temperature and pH at the threshold depth during threes. And the dark blue colors show you that the percentiles are really low so the dark blue color shows you it's below the first percentile. And so this shows you that, especially in the tropical Pacific, Eastern Tropical Pacific, during threes, the temperature also goes to extremely low values similar to the pH. And in the region where we had the anticyclonic eddy threes, there we get these reddish colors showing that the percentile are not so extreme. There it's rather normal. So calculating all this together, just here in the bottom, this is just a cumulative distribution function. The way to read it is basically just that here the 71% of the threes show pH values at the threshold depth below the first percentile, and this identifies them as compound events because they're at the same time threes and they're extremely low in acidity. And with this, I'm coming to my conclusions. These are basically all the summary sentences from the slides before. And I just wanted to point out again that the threes, they might really, first of all, they might affect the ecosystems right at the moment when the three is happening, but they might also provide us with a future, with a window into the future. And with this study, we might be able to anticipate the occurrence of threes. And so we might be able to sample them more and get a better understanding for how ecosystems react to this. Thank you very much.
Thank you very much. Time for question? Yeah, is there a question here? Yeah. Thank you.
Thank you, Eike, for a really nice talk. I'm really impressed by your work. I want to have an answer to, well, the basis of your work, defining these extremes. So if you have a relatively deep water column with low variabilities, it's a kilometer deep. I know that's not the scale, and it varies only a tiny bit, then your 99th percentile still leaves a huge water column for the species to swim around in. So I would not say it's necessarily extreme to those species if they lose only a few meters, right? So you run into this issue of defining your threshold. Can you say something about that?
Yeah, I mean, that's you actually see it here on the, oh no, there. I mean, you see it here on this slide, like if you look at the habitat reduction here, for example, in the Eastern North Pacific, where the hypoxic interface is rather deep in the water. I mean, there the habitat compression is still somewhat small. I mean, there we just get values of maybe 10%, 20%. So there, yeah, it's questionable whether that has really an impact on a species. But then again, I mean, oxygen is also not the only thing that affects the habitat's suitability, right? So I mean, we also heard talks yesterday about the interplay of oxygen and light, for example. So even if you have small shifts in where your hypoxic interface sits, this might have an impact on the species, their zonation and whatnot.
Thanks.
Yeah, thank you. Very nice talk. When you separate between cyclonic and anticyclonic, and I mean, a lot of the variability in that region is due to tropical instability waves and Rossby waves. And, you know, you wouldn't catch that with Okubo virus. And I mean, some of the patterns that you have, like the little snippet up there, look like could be wave propagation. So I was wondering, like, did you try to maybe your slugget might be actually a lot of it right at the latitude where you would expect those waves to propagate?
Yes. I mean, I don't have the plot here, but I hope it's soon available for everyone. But I mean, I look at Hofmiller diagrams. I hope you can hear me. I look at Hofmiller diagrams and I see some wave propagation that, I mean, drives already the seasonality in the hypoxic interface position, but also these threes, they sort of sit on top of this. So yes, I also think that, like, Rossby waves, for example, they play a big role in generating these threes.
You should add that more than just the cyclonic.
There are two questions here. And then, okay.
Have you looked at the relationship with the upwelling Kelvin waves in the equatorial area? Because these could be the main drivers of this showing of the oxycline. That's the first question. And the second, very rapidly, is I didn't see any long-term trend in your data. Is that surprising for you?
Yeah, maybe quickly to the second one. Yes, I don't see any long-term trend in an increase in threes. But I mean, I get this really strong interannual variability associated with ENSO. I mean, this might just potentially overrule everything else. So maybe behind there is some trend, but ENSO is just so strong in the Eastern Pacific that this might mask any sort of long-term trend. And towards the Kelvin waves, I mean, I haven't looked in detail, especially on the equator. I haven't really looked in detail into the processes there, but I mean, my imagination tells me, for example, in these snapshots here, along the Central American coast, I mean, you get these bands of habitat compression there, and I think, I mean, without having looked into this, but I would suspect that there might be something like an upwelling Kelvin wave propagating along the coast together with the La Nina event that was happening at that time, creating this habitat compression. Yeah, I think.
Okay, so we have to move to the next speaker. So thank you very much. The next speaker, Lars Sundlo, modeling hydrodynamics, control of sediment water fluxes.
Oh, can I switch slides? Okay, good. Okay, yeah, thanks to the organizers. So let's talk a little bit about hydrodynamic control of sediment water fluxes and the question about how this can be implemented into a coupled numerical model that contains a benthic compartment and also a water column compartment. This is work done in collaboration mostly with people from our institute, RUW, the Leibniz Institute for Baltic Sea Research in Warnemünde, but also with contributions from IGB in Berlin, PML in Plymouth, and it's all funded by the German Research Foundation. Okay, in the keynote this morning, we talked a little bit about breathing ocean, right? Now we're going to talk a little bit more about breathing sediments. And that's why I copied this figure here from an older paper by Andreas Lorca in his group. And this figure shows oxygen concentrations very close to the sediment. This is the sediment-water interface, sediment, the water column here. And if you look at the scale, it's just a couple of millimeters. And what you can see is that oxygen concentrations near the bottom are pretty well mixed. But then in a region that we call the diffusive sublayer, they almost linearly increase towards the concentration at the sediment-water interface. And this layer is very thin. In this example, it's of the order of less than a millimeter. And we know that inside the layer, fluxes are essentially molecular. So if you want to compute the flux of oxygen into the sediment, you basically have to estimate the gradient across that layer and multiply it with the molecular diffusivity that you know, of course. So the only thing basically you need to figure out is the thickness of that layer, and then of course the concentration difference between the top and the bottom. And that's straightforward if you work with in situ data, but if you want to have that process in your numerical model, it's getting very much harder because all these quantities that appear here in this fraction, they have dependencies and feedback mechanisms. So the thickness of the diffusive sublayer depends on your bottom turbulence. So you need a turbulence model that's able to deal with effects like stratification at the bottom, rotation rates, and bottom roughness, and so on. Then if you want to estimate these concentrations, especially here on the water side, you need to understand the oxygen dynamics on the water side, which basically means you need an ecosystem model. And the same thing for the interface concentration here, which depends on what is going on in your sediment. So you need also to understand benthic oxygen dynamics, and all these processes are intermingled, and you really need a coupled model to sort out how this all works. Okay, this is what I'm going to talk about, because I don't think that this process, which is important, is properly implemented in most of our coupled models that we have. So our model, and this is only a conceptual sketch here, I don't show you the equations because I don't have enough time, consists then of a water column compartment and a benthic compartment separated by the sediment-water interface, which is the black line here. And we solve transport equations in both compartments for the oxygen concentration, which is the first of two state variables that we have in this idealized model. And in this study, we really want to focus only on this hydrodynamic effect. So I try to strip everything from the model that is not directly related to this. So we, for example, ignore oxidation in the water column, and the only thing the model does in the water column is it mixes the oxygen that comes from the atmosphere down towards the sediment, right? So you need a turbulence model, which is part of this coupled system here. Then in the sediment compartment, we ignore resuspension, we ignore bioturbation, we assume cohesive sediments, and that means that our oxygen water flux is, sediment water flux is purely diffusive. Yeah? And we model this with such a bulk formula that is basically driven by a concentration difference at the lowest grid point, and the CB, which is the concentration directly at the sediment water interface. And then these models are usually constructed linearly in the friction velocity, and they include a transfer coefficient which includes all the dynamics of the diffusive sublayer. So that depends, for example, on the molecular Prandtl number, on the friction velocity, on the roughness, on a number of additional geometric parameters, depending on how complex you want to have it. And many people ignore that also on the distance from the bottom. So it really has to be implemented correctly, otherwise you get into trouble when you do this. Okay, then sediment is consumed in this simple idealized model in the upper part of the sediment, and this sink term here is spatially and temporally constant. Yeah, so it's as simple as you can do it, and we switch it off as soon as oxygen concentrations have decayed towards zero, right? So you only consume oxygen, obviously, where you have it. Okay, and then when you run it, it turns out that a lot of the oxygen that you actually need in the sediment is not directly expired, but it's used by reduced material that is generated down here and it's diffusing upwards. So it's oxygen used for the reoxidation of reduced chemical compounds. That's well known, and that's the reason why we have a second state variable here that we call oxygen demand units, and that basically sums up all the reduced material that we would have in the water column. So it's the most simple, idealized way to represent that. And this works physically exactly as the oxygen model, except that things are now moving upwards from the sediment where these oxygen demand units are produced. And again, this production term is constant in time and constant in space. And we switch it off exactly at the position where we start to have oxygen, right? So this is consumed. And when this is over, then you produce oxygen demand units. Okay, and then of course you have a redox-Klein where these two substances meet, where you have overlapping gradients. And in this redox-Klein region, you use the oxygen to reoxidize these reduced compounds. And that's in our model represented as such a second order kinetic term. And you see here the product of these two concentrations, and the KT is a model constant that somehow hides the time scale on which these chemical or biogeochemical reactions take place. Okay, just very briefly about the numerics, and you have to be really careful about this. So this is all based on our general ocean turbulence model, which is a kind of a toolbox for different turbulence models in the water column. Now, for this paper, extended by a sediment module, and then all biogeochemical reactions are done in the framework of aquatic biogeochemical models FABM. So this is coupled to COTM. A thing that can happen, and that actually does happen, is that if you take out oxygen from your lowest grid box and let it flow into the sediment, and you have a long time step, then your concentrations in the lowest grid box here on the water side, they may become negative, yeah, and then that messes up your model. So we grew a lot of gray hair actually when we ran into this problem, and we solved it with a special type to limit that sediment water flux. And it basically boils down to determining the order in which you solve the sediment and the water column compartment. Yeah? So you have to be really careful not to run in such negative concentrations. And then a similar thing happens as a result of this consumption term that we have here. If your time step is long, then that constant consumption rate can again take out more oxygen in your grid box than you have. Yeah? So this problem has been solved a couple of years ago by Hagen Radke, our co-author, and Hans Burkhardt, and they suggested a special type of a split time step or a forward scheme. Okay, and when you do this, and only when you do this, you can guarantee that your quantities all remain positive. That does not only apply to oxygen, but also to the oxygen demand units or whatever else you have in your ecosystem model, and you conserve mass. That is also very tricky in a situation where you have a redox-Klein that may move up and down across the sediment water interface here. So this numerics is difficult here. Okay, a first of two examples, and that then goes again back to the breathing sediments. So we set up a very simple idealized case that consists of a shallow water column, just 10 meters deep, unstratified, well mixed, and with oxygen concentrations close to saturation. And because that's so simple, I don't even show it here. And we drive the water column forth and back by a prescribed sinusoidal pressure gradient. And you can see the effect of this pressure gradient here. My mouse is gone. Here it is. On the velocities. And near the bottom, you see such a 24-hour, the time scale is down here, it's just a day, 24-hour variation of the speeds above the bottom of the order of a few centimeters per second. That's what you would expect in a lake, for example, in a fjord or in any low energy system. Yeah, and then this second panel here shows the benthic oxygen concentrations, and I'm showing only the upper centimeter here. And you can see that the concentrations are really strongly modulated by the currents above. So you can see that whenever you're close to the current reversal, then the oxygen concentrations collapse. They go very close to zero. And then as soon as the currents have reverted, accelerate again, then the sediment is breathing and it takes up oxygen again here. Okay. And the reason, oh, I'm sorry, can you still hear me? The reason why this happens are these variations in the diffusive sublayer thickness that you can see here in the third panel. So when the currents are strong, we have a sublayer that is less than a millimeter, and then around current reversal, it increases by more than an order of magnitude. And that directly impacts on the oxygen flux into the sediment, which is the full line here. So fluxes are negative when they go into the sediment, and you can see that at the current reversal, they are almost zero, and then they start again. So this is a nice illustration of this breathing concept that we have already heard about in the morning. And here it's even a rhythmic process. When you look very carefully, you see that this breathing is a little bit asymmetric around the points of current reversal. So after the reversal, you inhale kind of stronger, then you just do at the same current speed before. And the reason for this is that during the current reversal, you accumulate an oxygen deficit or an oxygen debt that you have to pay back as soon as physical control allows for it. Yeah? So this causes this asymmetry. And similarly, there is a dashed line, and that is the flux of reduced material of this oxygen demand units out of the sediment. And you can see that also this is stronger after the current reversal. So the system has kind of two responses to deal with an oxygen deficit. First, it inhales stronger, and second, it kind of exports reduced material to the water column where it's then oxidized. Yeah? So it's a different mechanism. I don't know how I'm doing time-wise, but that's my second last slide. Okay, good. All right. So this is just to show I don't know if it's the full complexity, but more complexity that the system can have. An example that consists again of a 10-meter water column, and now you can actually see it with the same diurnal forcing, except that we now run it for a much longer period, for more than a month, that you can see here. It's initialized, that's the water column here, initialized at 50% saturation, and we now include stratification effects by just prescribing a pycnocline. Yeah? Okay, and then we let it run, let it evolve, and you can see that the oxygen concentrations decay as a result of sedimentary oxygen demand, and then we prescribe a windy wind, water column is mixed, and the sediment that you can see here is oxidized. When you look closely, you see that many effects we've seen before are just repeated, so this rhythmic inhaling, exhaling of the sediment, modulation of the fluxes as a result of variations here in the diffusive boundary layer thickness. So the system reacts on this short time scale, which, if you recall that formula here, is just variations of the diffusive boundary layer thickness. But then if you look carefully on a longer time scale, you see that the oxygen flux decays and decays. And that's no reason due to variations of this difference that makes up the gradient, right? So you can see that the difference between the lowest grid point and the sediment water interface decreases, and we see the same effect here. Okay, good. I think I stop. So maybe we have time for some questions. And summing up, so I hope I could convince you that this hydrodynamic control matters. You need to be really careful about your numerical methods. Memory effects are important, so oxygen depth, loosely speaking. And then, and I don't know if that's known to others, it wasn't known to us, the system can react by exporting reduced compounds when there is an oxygen deficit instead of kind of inhaling more oxygen. And I think this could be important for the prediction of short-term deoxygenation events and also for seasonal variations of sediment water fluxes. Thanks.
Thanks for a very nice talk. I had a question about the how you parameterize the production of these oxygen demand units and how that relates to actual concentrations of ammonia and hydrogen sulfide.
Okay, so this quantity is difficult to measure, but what you can measure is how much hydrogen sulfide, for example, diffuses across the sediment water interface. So there are measurements for this, and then when you know the thickness of your sediment layer and the flux across the sediment water interface, you have an estimate for your production term. So, and we looked into literature to do that.
Okay, but maybe just if I have time. So is it determined then by the gradient in the oxygen, or is it related to some formulation of the stoichiometry of how much units you would produce based on the consumption of the oxygen?
In this case, the production rate is fixed, but of course you can make it depending on whatever you want, on temperature or on some other biogeochemical parameters. Yeah, but we fixed it because we wanted to have it simple. Yeah.
Okay, so I'm afraid that we have to move to the next speaker, so you can continue, of course, during the coffee break. So at the last speaker before the coffee break, Pierre Langemann, and so we'll still speak about modeling, unstructured grid modeling of the global coastal ocean. Yeah.
Can you hear me? Yeah, okay. That's there. Forward. So the title with consistent global modeling is maybe was a bit too optimistic, causing several panic attacks because it's an early stage of modeling. So you will not learn much about oxygen in the ocean right now, but more about modeling of, maybe I can convince you that this is a quite promising development in Hamburg right now. It's all about the ICON model. That's a framework recently created in Hamburg by the German Met Office, Deutscher Wetterdienst, and the Max-Planck Institute. And it's a framework. It's a Earth system model framework with the atmospheric part developed by the Met Office, the ocean part by the Max Planck Institute. Then there's the ICON L for the land river fluxes, and the HAMOC is the ocean biogeochemistry part, and the name ICON refers to this icosahedron, this one of these 2,500 years platonic solids, a body which is created by 20 triangle surfaces. And this ICON, this geometrical structure, is used here to resolve the sphere of the Earth with triangles. These triangles can be refined by bisectional in order to achieve the resolution you're interested in. So the basic motivation, I guess, for this ICON development is the technical advantages. So it's computed by experts, developed and programmed by experts to use this brand new machine we just recently got at the Deutsche Klimarechenzentrum, the Levante, very efficiently. So that means we can spread this computational effort to 200 cores, 200 single nodes, and it's 200 times faster than just running it on one node. So it's the scalability, which is very nice. And the ocean part is a very robust model. That is my impression as an ocean modeler. So I'm an ocean modeler, not an ecosystem modeler. So why these unstructured grids? So one of the big difference to the models used before is these different meshes, these triangles. First, it's avoiding the meridional convergence, this kind of what one of the other speakers this morning or this afternoon was showing, this kind of polar convergence or anomaly of the grid at the north and the south poles, which you can, as an ocean modeler, you can avoid by shifting the poles onto land masses. But still you have this kind of distorted grid, maybe resolution at areas where you're not necessarily interested in. And with an icosahedra, obviously you don't have this problem. Second, with having triangles, you're able to resolve a coastline like this much better than we're having this steppy structure in a structured grid. And third, you are able to resolve, to change your resolution at regions where you're interested. And I think a nice example is the Strait of Gibraltar, where you have to resolve the ocean quite highly to be able to get the fluxes through, which are influencing the entire North Atlantic by this high saline outflow. And if you don't want to resolve the entire North Atlantic with 1 kilometer, then such a grid would give you the opportunity just to resolve the strait highly enough and then having the rest in a more convenient, lower resolution. What we did at Geesthacht, at the Institute of Coastal Systems, close to Hamburg in the forest, we used this ICON-O, the ocean part of ICON, and are still on the way to develop a kind of coastal version of this ICON-O, called ICON Coast. And for these two things had to be imported into ICON-O, the tides, which weren't there at that time, and this kind of coastal mesh refinement. So we developed a tidal subroutine, just taking the tidal potential, inserting these accelerations into the equation of motions, and we wrote a program which puts out grids which are the right format and the right structure for ICON. This is a little bit, so you can't use any triangle for this model. The triangles need some quality for ICON. And this paper, I published it last year, where I just tested the performance of tidal waves on these irregular meshes with high coastal resolution. It worked. So we were able to improve the precision of tidal waves with this kind of mesh. And so we had the generator for ICON-O, following our coastal perspective, and the tidal subroutine. And then my colleague Moritz Mattis was going into this HAMMOCK, the ecosystem part, and was improving several parts like shelf-specific process representations like sedimentary suspension by tidal currents, temperature-dependent remineralization, riverine metafluxes, variable sinking speeds. So he submitted this, will be hopefully published this year. These experiments regarding, he's more in carbon fluxes. And what you see here is their vertically integrated netto primary production, and also computed on such a tidal coastal, high resolution coastal mesh, and works quite fine. Whereas because we at the Institute, we have a lot of applications traditionally around, the speaker in front of me, having this eCosmo model. He didn't use the eCosmo model, but the FABM framework. So we have a framework here and another framework on the other side. And the quite challenging task was to connect this, to couple these two frameworks that by this library linked to the ICON Ocean, that we are able to use the ecosystem parts which are already existing at our institute. And so we have this. Oh, that was the wrong direction. And then for this, of course, we are to add another subroutine which calls the eCosmo, the ecosystem part, and which overtakes some physical computations like the light attenuation, depending on plankton concentration, the sinking of particles, and the data transformation. So it's a bit tricky because the FABM framework needs a little bit more ordered data than the ICON, as far as I understand. It's a bit, yeah, a hurdle of this development, but it works, I think. So for, what I had to do with the ECOSMO model was used mostly for Northern Sea, North Sea, Baltic Sea, Northern Atlantic. But if you want to use it on a global scale, you have to add the iron. That's the iron limitation, or iron as an additional nutrient. And so I did this as well. It's also not so trivial to do. It's wrong direction. And here I just sketched. So and yeah, you have seen the ICON model. I probably don't have to the ECOSMO model. Here was these 3 kinds of plankton, zooplankton. Here we have the nitrates, the nutrients with the iron added, and this is dissolved organic matter and detritus and 3 kinds of sediments. There are also fish classes in my version of ECOSMO. On the right side, I try to sketch the oxygen fluxes as far as I understand this model. So we have the exchange at the surface, then we have consumption of oxygen by the detritus, the dissolved organic matter, the fish, the zooplankton, and chemical reactions like nitration or nitrification, consuming oxygen, and then we have the primary production as another oxygen source in this system. And yeah, okay, wrong direction. And this is the first result on my training mesh, homogeneous, low resolving mesh, just to have it fast and easy. And you see, that was really more or less the first run, and it looks quite realistic as far as I can say. That's in 80-meter depth, the oxygen concentration. So we have this classical structure. And even if I start the model, so it's initialized with the World Ocean Atlas data in this case, but to check it, I also initialized it with homogeneous oxygen concentration, just the mean value for the entire global ocean. And after a couple of years, the structure came out. And if I switch off, which you see in the next slide, so this, then I wanted to check, to test this on a more sophisticated grid, like this one, going up to 14 kilometers at the coast. And this is only, and here after a few days, the model crashes. I still don't know. I think it's a trivial thing, but I didn't have the time to check. But what I can see in this crash, so there was something like a missing iron everywhere and collapsing primary production after a couple of days. And then I could see the oxygen increasing globally enormously, very fast. For me, as a more or less outsider, quite surprising how fast the oxygen, these blue patches disappeared more or less after, just by switching off the primary production, but of course it's the mechanism in this model. So we have primary production and detritus sinking down and consuming the oxygen. Okay, so the next steps will be, of course, coastal. If we have this coastal focus, having riverine nutrient loads or just resolving the coast higher. The question is, will we be able to resolve these coastal low oxygen structures, these red dots in this graphic we have seen these days every 10 minutes, and then I just put it here. It was just an idea, maybe completely wrong, but because I've read somewhere that the global models predict an increase of precipitation over the Sahara, which could reduce the dust deposition over the ocean and less iron. And would this cause an increase of oxygen if we would have some these kind of missing dust? And but probably you, if you believe me that this is this model development will be successful, maybe you have great ideas. Just write me an email. We can make some cool simulations in future. Thank you very much for your attendance.
Thank you. Okay. Time for one quick question. No?
No.
Yeah. Yeah.
Sorry, use the mic.
Okay, I repeat it. Do you have to run it all the time in a global mode, or could you also run a sub area around Europe or somewhere else? Yeah, good. Yeah, let's also discuss to make it regional. The German Weather Service also wants this. They are not so much into this grid refinement things. Want to make these regional models open boundaries are at the moment not included in IconOcean. So you.
Okay, one last question by
Andreas. How do you deal with the time steps if you have very different grid resolutions?
Yeah, of course. That's a good point. The time step has to be adjusted to the smallest triangles. That it's no magic. So, but we save a lot of computation still. It's like music compressed in what is the format? This MP3. You don't go back to WAV through. Meanwhile, our players have so much memory. We could also use WAVs, but it's like MP3 mesh.
Thank you very much.
So it's our pleasure to start again that session. And the first talk will be with Abdul Wahab Tal, and he will be talking about the modeling of the dissolved oxygen cycle, budget, and Lagrangian analysis on the Senegalese shelf. The floor is yours.
Thanks. First of all, I'm Abdul Wahab Tal. I am a PhD student from Senegal. First, I want to thank the PAGE program which finances this trip. My talk is entitled Modeling the Dissolved Oxygen Cycle, Oxygen Budget, and Lagrangian Analysis on the Senegalese Shelf. First, why are we studying the dissolved oxygen in particular on the Senegalese Shelf? Several studies are alerted to the global trend of oxygen depletion. You can see in this figure, the deoxygenation trends are very important in the coastal areas. Of the West African continent, the study of oxygen is focused on the variability of oxygen minimum zone, in particular of the Mauritania, where it is very strong. You can see in the rectangle, there are very few observations near the Senegalese coast. Our understanding of this upwelling system is very limited. However, a previous study showed a mortality of Serendai fish, and it was attributed to low oxygen concentrations. Recently, during the upwelling season in March 2012, anoxic waters were measured on the shelf for the first time. And recently, a dissolved oxygen time series was collected by a Melach buoy at the bottom layer of the Senegalese shelf. We use a model to analyze the variability of oxygen on the Senegalese shelf over the entire shelf. Our objective is to study the bottom dissolved oxygen variability during the upwelling season over the Senegalese shelf using a regional model. We seek first to understand the role of the physical and biochemical processes controlling the dissolved oxygen budget, and second, to study the characteristics of the upwell source waters using a Lagrangian approach. The Croco model Pisces architecture is presented here in this slide. First, the oxygen, the processes that can vary oxygen is represented here in red. The biochemical Pisces model is the NPZD model type with several compartments and several nutrients. Oxygen is produced by photosynthesis and then consumed by remediation and the elimination of organic matter and by the respiration of the plankton. Physical processes are advection and mixing. The oxygen flux at LC interface is also taken into account. Let's now go to the model characteristics. The model is made by a large domain at 10 kilometers resolution and high resolution at 2 kilometers resolution. As physical forcing, we use the daily Ascot wind over the period for 2014 to 2019 and a climatological heat and freshwater fluxes. The initial and boundary conditions are the Mercator model for the physics and the WOA and GLODAP data for the biochemical parameters. Model outputs are model variables, oxygen budget terms, and the biochemical terms. For the evaluation of the model, I show you in this figure the comparison with a time series of collected by the Melach boy on the shelf. A paper analyzing these observations has already published and presented on the CSIR question, and the poster session. And here in RAG, I show you the position of the station and the Melach buoy picture. The comparison shows a seasonal variability with low dissolved oxygen during the opening season and a strong intra-seasonal fluctuations related to the synoptic wind events. And it shows also the dissolved oxygen occasionally below hypoxic threshold. For overall, the model show a good match between the model and observing dissolved oxygen. In the rest of the talk, I will focus on the opening season shown here in the blue rectangles. Let's now go for the results. All these figures are average on the opening season and at the bottom layer. The bottom dissolved oxygen distribution shows a high oxygen waters in inner shelf and low dissolved oxygen offshore. And also it shows a strong cross-shore gradient. As expected, dynamic brings oxygen while biochemical consume it. The contribution are important in near the coast. Let's now focus on the physical processes represented here by this equation. The first term in blue is advection, and in red the vertical mixing. As I show, that advection is a globally negative, taking out oxygen. The maximum advection is due to strong dissolved oxygen gradient near 20 to 30 meters isobars. Vertical mixing brings oxygen to the bottom layer by mixing surface oxygenated water and low dissolved oxygenated bottom waters. Oxygen, let's now focus on the biochemical processes. Oxygen is produced by photosynthesis at the shallow waters. And if you compare these two, the regenerated production and new production, we see that the regenerated production is significantly higher than the new production over the entire shelf. If I focus on only on the new production, This is strong in the north, where the upwelling of subsurface nitrate is located. Oxygen sinks are remineralization and zooplankton respiration. The comparison shows the dissolved oxygen of biochemical dissolved oxygen consumption is largely dominated by organic matter remineralization, which largely compensates production by photosynthesis. It shows also zooplankton respiration levels are lower than the organic matter respiration remineralization. Note that the mesozooplankton respiration is largely dominated the microzooplankton respiration. The second part of this presentation is to analyze the characteristics of water masses reaching the shelf. The methodology is following. We release 500 floats transported backward in time for 30 days. They are released between 30 and 40 meters around Mela station, shown here by the red rectangles. They are released every 5 days from February to April in each year. We analyzed in total 45,000 floats and oxygen concentrations. Depths, waters, vertical mixing, and biogeochemical terms are registered along the trajectories. In the figure in left show the source water transport converging towards the shelf, and in right, the evolution of the depths of the floats. This slide shows the evolution of dissolved oxygen source waters, and it shows an increase of dissolved oxygen during a month, and a low dissolved oxygen during the first 15 days, like minus 13 to minus 30 to 15, minus 15, and it increased rapidly during the last 15 days. This figure shows also a strong interannual variability, in particular in 2017, in the last 15 days. Why is that? I show you in the figure on right, the vertical mixing as positive terms and the biochemical contribution as negative terms. And this figure shows a low dissolved oxygen supply by vertical mixing in 2017 and 2016, and a low dissolved oxygen consumption by biochemical in 2016. The combination of these two processes can explain the low dissolved oxygen in 2017 in the last 15 days. This figure shows also a strong vertical mixing and consumption in 2015 because of shallow source waters. To conclude, physics brings dissolved oxygen and biology consumes dissolved oxygen in the bottom layer on the continental shelf during the upwelling season. Advection decreases dissolved oxygen by transporting low dissolved oxygen subsurface water onto the shelf, and vertical mixing ventilates the bottom layer. Consumption of dissolved oxygen by remineralization of organic matter strongly compensates production of dissolved oxygen by photosynthesis in the bottom layer. The source waters show high interannual variability with the lowest oxygen levels encountered in 2017 associated with reduced vertical mixing. Thank you.
Thank you, Abdul, for this nice talk. Are there questions? Yes.
Hi, you showed that in the model you could differentiate the new production and the recirculation production. I was just curious how you do that from the model.
Yes.
Sorry, Tal asked me to help for the questions. I'm one of these supervisors. So your question was how we separate new production and regenerated production? Well, in the model, there is ammonium and nitrate. So we separate these two terms in the equations, and they are stored in different parts. So we can diagnose this. We diagnose this that way.
I have a quick question. Is there any impact on those low oxygen level on the artisanal fisheries? Have you looked at that?
As Tal said in the introduction, there was an impact. I mean, there has been documentation of a dead fish due to low oxygen in the past. For the moment, no, but there is a lot of fishery, artisanal fisheries, so it's really a concern of the local authorities. So that's why we did this study. And also, as you saw, that the mooring is quite offshore at 35-meter depths. So maybe there are like more anoxic events or hypoxic events near the coast.
Okay. Well, thank you, Abdul. We'll move to the next talk, Christina Schulz. Thank you. So Christina will talk about using model output to understand oxygen variability and inform agro-sampling strategies in the California current system.
Okay. That's the slides, because that's bad. Okay. And can people see this? Okay. Thanks everyone for listening to me. So I'm going to be talking a little bit about part of the work that I've been doing with John Dunn at the Geophysical Fluid Dynamics Laboratory. So we're using models to understand oxygen variability and decide where do we put the biogeochemical Argo floats in California. Do I just press this? Okay. So a little bit about the Argo project for those who are not familiar. The Argo floats are just these floats that you put them in the ocean, and then they will sink to about 1,000 meters, and then they're there for a predetermined amount of time, like 5 or 10 days. And then when it's time for them to sample, they sink to the depth that you want to sample, and then they sample from that depth up to the surface and basically phone home with the data. So you can put these floats in the ocean, and they will just like go around for up to 4 years collecting data. So they're incredibly useful to fill in the gaps of some of the regions that it's high to get data at this type of resolution, right? So this map that I'm showing is the location of Argo floats in December 2021. The red points are the places where you have floats with physical data, so temperature and salinity basically. But you can see that there are some green dots that are popping up, that those are the places where you have biogeochemical floats. So you have sensors like oxygen, nutrient, chlorophyll, backscatter, and those are the ones that are available for now. So what we want to do is see where do we need more information on biogeochemical variables and at what frequency in the California current? What mechanisms are causing these biogeochemical variations? And how can we use Argo data to help improve models so that we can work towards having an ecological forecast model, which would be really important in this region where you have a lot of fisheries and a lot of people using the coast. So you really want to make sure that you don't have harmful algal blooms. And, I mean, you can't prevent that, but that people are not there. So just a very basic model class, just so to say. So I'm working with different models, and part of the reason I'm doing that is because there's always a little bit of a trade-off between how many processes you're solving and what is the resolution that you can get to the model. So here, just if you have an Earth system model, I'm using 2 different Earth system models. You're solving for the ocean, you're solving for the land, and you're solving all these processes in the atmosphere and the ocean are like fully integrated with each other. But then you can't, because it's so heavy, you have to compromise on the resolution you use, versus if you just use an
an ocean model, excuse me, and force the atmosphere, you're solving for a subset of those processes, but you can have a higher resolution. So if you paid attention to the video, you saw that if you use an ocean-only model, you can have a high enough resolution to solve for the eddy dynamics, which might be important in redistributing some properties. So as I said, I'm working with 3 different models. What I have here on the top left is the surface world ocean atlas salinity at the, and then I have the model results, the model averages for the surface salinity interpolated to a 1 degree. So I'm working with 3 different models. ESM 4.1, which runs at a ½ of a degree latitude longitude resolution, but has very comprehensive biogeochemistry, has cobalt biogeochemistry with 33 different variables. Then we have CM4, which is another Earth system model, but that has double the resolution, so a ½ of a degree, but it has a much simpler biogeochemistry. And then MOM6 only, so just the ocean model, which has an 1/8 of a degree latitude longitude resolution and the comprehensive biogeochemistry. So the first thing we did was to calculate the standard deviation of oxygen for all of the models using monthly means from 1965 to 2017 to get an idea of where you're going to have more variability. So what I'm showing here, each column is going to be a different model with model resolution increasing from left to right and depth increasing from top to bottom. So you see that at 50 meters, most of the variability is focused at the coast. And then as you move to deeper regions, you're going to have this variability going offshore. So that's the main takeaway from this message. Some other things that you can see here is that as you increase the model resolution, this region of high variability is smoother, so to say, which so far we're attributing to the fact that the high resolution is better at capturing eddy dynamics, but I would love to talk to people some more about it. And then what I did next was to see, and another thing is that below 100 meters, if you remove the seasonal cycle, you have pretty much the same results. So the seasonal cycle is mostly important down to 100 meters. And then below that, this is mostly interannual variability. So next, what we did was to choose a transect, which is this black line on the left column, that goes through this region of high oxygen variability. And then what we did was plot a Hofmöller diagram to see the temporal evolution of the oxygen anomalies. So what I'm showing here is for this transect that's shown again here on the top right, a Hofmöller diagram. So you have time on the x-axis and latitude on the y-axis. Again, the different models. So increasing model resolution to the right and increasing depth to the bottom. And this is the oxygen anomaly relative to the monthly climatology. So red is where you have more oxygen than usual, and blue is where you have less oxygen than usual. And what you see here is that at 100 meters, you have a larger signal of anomalies towards the coast at the northern part of the transect. And as you move deeper, this moves offshore. And the interesting thing is at 200 meters, and again, you can see that the MOM6 has a messier profile, which is different from the Earth system models. But at some point around 200 meters, it seems like these anomalies get captured and then they get transported towards the southwest, right? Which we saw on the previous figure as well. And this is consistent between all the different models that we see here. So you might be wondering, okay, how does that compare to data? So this is just a quick rough comparison with the Argo data. So on the right, you have some of the Argo flowed tracks. And then I did for the Argo flows that we have 4 years of data, just comparing the standard deviation with depth for each of the models and the Argo data. So you see that sometimes the models do really well, sometimes they overestimate or underestimate. So MOM6, which is the high resolution, tends to underestimate the standard deviation for the oxygen data, and the Earth system models tend to overestimate. But mostly what I get from this figure is that we need more of this type of data. A different analysis we did was to look at to see which is the main axis of transport from these oxygen variables. So I chose two different transects. One is north-south, which is shown in blue on the figure to the top right, and one is east-west in red. So this is the north-south profile, and what I'm plotting here is the oxygen anomalies for each of the models. On the left, you have on the right, you have the coast, and then on the, this is for 128 degrees west. So north to the right, south to the left. And you can see that these anomalies, they kind of pop up and disappear not very far from where they started in all the different models. And the black line is showing the potential density range for the mode water. So that would indicate the large scale basin circulation. And if you look at the east-west profile, you see again that these anomalies are located in the density range of the large-scale circulation, but that they tend to go farther. So what we get from this is that you have these anomalies being generated near the coast, and then they get transported offshore, downwell at some point, and then captured into the large-scale circulation. So one thing we did to try to get to what's causing these anomalies and like how do we see where this is coming from was to do an extended optimal multi-parameter analysis. So a water mass analysis that has been done very successfully in the California Current by Steve Bograd and Natalia Evans. You'll hear from Natalia tomorrow. They did it using the CalCoffee data in the California Current and near California. So basically what this method is that you use a mixing line to, you choose some end members of interest where you think the water masses are formed that are influencing your region. And then you calculate a mixing curve from temperature and salinity. And we also use nutrients. And then we add a term here, which is related to the age of the water mass and the rate of oxygen consumption. And then you can calculate this, you can have the same term applied to the other nutrients using a Redfield ratio. So basically it's going to give you the fraction of the water mass and a term that represents how much oxygen was consumed along the way. And so what I'm showing here, I did this analysis for ESM 4.1, which is the only model for which we have all the variables that we need for all the time series we're looking at. And so I'm showing the mean and standard deviation of the subpolar water mass, the central water mass, equatorial, and then oxygen at the bottom to compare. So what you can see here is that the places where you have high oxygen variability, you're also gonna have high variability on the subpolar and equatorial water masses. And then if you look at these black dots here that kind of go along where you have the high oxygen variability and calculate the correlation with the water mass anomalies, you see that they're all like pretty high. So reminding that the equatorial water here in green, I multiplied the correlation by -1 just to get the magnitude of it. So basically what this tells you is that where you have equatorial water, you have low oxygen. That's a low oxygen water mass. If it's there, you're gonna have low oxygen. So then another thing that we did that you can do with the models that's kind of cool is to get to how much biogeochemical transformation is influencing the oxygen. You can get the diagnostics from the model and then calculate what would your oxygen concentration be if you didn't have any biogeochemistry. So just get the fraction of each water mass and multiply by the concentration of the end member. So on the top here, you have the diagnostics oxygen from the model. And then you have the end member mixing, which would be just the physics if it was just physical mixing. And then the difference between these two quantities is going to give you how much biogeochemical transformation you've had by the time you measure that you get this data. So you can see here again that the physics is going to have this range here where you have a high physics, a high variability on the end member mixing. so that your oxygen concentration here, where you have this high oxygen variability, is related to physics. And then the biogeochemical, most of it is at the bottom, which is where you're gonna have most of the respiration, the older water masses and a lot of respiration. And then you have these two different areas where you have high biogeochemical transformation at the surface, which could be related to primary production, and at the bottom where it's most likely related to this respiration that you get here. So some of the conclusions that we get from this analysis so far is that this highlights the importance of coastal processes in driving oxygen variability in the open ocean, with the implication that there's limited predictability of coastal variability through the understanding of the offshore variability. So what I mean by that is that we started by thinking, okay, if we put all these Argo floats where we want our boundaries for a regional model to be, then we're going to get all this information and guarantee that the information that's coming into the model is going to be more accurate. But if you have these coastal processes transporting, so to say, this oxygen variability offshore, there's just so much you have to gain from that. So what my recommendation would be so far is that if we want to move towards increased predictability for this region, we really got to make sure that we get the coastal processes right, and we really got to sample this this place where the oxygen anomalies down well to make sure that we fully understand what's happening there. So some of the next steps we have is to use the water mass, simulated water mass age from the models to calculate these remineralization rates, compare these to the Argo floats, and further investigate the role of eddies in smoothing this variability signal in the high resolution models. So thank you very much.
Thank you, Christina. One question, Sarah.
Great talk, thank you. I don't have a modeling background. I'm a bacterial physiologist. So I'm just curious about how modelers calculate the oxygen consumption variable, and if that varies based on the oxygen concentration of the water mass.
So yeah, the oxygen concentration of the water mass, that's something I didn't get to, but this what's one of the things that were interesting when we're doing this analysis is that where you get the variability of oxygen is not necessarily where you're going to get variability from temperature and salinity. So that's when we're like, okay, there's something else going on there that's not just local processes. And it turns out that because you have different gradients between the water masses, that's gonna, so if the oxygen gradient is higher than the temperature gradient, you're gonna have a larger signal in oxygen than you're gonna have in temperature. So that's one of the things. But the way you calculate the oxygen consumption is just iterative ecosystem respiration. So let's add those up.
Okay, thank you, Christina. I think we need to move on. So our next talk is Evgeny Yakushev, and he will talk about fish farm emission impact of oxygen depletion in a Norwegian fjord by observing and modeling.
Thank you. The title is here, and actually that's a result of studies of several people that I present here. This is a story about how anthropogenic impact influence oxygen regime regarding this conference. So it's needed to have a short introduction. Fish farming industry faces challenges connected with environmental management in aquaculture site, and it's important to investigate the potential impact of waste product on the coastal and marine environment. And models are cost-efficient methods to evaluate impacts of the site selection and production levels of the environment, and can be used as environmental impact assessment. So actually what is needed in many cases. And the oxygen condition is usually a main criteria that usually taken into account in fish industry planning, because in many countries there exist certain numbers, levels of oxygen that that can be used as to allow and not to allow fish farming. So the objective was to develop and configure a model to simulate the effect of fish farm organic loadings at 1 kilometer horizontal scale, optimize using field data, and quantify the effects of organic loading from fish farming on water column and sediment biogeochemistry, first of all, on oxygen condition. So for this, we used a model named BROM. That's a pelagic, benthic pelagic biogeochemical model. So it is a fully coupled pelagic benthic model. That means that domain includes both water column, bottom boundary layer, and sediments. And for example, redox can go from the sediments to the water column and back. And from biogeochemical point of view, we have a very simplified ecosystem model with autotroph and heterotrophs, but with several bacteria that are needed to parameterize organic matter production during the chemosynthesis. There's autotrophic and heterotrophic bacteria that are typical for oxic and anoxic conditions. And also we have organic matter, and we have connected cycles of elements written here. So it's oxygen, nitrogen, manganese, iron, sulfur, carbon, and phosphorus. Also we have alkalinity and DIC, and we can produce pH. And in the upper in the sediment in the water air interface, we have fluxes of oxygen and CO2. And as a result of application of this model, we can calculate pH, we can calculate oxygen, and we can calculate fluxes of many elements. So this is a more detailed figure of our biogeochemical model. So we have Redfield stoichiometry for phytoplankton, zooplankton, and labile organic matter. Then we have semi-labile organic matter, and so we have non-Redfield stoichiometry in this block. We have basic nutrients that are forms of nitrogen, phosphorus, silicate, carbon, and actually we take into account all possible processes of organic matter mineralization, that oxic mineralization, denitrification, reduction of manganese and iron, sulfate reduction, and methanogenesis. So this is a classical reproduction of a process occurring in marine sediments, and the same can be found in the water column. So all this is fixed. And on top of this, we have cycles of several elements, as I said before. And from point of view of oxygen, so oxygen is not only produced by phytoplankton and used for nitrification and mineralization of organic matter, but also it participates in redox reactions with reduced forms of nitrogen, manganese, iron, and sulfur. For carbon system, we consider alkalinity, and we take into account changes in alkalinity due to redox reactions when proton is produced or consumed. So there is also a poster presentation with this model, and I will be happy to describe about details if you will have questions. Then several years ago, we were happy to start to work with FABOM. Already in two presentations, this framework was mentioned. And actually all our model is written in FABOM, let's say, language, and all these biogeochemical cycles are represented like blocks. And for example, everybody can download and take them, for example, cycle for sulfur or manganese that we have here. And also a transport model. And we started from 1D model that was written here. And actually how it works. So we couple BROM biogeochemistry and BROM transport with FABOM. But then, because we don't want to be responsible of hydraulic process, we usually take forcing from existing models, such as we had experience with ROMS, GETM, FECOM, and Telemac up to now. And then in standard way, we produce resulting NC files and somehow process them. So from point of view of vertical resolution and processes, in the water column, we have the same vertical resolution and processes of transport that are in hydrodynamical model we force with. In the bottom boundary layer, we have linear decrease of diffusivity to 0 at sediment water interface. And then we have only molecular diffusivity in the sediments. Then for this task and some others, we made a two-dimensional vertical benthic pelagic model that again considers as in one model the main processes in the sediments and in the column. And this 2D model allows to use also advection components that allows us to transport some impacts in a horizontal direction. So in this 2D model, we have actually our area of interest where we have different injection, where we have a smaller horizontal grid, and then larger horizontal grid near the boundaries. And actually then we just have a periodic horizontal boundary that allows us to avoid problems with horizontal boundary conditions. So for this task, we modeled Hardanger Fjord, this fjord in Western Norway. And we use results of ROMS calculations, the Copernicus database for forcing with nutrients. So our horizontal resolution started from 25 meters. That's a typical diameter of a fish cage. And the maximum was, as I remember, about 100 or something. This is Hardanger Fjord. It's positioned here. In this fjord, there are several farms, and we were happy to be involved in field studies there, where we took samples from the water column and from the sediments. These are distributions with depths of temperature, salinity, oxygen, pH, phosphorus, ammonia, nitrate. It should be somewhere silicate, organic form of nitrogen, phosphorus, and DOC, and TIC and alkalinity. So this is a typical distribution for this fjord. Then we were allowed by owner of this farm to take one sample just near the cage, and we had these crosses. So just near the cage, we had decrease of oxygen. So this decrease was to 45 micromoles, and very clear increase at 7 micromoles of ammonia. So this is ammonia, like violet. For other parameters, also it was a reasonable change of concentration, for example, decrease of pH, but not so significant as for ammonia and for oxygen. So for modeling, modeling was made in 3 stages, actually as usual we do. So first we calculate basic seasonality with spin-up period to be sure that we model this fjord and not something else. Then we compare or validate with observations our results, and then actually we parameterize fish farm. So for this spin-up period, we started from uniform conditions. For example, this vertical distribution of oxygen in the water column and in the sediment water interface. So this is inside the sediment, and this is in bottom boundary layer. First, it was one number, as I remember, 100. And then next year, after 1 year of calculation, we had already better condition where we had no oxygen in the sediments, for example. And we continued this for many, many years, and then tuned our have distributions close to what we have in nature. So this is oxygen, phosphate, nitrate, ammonia, alkalinity, and DIC. For example, for oxygen, we had no observations in the sediments, but oxygen disappears in up millimeters of the sediments as it should be, as it was shown already in our conference. So we modeled 3 cages that stayed in the middle of our insect and that produced waste in the low boundary at 60-meter depth. So these points are observations of POC discharge during the installation of our fish farm. So this is time in days, and usually installations are in April, and they stay for 2 years. And next November, fish is taken from this cage. So we made parameterization of this flux. And actually, this flux corresponds to this percentage that actually goes through the fish cage to the bottom. On top of this, so these red numbers are from literature estimates. On top of this, there are estimates about oxygen consumption and production of nutrients by living organisms of fish. So now some short film. So this is this 2-year periods. So blue is winter, yellow is summer. And this is distribution of waste that is produced here. In low plot is its depths, the depth of water column. This is sediment water interface. So sediment is there, and this is bottom boundary layer. This is distance in meters. And now you will see from the beginning how this waste starts to precipitate down. And in oxygen, this oxygen penetration depth actually decreases and decreases to 0 during the fish farm installation. So then with time, so these are these 2 years. So this is a baseline condition. This is with fish farm. So we have accumulation of fish waste, fish waste in the bottom. And also we have for oxygen and ammonia and what is this? Sorry, I had forgotten my good glasses, so I have problems to read all this. So it is possible to distinguish this region affected and not affected. And if we look from top, this is like, this is distance, this is time for 10 years. This is at 10-meter depth, the depth of the fish cage. So we detect their waste, we detect the decrease of oxygen and growth of ammonia. And also we have even this small increase of phytoplankton. So that demonstrates that this is irrigation by fish farming. And also for those who like ocean acidification, so we should have some kind of decrease of pH because of certain reasons. But cancel this farm, so the system returns immediately back in the upper layer. In the bottom layer, we had surprisingly results that all this again potential changes. They can be traced for many years, actually up to 10 years. When first we spoke with our colleagues in Norway who works with fish farming, they said that about 2 or 3 years it is possible, but they don't know is it possible more. And then we found a publication of work made in Australia, and they said the same. So it might be about 10 years. So that's one of example when models can be useful for something. So, and of course, model shows appearance in the water columns, this reduced forms of manganese and iron and a huge concentration of ammonia. And I showed here very, very small concentration of hydrogen sulfide. So model works and reasonably produce what happens there. So this is again horizontal transect was going on. It's showing how this accumulation of organic matter takes place in the sediments and how it disappears and is being buried later. So, and the last slide. So the model seems satisfactory, reproduces the mechanism of changes of pelagic and benthic biogeochemistry due to injection of fish farm waste. Model allows to analyze consequences of the fish farm and predict volume of water affected, area of the bottom affected, extreme concentration of substances in relation to the injection of waste, changes in redox-dependent transformation of pollutants. So potentially, for example, when we have suboxic conditions, we should have efflux of mercury and formation of methyl mercury. In other tasks, we made this model also, and actually it can be added if it is needed. So, and it means that the modeling can be an effective instrument of estimating of carrying capacity of the water bodies. And what's about this conference? So model demonstrates that all major organic matter decay processes can be involved from oxic mineralization to sulfate reduction. The study scenario is, let's say, minimum impact, because usually people make farms in the same location several times with a break of 1 year. And as we showed, there are changes in the bottom that can be accumulated in future. Of course, we don't know this connected with man. Deoxygenation is an important point of view compared with other processes. But we can say that for at least at local scale, fish farming is an important factor of eutrophication and deoxygenation. First of all, for sediment water interface and in the water column. So this description of the model on this work is published, and we have a site in GitHub where you can the software and also copies of these papers. Of course, there were many projects involved in this work. Thank you.
Thank you, Egveni. It's interesting to introduce the fish farms in the modeling as a very efficient tool. We have time for a quick question.
Oh, thank you. I make it quick. Your model is so complicated with a lot of constituents interacting with each other, and there's a lot of exchange rates, basically. How do you get them? And how do they change the model results if you have them differently?
If you mean a biogeochemical model, so first it was made for the Black Sea, and probably that's the best place where a lot of measurements of, let's say, rare measured substances was made. For example, thiosulfate and elemental sulfur, they are present in water in 10 micromolar concentration, and they react the same stoichiometry as, for example, ammonia. So this just not included in majority of the model think of oxygen. That is why model underestimate oxygen depletion. So all of, so probably there's some kind, let's say, of experience. Also, for example, this sulfur species was measured in the Gotland Deep. So if we don't, if we cannot measure this process very frequently, it doesn't mean that it doesn't exist. So I had to put them in the model if we want to calculate oxygen precisely.
Thank you, Egveni. And I'm sorry, we're running a bit late. So our next speaker is Subhadeep Rakshit, and he will be talking about quantifying oxygen dynamics in a seasonally hypoxic fjord using a couple benthic pelagic model.
I'll leave it here. Okay. Great. Okay, I'll just take it here. Okay, hold on. Hello, everybody. It's a privilege to be here in this grand hall. Actually, it's a wonderful place. Also, I'd like to thank the the conference organizing committee for giving me this opportunity, and also thank you in general, because this is, I think, one of the first conferences, like in-person conferences after pandemic for all of us. And it's a nice change from the a lot of online events. So thank you. I am a PhD candidate at the Dalhousie University, and currently I'm in my final year. And today I will talk about a part of my PhD thesis, which is on one of the part is that I will be talking about today is quantifying oxygen dynamics in a seasonally hypoxic coastal basin using a benthic pelagic coupled model. So this figure is back again. So as we all know that the hypoxic regions in this world are increasing, and more so in the coastal regions. And in coastal regions, as the water column is shallow, the sediment becomes a very important part of this controlling this oxygen dynamics. So which brings me to my objective, where we try to understand the oxygen resupply and consumption processes in both water column and sediment. So my study is based on, or like mostly focused on Bedford Basin, which is a fjord and located on the northwest Atlantic. Here you can see this is the fjord, and this is the city of Halifax, where Dalhousie University is, where I'm from. And you can see this, this basin is just our neighbor, so we can go anytime there and do measurements. So if we just take a cross section of this fjord in details, we can see this is actually it's a fjord. So it's a 70-meter deep fjord, which is connected to the Scotian Shelf this side through a 20-meter deep sill, which restricts the bottom 50 meter, which is from 20 to 70 meter water. This water. So due to the presence of the sill, this water becomes a little bit more isolated from the bottom, the top disturbances. And for that, this basin stays relatively stratified for most of the time in the year, except in winter. This becomes interesting again because this site is also the location of a long-term time series, where as a part of a monitoring program, where they are collecting weekly CTD and water column Niskin bottle data for the last 20 years in a weekly resolution. So this is a very well understood in terms of the the dynamics of the water column, what is happening there. What is left in this place is the contribution of the sediment, where not a lot of work has been done yet. So that's where I come in, and through my PhD I try to resolve this, what's the contribution of the sediment in controlling the different dynamics of this water column. So today we'll be focusing definitely on oxygen. Well, nitrogen is fine, like anyway. So on oxygen, because this is oxygen conference. So what we do, we go to the field, definitely. We collect a lot of sediment core, and this we did seasonally in 2019. And we bring the core into the lab and measure the oxygen microprofile and through a microsensor, and measure the diffusive oxygen uptake from that, and also determine the oxygen respiration, sediment oxygen respiration rate from those microprofiles. So we also for a part of this study, we deployed a benthic instrument pod that we call it this one. So this records the CTD data at the bottom water at minute resolution. Why we are doing so, we'll come into it like a little later. So we have this, for a period of time in 2018 fall, we had this instrument which recorded the temperature, salinity, and oxygen data from the bottom water at minute resolution for 4 months. Okay, and we use a lot of this, the data to constrain our models. So to go into more details, first I would like to show you the climatology of this long time series. So just to orient you with this figure, so this is here you can see the climatology of the bottom water of oxygen, temperature, and salinity. And on the top panel, you can see the climatology of the sea surface temperature and this water column stratification. So you can see that here the oxygen is increasing during the winter months, here and this happens due to winter mixing. And you can see that during this time, you can see from the sea surface temperature, it is at its lowest. Also, the stratification index of the water column is also at its lowest. And during this mixing, you can see the whole water column mixes where surface water becomes mixing with the bottom water. And so you can see there is a drop in temperature as well, and also there is a drop in salinity, as it's a fresher and colder water is mixing. And also here you can see there is a little bit more variability towards the end of the year during this time. So this happens due to intrusion. So the intrusions in Bedford Basin happens. I'll go to this one. So this is a very quickly made cartoon. So the intrusion generally in Bedford Basin happens due to the coast of intrusion of the Scotian self water, which are denser and is more oxygenated generally. During so this happens in the fall, as you can saw here. So where there is a nice drawdown of oxygen because there is no supply of oxygen from there. We'll come to it later. And so you can see when these intrusions comes in late in the year, it brings oxygenated water, and as this is dense, it replaces the bottom water and kind of sits there, which we can see in our CTD profiles. But this event is not as regular as the winter mixing. So this is an infrequent event. So in this study, we focus on 3 different years. So so first is 2018, where you can see there was a winter mixing and there were 2 intrusions. So this is the time when we also, what happened during this intrusion, you can see this is sudden jump of temperature and a sudden jump of salinity. So this data is from week interval, like in every week. So you can see this jump, okay. So now, as I mentioned, that in 2018 we luckily deployed this this instrument during the period of time when the intrusion happened, and we could very luckily actually were able to capture these intrusions happening in real time. So as you can see, so these data points are in a minute interval. So you can see within a minute, within a matter of hour, the water property changes dramatically. Like here, you can see the oxygen concentration almost increases from 0 to almost how much? Like 150 or 200 or something like that. And similarly, temperature and salinity also changes. Okay, so going to the next year. So the next year of our study is 2019, where you can see there was a long winter mixing during this time when, in the green, and you can see there was no intrusion happened. And also in 2020-21, you can see that the winter mixing seems to be very low, probably the weakest winter mixing in the whole 20 years of time series. Also, there was no intrusion. And in all of these years, in the winter it was oxygenated, and towards the end of the year, towards the end of the season or the year, it entered the the hypoxia. So the seasonal hypoxia was, getting generated. So now we, designed 1D, the water column sediment coupled reactive transport model. So this is our, the model domain, which, extends from 20 meter to 70 meter of the water column and 0 centimeter to, the 10 centimeter of the sediment. So I mean, as this the below seal depth water is stable and is is not that influenced by these disturbances of the top layers, we can assume this is only there is no horizontal advection, and we can express this in 1D. So here you can see these 2 terms. This is the transport term, and this is the consumption term. So this is the KJ, which is the ED diffusion. So the ED diffusion is high during the winter when the oxygenation happens in the bottom water, and in the summertime when the stratification increases, it becomes it is very low, where during which time this term dominates when the net consumption of the oxygen happens in the bottom water. Similarly, in sediment, we model this using just a normal standard diffusive and reactive reaction. And here this is a constant which we can determine from our microprofiles. Here we know all these values. This is determined, empirically plus from the literature. And there is one unknown parameter, which is this one, which we will determine later. We'll come to it. It's basically by model fit. So here are the microsensor profilings. As you can see, in May in May, the oxygen concentration was very high, and there was a the big deeper oxygen penetration. Oxygen penetration was deep, and in other times the oxygen penetration was shallow. So when we determined the diffusive oxygen uptake from these profiles, we saw there was a significant correlation of diffusive oxygen uptake with the bottom water. However, no such relation we could see with the volumetric oxygen consumption in the sediment, which is good for us because we can then assume this is a constant, and we can apply in our model. So that K in the water column that I talked about. So to get that, we've model fitted our data, our model with the 2019 data, which we kind of consider normal because there was no intrusion and there was a kind of a normal winter mixing, which you can see. So okay, let me explain this figure first. So this shows the dots shows the observed oxygen concentration at different depths, and the line is our model output. So our model well predicts the data, and then we applied that particular rate constant for the other 2 years as well, and we can see that for all 3 years, it's actually our model could generate this seasonal change in oxygen concentration very well. Also, the sediment fluxes. So now, as our model is working, now we can use this model to understand some budgets of this oxygen, which brings me to my next slide, where this is in this plot you can see the relative contribution of different processes in supplying oxygen in the bottom waters. So you can see, even though the winter mixing lasts only for a few months, it actually supplies majority of the oxygen to the bottom water. Intrusions, on the other hand, even though they only last for a even though they happen infrequently, but they could be as important as winter mixing in terms of oxygen supply. Now, when we look at the absolute term, we can see that 2021 had almost 50% less oxygen supplied to the bottom waters than the other 2 years. I actually forgot to show you one thing, which I'll have to go back. Oops, sorry. Don't want to show this one, sorry. Here. So just to bring for a note that if you see the sea surface temperature of 2021, it was kind of towards the higher end of the whole climatology. Okay. Now, so which kind of explains that this might be due to that. And we also found that the sediment contribution of the total sediment actually contributed around 20% of the total oxygen uptake in our basin. So some of the takeaway points that I really want to point out is that winter mixing plays a very important role in this oxygen supply in the bottom waters of this kind of restricted basins, and these are very sensitive to the sea surface temperature. And a little bit change in the sea surface temperature might have a drastic effect, which actually happens in this Bedford Basin. So in this 20 years of climatology, we can see that the mean sea surface temperature is increasing very very fast. The trend is 0.1 degrees Celsius per year. And also, if you see the 2021, which sits at the very top of this trend, so it can have like a drastic effect, and it can reduce the oxygen supply almost by 50%, which is a lot. And this also echoes the a lot of talks that we have here. Heard from since the morning, that sea surface temperature plays a great role in supplying bottom oxygen towards the deeper waters. So next, I would like to point out that this kind of models can easily be upgraded to incorporate different other biogeochemical processes, which could be useful in monitoring and to understand different kind of budget kind of thing, you know, all those kind of stuff. So which actually I incorporate in my PhD thesis as well, where I incorporate like more complicated reactive transport models such as, you know, sediment diagenesis and all to couple the water column and sediment processes really well. Last but not the least, I would like to emphasize on the, you know, the importance of high resolution time series data, except which we can't know that if our current world is changing compared to the, the normal previous world. With that, I would like to thank you, Solus, for providing me the travel grant, which made my travel to Liège possible, and BIO for maintaining the time series is very important. And well, thank you. With that, I would like to, by the way, I am finishing hopefully by this year. So in case anybody has a position, so feel free to contact me, or like I'd be happy to discuss opportunities. Okay. All right,
great conclusion. Time for one quick question.
Okay.
If any. Vincent.
Yes, thanks a lot for your nice talk. I was wondering, because you have a very simple model, did you have to play a lot with the parameters? Or did you keep the same set of parameters for all the different years?
So that's a good point. So the main parameter that we changed is wintertime KZ, like the ED diffusibility. So we try to, as we don't know that what exactly is the mixing, so we play with that parameter to match the bottom water oxygen level. And that's the only thing we kind of change. But for the stratification time, we keep it a constant for 2018 and 19 when there was a normal one, but for 2021 we had to reduce it by almost by 70%, because it was a very warmer year, so we thought that maybe that's a good idea to do it. And with that we could predict, we could see the model matched with the observation, otherwise we were not able to get it. So it's kind of fits the story. We thought that, well, it's fine then.
Well, thank you, Subhadeep. No, I'm afraid we have to move on, but okay. You have the dinner to ask questions, no worry. So we move to Andrew, who was patient enough. Hi. And Andrew is going to talk about his high resolution atlas of the Eastern Tropical Pacific oxygen deficient. So the floor is yours.
Thank you, Veronique. Thank you, Marie-Laure. Thank you, everyone who organized this wonderful conference, the technical team, and thank all of you in advance for listening to me ramble on and on about oxygen deficient zones before dinner. And so I'm going to be talking about big ocean, these ODZs, as I will talk about, as I'll mention over and over again, and get us away from sediments. Sorry for everyone who is hoping for another sediment talk or more models, because I'm not doing anything with a model, although you could make a claim that any interpolation is a model. So we'll see. I ramble on and on, but I have to thank two very amazing students, Jarek and Timur. This work is mostly done by Jarek, but I will highlight something that we're going in a direction that Timur is taking on at the end. And so on the right, you see a map of oxygen saturation at a depth of 200 meters in the ocean. You can see there are large areas that are blue, that is, they're very low in oxygen. These are the oxygen deficient zones of the Eastern Tropical North Pacific off of Mexico, the Eastern Tropical South Pacific off of Chile and Peru, and then the Northern Indian Ocean, the Arabian Sea on the left, the Bay of Bengal on the right. And these are where you end up having very, very low oxygen concentrations. On the left-hand side, I'm showing a vertical profile of oxygen. This one's actually taken off of Hawaii in the central North Pacific Gyre. And you can see a characteristic profile where you have saturated waters in the surface, they're in equilibrium with the atmosphere. Oxygen gets drawn down with respiration. It reaches some minimum value before the biological consumption of oxygen is now outpaced by the physical ventilation laterally of cold polar waters that are coming in to ventilate the water. This is what I would call an oxygen minimum zone. And the oxygen concentrations here are 10s to 20s of micromolar in concentration. This is well above the threshold you would need to actually start turning on some of these anaerobic microbial metabolisms. In contrast, if we were to move a couple thousand kilometers eastward in the basin, you'll see the Eastern Tropical North Pacific. Hopefully, there we go. You see a similar profile, saturated in the surface, but the oxygen concentration gets drawn down much, much more quickly, much closer to the surface. And this is because these are zones of very high productivity and very sluggish ventilation. And so you end up with this swath of water here. I'm showing about a few hundred meters in thickness, where there's very, very low oxygen concentrations. And these are the zones that I would call an oxygen deficient zone. Although Don mentioned yesterday, he used the term anoxic OMZ, and that's fine too. Other people have called them nitrite accumulation areas because there's a correspondence of when you have very, very low oxygen and the accumulation of nitrite. But in essence, these are the same thing. In essence, they are areas of the ocean where the oxygen concentrations are low enough that processes like denitrification, anammox, dissimilatory nitrite reduction to ammonium can take place. I'm now showing a map. This is older than the one that people have been showing throughout this conference by Strom et al. In 2008, where I'm just showing that these areas are on this about 10 micromoles per kilogram contour. But that, as Don mentioned yesterday, is somewhat artificial. This is kind of the limit of our ability to detect with conventional means, meaning sampling water from meniscin, doing a Winkler titration, or even with some of these standard Clark type electrode sensors. I also changed the presentation at the last second because everyone else was showing this. And I wanted to prove that I'm not a Luddite. But in essence, Breitberg et al. Shows a very similar thing. This 0.07 milligrams per liter oxygen translates to about 2 micromoles per liter. And that is pretty much the limit of our ability to detect oxygen concentrations without a Stock's electrode or one of these optode sensors. You also can see from this figure, if you were to look in the Eastern Tropical, very sparse points. And that is a key feature that we're trying to overcome in trying to understand what do these zones really look like. We care because the oxygen concentration is in fact very hard definition. There are a number of studies that have attempted to define a threshold on the basis of what the measurements themselves say. But we want to get away from that definition simply because we're really looking for areas where the gradient with respect to depth, and I'll get back to this in a little bit, collapses to zero. And so here's a typical profile that we'll see. This is taken off of Mexico. In the gray line is the continuous trace from the CTD sensor. This is a Seabird 43 sensor for those who care. And then the green dots are showing where we triggered a bottle closed. So there's 24 of these because we had a rosette of 24 bottles. And you can see that, yes, the bottles more or less capture this continuous trace fairly well. But when you zoom in on the oxycline, that area at the top around 50 to 100 meters of very steep gradient, you can see that you don't perfectly capture this extraordinary feature very well. And that might be very important if you care about the migration of fish or the habitat or any of these things that define this very steep layer, which is tens of meters thick, and you have a gradient from saturation to zero across that. There's a further issue that comes into play where a lot of the gridding products tend to obscure these sharp features more because you tend to regress to a mean value. The extrema get reduced in size. And that really means that the minimum oxygen concentration you might see in a gridded data product is slightly higher than it would be if you were to measure at that point in space and time. And so we decided to change the strategy a little bit and look at all of the CTD casts and Argo profiles that we could get our hands on from the area and make use of the more continuous data that get reported back at, say, 24 hertz or so, in order to try to resolve better this steep gradient at the top of the oxygen deficient zone, as well as the swath of water that has no oxygen. And so here I'm just showing a typical sensor profile. The seabird is in the coral color, and the STOX electrode, this is thanks to Emilio Garcia Robledo, who spoke beautifully yesterday. The STOX is more or less the true value, about 0. So somewhere around 2 to 10 nanomoles per liter. And you can see that the two sensors more or less collapse on top of each other at a few micromoles per kilogram, something like 5 to 10. But it's at this very low limit where there's a bit of an offset. And that has to do with calibrations. It has to do with the fact that the sensors are not necessarily the best at detecting very, very low levels, but they are beautifully precise, if not accurate. And so this one happened to have a minimum of around 1 micromole per kilogram. That's not a real value. I will just mention, we've been on a number of cruises where that value might be 5 to -6 on one of the cruises. And that just shows that the variability that exists in how you start calibrating these electrodes. But that's only an offset in the absolute concentration. You can see that that gradient is still very, very steep. That is, if I were to take the derivative with respect to depth, you get pretty much 0. And that's what we're looking for in defining an oxygen deficient zone. And these are in fact crucial for climate and sustainability. These are the regions where these reductive nitrogen metabolisms take place, like denitrification, Anammox, DNRA. You can see this in global data products as a nitrate deficit. That's all of these very purple areas of the Eastern Tropical North South Pacific, the Arabian Sea. So this is what in fact limits lots of the fertility of the global ocean. In addition, these are zones of nitrous oxide production. So that's what I'm showing here. Everywhere you see red is an area where you tend to release nitrous oxide to the atmosphere. And since N2O is a very potent greenhouse gas and an agent of stratospheric ozone loss, this is very important climatically as well. There's also a debate as to whether or not the ODZs will expand due to the fact that temperatures are warming and oxygen is less soluble, or they might contract due to the fact that temperature is warming and the surface waters will become more stratified. As waters become more stratified, you can curtail the upwelling that drives the intense productivity, which might reduce the biological oxygen demand in the interior, which could actually lead the oxygen deficient zones to contract. And so we don't really have a perfect idea of what the sign change will be of oxygen in these oxygen deficient zones. So we may as well figure out what the base case is now in order to better monitor and predict going forward. And so let's try to do that. And that's what Jarek and I did in this paper that was published a few months ago. I encourage you to check it out, utilize the data as much as you like. But what Jarek did was he took about 10,000 CTD profiles, 15,000 Argo profiles, something like 15 million measurements from this region, and looked through all of them for a place within the water column where the vertical gradient was close to 0. Once he did that, we were able to define this binary of, is it an oxygen deficient zone or not, rather than dealing with the absolute concentrations. And since this is a lot of data across 20 years, a lot of the grid cells were visited multiple times, and we were able to define a parameter known as the fraction of oxygen deficient zone, or the fraction of times that a sample was taken that it was identified as an ODZ. We then mapped this data set in a 1/2 degree grid resolution, either in depth space of 20 meters or in density space of 0.1 sigma theta units. And now this product is available for anyone who would like to use it. And you can see some interesting features like the fact that the Eastern Tropical North Pacific is a bit shallower than the Eastern Tropical South Pacific. It's also thicker. What's really neat is we're able to come up with a pretty good map of what we would say the oxygen deficient zones look like. And so indeed, the Eastern Tropical South Pacific is much narrower in space, and it's also shifted shallower, even if the top is a bit deeper, whereas the ETNP is much, much wider. We were able to compare volume estimates, and I will not dwell too much on a slide that has a big table on it, but just to say that our volumes are very, very much the same as the volume estimates that have been calculated previously. The only difference is that we didn't have to make a determination on the basis of the data as to whether we were using a 20 micromole per kilogram cutoff, 5 micromole per kilogram cutoff. Rather, we just use this vertical derivative. And by doing this, it actually reduces the error uncertainty in the volume of these zones substantially. And so if we look at some of the data, this is a meridional perspective of the Eastern Tropical Pacific, the Eastern Tropical South Pacific on the left, the North Pacific on the right. You can see that the ETNP is reading out these fractions ODZ of close to 1. That is, the water is permanently anoxic. Whereas the Eastern Tropical South Pacific has a bullseye in the middle where you have no oxygen pretty much ever, but a lot of this transient anoxia around that. In addition, because we're looking at gradients, we can look for areas where there's a little intrusion. That is, that there's some oxygen coming in laterally that is sandwiched between two ODZs. And so that we defined an intrusion, we can define an intrusion rate parameter. And then when we map that in a similar way, you can see that these zones are in fact ventilated as you might expect on the outside, so on the boundary. But closer to the equator, on the equatorward side of these zones at the surface, you have a huge amount of ventilation going on, of intruding water masses. And this we attribute to eddies that are coming in. This highlights the fact that we don't really have an idea of how these zones might change into the future, because the ventilation pathways are dominated in a lot of contexts by the eddies themselves. And I have no idea how those might change moving forward with climate change. That matters a lot because the intrusions themselves will start impacting the biogeochemistry of what's happening. Don talked a lot about this yesterday in regards to nitrite oxidation, as did Emilio, I should note. And we see the exact same thing. So here's just a number of profiles of nitrite in the top row, oxygen in the bottom row, from off of the Eastern Tropical North Pacific. I'll mention station 1 is at 14 degrees north latitude, and all the other ones are around 10 degrees north, so at the bottom edge of the Eastern Tropical North Pacific. And what you see is a whole lot of intruding oxygen water masses, which changes whether or not nitrite accumulates or not. And what's really cool is that the station 1, which we would identify as the core of the ODZ, has these triple nitrite maxima features. So this first one is a primary nitrite maximum, which I don't really want to talk about in this context. But then the second one and third one is what we would call the secondary nitrite maximum. But there's this minimum of nitrite, it's actually 0 in between. And if I were to zoom in really, really close on oxygen, you can see this blip of around 0.5 micromole per liter that was percolating into the ODZ at this point. And so if you have this oxygen coming in, that acts as a oxidant for nitrite to turn it back to nitrate, as we learned about yesterday. But it changes all of the chemistry that could happen, all of the microbiology. And so how these zones change into the future will have a dramatic effect on what the ocean will look like. So to summarize, this is, we think, the most accurate resolution map of the ODZs themselves, just on or off to date. We hope that this will help modelers to validate some of their model predictions, as well as identify the sign change of what's to come. And we implore anyone who's reporting back data to keep it at least within these zones at meter scale or better. And that's because it helps resolve the very, very strong gradients that exist within these regions. And so having just bottle data or having Argo data that's downscaled to 50 meters or so in resolution within this layer can be a little bit difficult to deal with in order to predict how the ODZs will change moving forward. Thank you very much for your time.
Thank you, Andrew. One quick question done, and then we'll move to the panel.
Thanks for a terrific talk, Andrew. That's really great. I'm really happy about how that's developing with trying to understand and define these OMZs. One thing that concerns me a little bit is I think it's really important the sensitivity of the sensor that you use. Take the Bay of Bengal as an example. If you had a sensor that you're using there with a sensitivity of about 100 nanomolar, you might actually decide that it was a real anoxic OMZ, but in fact it's not, and that would impact the relationship further onto the nitrogen cycle.
You're absolutely right that if in the Bay of Bengal, you also see minimal gradient with respect to depth, it's just a little bit higher. And that relates to the drawdown, whatever the oxygen concentration the organisms want to draw down to. And so in order to contextualize whether or not these anaerobic metabolisms are happening, you need additional data, things like nitrite, things like N-star. And so you're absolutely right in that regard.
Okay. Okay, thank you for all the speakers of the session. So we're going to get organized for the panels. So we have two of our panelists who are here and two are remote. So may I ask Fabien and Dacia to come here, and we have two, Eric Dupont and Stefan Raymond, who are online. So this panel is called Citizen Science. So in other words, how can we augment the observation for biological essential ocean variables, getting help for every sector? So our panel, so Marie-Laure and I will be trying to moderate that. So here we have Fabien Roquet. Can you just, hello. So he's a physical oceanographer as a background and is working in University of Gothenburg and is a co-chair of ANIBOS, which is an emerging network from GOOS with animal ocean-borne sensors. And we have Dacia that is a scientist. So she's a scientist of the panel and you heard. So she's a chemical oceanographer from the Ocean Frontier Institute at Dalhousie University in Canada. So online here, we have our two other panelists. So top one is Eric Dupont. So he's director of the Ecopole company, and he's also representing here the Ponant Science Program. And below, we have Stefan Raymond, who is a scientific advisor of the Ocean Race. So these companies are part of the UN Decade Program Odyssey, which was launched in February 22 about citizen science. So trying to get more observation from any part of the civil society. So companies, public sector, and so forth. So the way we're going to organize, maybe you can, we ask each of the panelists to give a few words of what their activities are for, I mean, Dacia, you already heard, but she has a couple of slides too. So maybe we'll start with Eric top there, and we'll switch to And then what we did with all the questions, the poll. So you kind of put your favorite questions. So after the very short presentation of each of the panelists, we'll ask them the question that you rank first, second, and hopefully we'll try to get them all. So Eric, the floor is yours. Quick, 2 minutes.
2 minutes, thank you, Veronique. Hi everybody, sorry to be on a remote area. It was a really stretch for me to be here with you today. I was involved in a big crisis, pollution crisis yesterday. So actually I was willing to talk about opportunity ships for ocean data. So as Veronique mentioned, I am working for Ponant company, and especially with the latest Commandant Charcot ship, and also founded a company named Ecopole for pollution and I would say coastal data. So next, you are switching my, okay, thank you. So actually for Ponant, they have some ships that are sailing in very remote areas and basically polar region where very few data are existing and where standard oceanographic ships have some difficulties to go. So next, we have on board science officers. That means that they are able to follow pretty stringent scientific protocol, and that's ensure the quality of the data. Next, please. And also on this very brand new ships, we have Ponant has invested some very cutting edge equipment, such as you all know the ferry box. So we have pH, dissolved oxygen, CO2, chlorophyll, of course the standard sea surface temperature. We have also very innovative. I don't know if you know it. It's a partial pressure CO2 measurement box that has been designed by a national General Oceanics, and it has been set up by the National Oceanographic Administration. We have a CTD, we have a microscope, we have also a freezer until -80 for biologic constraint. Then the next, we have also 13 ships. So the interesting fact here is that if we can set up all this equipment in all the different ships, we could build a consistent data network so that all the data are pretty consistent and can be used as an open source for any laboratory who would be interested in those data. Next. And then we have the Ecopole company. So we are operating pollution ships basically for hydrocarbon and plastic pollution. But actually we have instrumented these small ships with scientific sensors, such as, of course, the pH, oxygen, and so forth. And the skipper basically are scientific or have an oceanographic background. So that, again, that just ensure that the data are collected with good scientific protocol. This is a small ship, but the interesting here is that we can put it a lot of different instruments. We have even CTDs that can go to 100 meters depth. So next. So as a conclusion, I would say that the Ponant ships, obviously the Charcot, is open for data for any different laboratories that would be interested in getting data or conducting or operating a scientific project in remote areas, especially where the ship is going in North Pole or Antarctica. And the next, this is for Ponant. And for Ecopole, you could have specific studies. Basically, the ships are located in South Mediterranean because this is the most polluted sea. And so we are open to welcome any laboratory or to provide any data that would help to calibrate your model for a coastal model. that's pretty much it. Thanks very much for your attention.
Thank you, Eric. And we'll move to Stefan. Can you say a few words? Your PowerPoint is on the screen.
Yes, thank you very much. Thank you very much to have the opportunity here to speak. I will make it quick. So next slide, please. The Ocean Race is a race event which takes place every 3 to 4 years with sailing boats racing around the world. Next year we will start in Alicante in Spain. We will have multiple stops like in Cape Verde Islands in South Africa, then we sail straight to Brazil, then north to the US, and then we cross the Atlantic back, having stops in multiple countries in Europe. Within this Ocean Race, we have a team, a dedicated team, what you see here on the slide, and I'm responsible for the science program. Next slide, please. The science program consists of some packages on board of the IMOCA class, which is a subclass during the race. We will have small ferry boxes. So something what you saw on the Ponant, but much smaller. We talk here about an instrument with a weight of 15 kilograms. With dimensions of 80 centimeters times 25 times 50 centimeters. So it's very compact, very lightweight, and which can be deployed on this high-speed sailing boats. We measure basically the same parameters what you have on this big instruments. So we have an instrument measuring sea surface temperature, sea surface salinity, PCO2. We will have a sensor for measuring oxygen. We can have also chlorophyll A and other pigments measured. And on the VO65 boats, we will have 2 samplers to measure or to sample microplastics. Also on all boats, we will submit data from the boat itself, weather data, which will help to do better weather predictions. Next slide, please. So all boats or racing boats can be part of this global approach to obtain data in remote areas. And as mentioned before, our aim is to measure PCO2, salinity, temperature, oxygen, and other parameters. Next slide, please. Here you see an example what we did between the races. So last year we had a couple of race events in Europe, and each time when the boats went out, they gathered data, if you like, for free, because you don't have to pay any research vessel or something like this. The boats are out there anyway, and they come back with this high quality data. So you see here race events taking place in the Baltic Sea, then in Mediterranean Sea, and for sure also when the transit from the Mediterranean Sea to the Baltic Sea, we got data. So here in this case, we collected PCO2 data, but also we have the other parameters. Next slide, please. And I think that's the end. So we will start the race January next year, have the first stop after Alicante in Cape Verde Islands, and then sail to South Africa. Yeah, thank you very much.
Stefan, for keeping us up to date of what's going on with the Ocean Race. So maybe now we'll have Dacia. We can navigate your slide here, so you don't have to.
I have just 2 slides because I think it took my time this morning. So I'd like to give a short update on the status of biogeochemical observations on ocean sites. Ocean sites is the sites of permanently deployed moorings. And as you can see from the statistics, still there is really very little of biogeochemical sensors that have been deployed there. Sensors for salinity, temperature are more abundant, but sensors, especially for the carbonate system and nutrients, are not there yet. And I would say that the technological development is what's limiting deployment
of these sensors on the platform. So next slide, please. And the limitations of the sensor technology is, of course, its size and power consumption. You can imagine that especially mobile platforms cannot accommodate large analyzers. And if you want to measure nutrients, for example, that's what you're looking for. And the carbonate system itself. Also, there is a lot of questions towards accuracy and stability of the sensors, but this is a work under development, and sensor companies together with the scientists are doing enormous amount of work validating and the sensors and bringing sensors to a point where they can be commercialized and being sold. So that's where I see contribution that sensor companies and academia doing together through this tight collaboration. And just to give you a couple of examples of the sensor development that's been done over the last, I would say, decade or so. Optode technology has been progressing really well. And you all know about the oxygen optode sensors, but now we have even a pH optode sensor from the company Pyroscience has been displaying their products even here at the conference and sponsoring this event. And then one of the most promising developments that I see is that the development of these miniaturized analyzers, a couple of companies, well, two, not a couple, two, there are only two that are doing using this technology. So basically packaging an analyzer that we would normally use in the lab into a small volume. Power consumption is low. They can be deployed even on the gliders. I'm not even talking about the Morins here. And the example of the sensors where we find a new way of measuring something. Instead of packaging analyzer, nutrient analyzer in a can, we may use a fluorometer such as nitrate fluorometer that's being used in the sensors like Satlantic's ISOS or Trias Opus sensor measuring nitrate and DOM as a byproduct. Just to summarize that, that the expansion of our observing network relies heavily on the progress in sensor technology, which in turn relies a lot on the sensor companies and industry collaborating with the scientists on proving the sensors for further use. That's about it.
Thank you, Dacia. Fabien now. We'll say a few words at Anibos.
Hello, yeah, my name is Fabien Roquet, professor of physical oceanography at the University of Gothenburg. And I'm here invited, thank you very much for that, because I am chairing Anibos, the Animal-Borne Ocean Sensor Network, which is a GOOSE network. And maybe I need some slides so I can show. But the transition would be very smooth. Yeah, so I will just talk. So Anibos just was created in 2020, but it is the result of almost 20 years of work now, research, a very fruitful collaboration between biologists and physical oceanographers. So using instruments that we put directly on animals, and in particular, we used a lot elephant seals, different other species of seals, and now more sea turtles. We've been able to gather a very large amount of data in the Southern Ocean, in some tropical regions, and in the North Pacific. And more recently, we've also used oxygen sensors. So that's also why, oh, wow. So here is what I was talking about. So here you can see some Weddell seals, and one of them has satellite relay data logger on the head, ready to send us some very interesting data. Can you change the slide? Yeah. So the vision of Anibos to enhance understanding, describe our changing ocean. through the eyes of marine animals. So Anibos is here to try to coordinate these activities internationally and to help foster more work, more activities. You can see a map that represents all the CTD profiles that are currently available worldwide. So you can see it's mostly in polar regions right now, especially in the Southern Ocean. Has been extremely powerful. It is right now already the main source of CTD data south of 60 degrees south. So it's a very important source of data. Yeah, please. Here, yeah, just to see, it's quite recent, but now there's been an oxygen sensor has been developed, so it's been tested. This is a work that's done by Christophe Guinet at Chizé in France, and this is very promising. Can you? Yeah, and that's a summary. Yeah, Anibos, we hope will help to develop this further. Yeah, and I will stop here. Thank you very much.
Okay, thank you, Fabien. So I'm going to ask to each of the panelists at least a couple of the questions that were ranked by priority. And of course, the one that comes into mind right away is what about companies' contribution to science? And there is a big debate in France about that. How to avoid a potential perception of greenwashing. So Amai has the question to Stefan, and I will ask to each of you.
Yeah, so I would reply to these questions by contact questions. What should we not do? Why we should not do this? I mean, we're sailing anyway in these areas. There's no extra carbon footprint when doing this kind of observations. And my question would be on the scientists asking those questions. What is your footprint when you start a campaign, let's say in Southern Ocean, starting from Chile? So you fly in a complete scientific crew to Chile, going on board on a research vessel, using or burning lots of fuel. We use our sails, so our boats will go anyway. That's one thing. The second thing is with our outreach due to the sports event, we can communicate with the public in a very different way. So we can transmit the message much more effectively than maybe a scientist can do. And maybe in the end, the company looks a little bit greener, but why not? I mean, it's a nice way to get data.
Sure, I fully agree. Eric, your point of view.
So you have to unmute.
You're muted. You're still muted.
Yeah, this question is really key. It has been a very long debate, even internally in Ponant at least, because Ponant is more well known for luxury cruise and scientific ships. But actually, I am not from Ponant initially. I am a science from the French Geographic Survey, the CHOM. And when I joined Ponant, I was a little bit skeptical about that science program. But now I'm working for them. I done at least 3 months on board. I did a lot of science project, and I saw that there is a real will of Ponant management to help science and to help scientists to get good science data. Definitely the equipment that are on board are really at cutting edge equipment. They invest something like 300K last month just in a ferry box, CTD, microscope, freezer, a lot of different equipment. We have at least 150 square meter of laboratory, and we are welcoming to 4 scientists per cruise. So obviously, maybe at the initial funding of this program, that was could be a greenwashing or a justification to say, well, we are going in remote areas to do science, so we have the right to do it. Maybe, frankly speaking, I would say that could be a point. But today, in 2022, I think it's real good science that we can offer to science. And it would be a pity not to take advantage of these ships, because the infrastructure on board are really good for your model, for the lab, and for the whole ocean understanding. So maybe just to show you something, okay, I cannot share my screen, but we have in 2022, 33 science projects on the Charcot ships, and we are welcoming 84 scientists on board. And I would say most of them, not to say 100% of them, are really happy to be able to be on board. Obviously, there is, you know, I've got some feedback from science lab, and they told me that they were so happy to be on Charcot. I would say there is no antinomic between to be in a comfortable ship and to do good science. We can do both, and definitely Charcot is really a great ship for that.
Thank you, Eric, for your point of view. And Fabien, so now the scientist, what do you think about that question? How do you feel about that?
So the question is about greenwashing?
Yeah.
Yeah, I think it's clearly true. I mean, every scientist believe that they are here to do greenwashing. I think there's no question about that. Is it useful then for science? I think that's an open question. It's not proven yet, but maybe, I don't know. But I have my doubts, but maybe in the long term, we'll see.
Okay, that's interesting. Dacha.
From our experience working with the big shipping companies and others, of course, there is that component of, you know, we want to look green, we want to, or our corporate tell us to be green. But from communication with people who actually run the ships and people who work on the ships, there is a genuine interest in making or being a part of the solution and helping as much as they can. So I think there is a goodwill. I have questions about execution, so I will count Fabien on that.
Yeah, I think we're learning how to really work together. And so maybe the beginning will be a bit hectic, but I think by the end, it's another possibility to increase the number of observations. So that's my point of view. Okay.
Yeah, just a quick point, Veronique. If you think about the weather or the atmosphere study, you know that a lot of airplane or air flight that are, you know, getting tourists in Maldives or whatever, they are using the flight to get some data, and nobody is blaming Air France or KLM or Qatar Airways to give some data about the atmosphere. It's kind of the same thing, I think. What about the ocean? Why we cannot use ships and commercial ships to get science if the data are really clear and clean and acquired following a clear scientific protocol? Why not?
Sure. Now, may I ask some people of the audience to give their opinion on that question about, you know, the company's contribution could be perceived as greenwashing? Any volunteer? It's been a long day, huh? Okay, we'll just go for another question about what you think. So I'll start with you, Fabien. What is the contribution? What do you think could be the contribution of the civil society to stop the decline of the ocean health?
Yeah, I think the most important thing is that people vote, especially the young generation is not voting enough, and they should vote, because in the end, that's all that matters. And if we can get politicians that are more caring about the ocean, of course, that would be probably the best way to improve this decline. Now, yeah, there's very deep underlying cause to this decline of the ocean health. This is a very complicated issue. So yeah, I am not sure there is in the end very much we can do. We can observe. As scientists, we certainly observe it. We can be hopeful that something will come. But yeah, I would insist that as far as I can tell, it's only democracies that care about environment nowadays. And so, yeah, vote.
Okay, got the message, Dacha?
I'll take over. In order, I'll just comment. In order for this democracies to care,
get closer.
In order for the democracies to care about climate, they they need to care about no war to start with. So that's just my first point here. And the second one is I see our role as a community, not only in collecting more observations, doing more observations, but also to effectively communicate science with the society, with the stakeholders. For that, I think, judging from experience, we all lack in these communication skills that I think are needed in the modern society in order to be successful and impactful, where the problems of deoxygenation, ocean acidification, and other related issues are well articulated and being find the listener and being actually comprehended, and then those people have to go and vote. So,
okay, so the message here is we should vote. So, what's your point of view for the contribution of the civil society to try to stop decline ocean health?
Well, definitely the civil society has a big influence on politics, and voting is a key part of this change. I would say there is a missing link between all your studies about this interesting oxygen and other type of key parameter of the ocean health and the civil society. For example, Ponant is welcoming 50,000 passengers per year. The fact that we are able to vulgarize, to speak about science on board as a very simple word to these passengers. Each time I am on board, I'm welcoming workshop and people who are coming and are very, very interested in all the science project. We call it citizen science, but it's even more than that, because they can touch, they can see what is a ferry box, what does that mean, an oxygen decay or an oxygen decrease, what does that mean, microplastic in the Antarctica, what does that mean, everything. And when they see that, definitely they are able to disseminate the message. They are able to really be aware and understand that this is the last chance for us to change the things for the ocean. It's a decade. And so I think above, there is a clear missing link between the pure academic science and the civil civilization or the civil population.
Thank you, Eric. Stefan?
I completely agree with Eric. So education and communication is really key here. I'm surprised how unclear for some people are the basic information, like the link between atmosphere and ocean. That when you have more pollution, more CO2 in the atmosphere, that your oceans get more acid. Those basic information which are clear for the scientific community are not each time or not so clear to lots of the population. So that's why the Ocean Race organize events in each port. So when we, for instance, sail to South Africa, we will have a race village and shows demonstrating those facts. We have beach cleanup events. And I think those things are very, very important to communicate with the audience.
Okay, thank you, Stefan. I'll ask a last, unless anybody wants to, Andreas, you want to give it?
Yeah, thanks very much. I think, well, voting is very important, but I don't think voting will save the planet. So politics is too slow and has too many make compromises all the way. So health system and social system, many very important things for most people who are voting today, I think. And the next generation will not vote. And so we, therefore, I think civil society and also companies are very important, so they can create new markets. They will see new markets and new opportunities, and that's maybe much faster than waiting for the political system. We have seen that, I think, in the food industry, that has changed quite a bit. At least parts. There's still old parts surviving, but when we go to the supermarket, we see organic food and fair trade coffee and many things which we didn't achieve by voting, but by the markets. And there I hope that talking or communicating, collaborating with industry, with companies is very important and is probably the only way that will move it fast enough. And it will include the big oil industry, coal industry, probably, since they can move big amounts of matter, of CO2, of whatsoever, and produce energy, which we all need. So it is painful, I think, but science then has the opportunity, the task to make it transparent. And that's, so we are paid by the taxpayers, and we have to make everything transparent, and thereby also contribute to avoid greenwashing, but allow companies to open new markets and take opportunities. And some of these may hopefully work, not all, but and that's again up to the scientists to really assess everything critically and transparently. Thanks. But we should still vote all the time.
Well, I think we will close up here because it has been a long day. And we would really like to warmly thank you for standing online. And sorry we were a bit late with the program. But I think it's very important that companies are, like Andreas was saying, are part of the game because we are all on the same planet. And I think we need to work together and try to convince scientists who are really into the greenwashing without, seeing that there are opportunities to be taken, and of course want us to be cautious. But I think it's a great opportunity that you offer. So personally, I do thank you, and I want to thank also Fabien and Dacia for being here after a long day of talks. And well, maybe we can upload everybody. Thank you. Thank you.